అయిందా?
అదే ఒకసారి బ్లూటూత్ ఎనేబుల్ చేసుకోవాలి. ఉండు నాది క్లోజ్ చేస్తాను. కొంచెం అదే ఇట్లా అట్లా చేసుకోండి.
యాడ్ డివైస్ అన్నాడు కదా.
బ్లూటూత్ అని ఉంటుంది చూడు,
అదే బ్లూటూత్
కనెక్టెడ్ అని వస్తుంది సార్.
ఆన్ ఉంది.
ఆన్ ఉందా? వాళ్ళు మాట్లాడేది నీకు వినపడుతుంది సార్.
చూస్తాను.
OnePlus అని వస్తుంది చూడు. ఇక్కడ చూడు,
అవునా?
ఆన్ చేయి.
OnePlus కనెక్టెడ్.
కనెక్ట్ అయ్యి చూడు 100.
నేనే ఇంకా ఎవరు జాయిన్ కల.
ಹಾ ಇದೆ ನೋಡಿ.
ಗೌಡ್ರೆ ಕೇಳಿಸ್ತಾ ಇದ್ಯಾ? ಮಂಜುನಾಥ್ ಗೌಡ ಕೇಳಿಸ್ತಾ ಇದ್ಯಾ?
ಕೇಳಿಸ್ತಿದೆ ಮೇಡಮ್, ಕೇಳಿಸ್ತಿದೆ.
ಓಕೆ ಓಕೆ ಸರಿ, ಥ್ಯಾಂಕ್ ಯು.
We are about to begin, and I'll request all of you to please keep your mobile phones on a silent or a vibration mode. Very good morning, one and all. Respected guests, distinguished dignitaries on the dais who have not come yet, but I'll be inviting them very soon. Esteemed scientists, researchers, academicians, industry partners, dear delegates, students, and friends, a very warm and hearty welcome to one and all. It gives us an immense pleasure to welcome you all to the 8th International Conference on Sustainable Agriculture for Food Security, being organized under the vision of Agri Vision 2026 in collaboration with SARM and ICAR, IIRR, CHEC, Bhubaneswar. At the time when the world is facing unprecedented challenges, climate changes, shrinking natural resources, population pressure, and food insecurity, the theme of this conference is just not relevant but very crucial. So, sustainable agriculture is no longer a choice. It is a shared responsibility towards ensuring food security, environmental balance, and the well being of future generation. This conference serves as a powerful platform where the science meets practice, where innovative ideas, research findings, and the field experience converge. Over the next few days, we look forward to meaningful discussions, knowledge exchange, and collaboration that will help shape resilient and sustainable agricultural systems. We are honored by the presence of eminent scientists, policymakers, industry leaders, and young researchers from across the country and abroad. Your participation truly enriches this forum and strengthens our collective mission. May this conference inspire new perspectives, foster partnership, and sow the seed for impactful solution in the sustainable agriculture. And now, I would request all of you to give a warm welcome to our dignitaries on the dais. I'll be requesting everybody to please give a good round of applause for Dr. Tushar Kanti Behra, Director ICAR IIHR Bangalore. We also have with us Director Horticulture Government of Odisha IS Dr. Gorak Kalungeji. We also have with us Dr. Ram Gopal Agrawal, Chairman Emirates Dhanuka Agritech Private Limited. We have with us Dr. Sain Das, former Director ICAR DMR. We have Shri Dibyanshu Prasad Das, Organizing Secretary AgriVision 2026. A good round of applause for all of them. I'll be requesting. And now, as it is said, that every humble beginning should be started by seeking the blessings of the Lord above, I'll be requesting all my dignitaries on the dais to please do the honor of lamp lighting first, and then they may take the chair. So, first lamp lighting. And ladies and gentlemen, as we say, this day is just not of theirs. It's of ours efforts that we will be making this AgriVision 2026 a successful one. So I'll be requesting all of you to please stand up from wherever you people are sitting and be a part of the same. A warm welcome to AgriVision 2026. Let's have a good round of applause for all of us, and let's chant his name first. Samaste misiki tike ujor swarare tike kohiba Jai Jagannath Swami ki Jai. Let's hope for a wonderful conference in the coming days. Thank you so much, everybody. You may take your seat now. Now, as we proceed, I'll be requesting our organizing secretary, AgriVision 2026, Shri Dibyanshu Prasad Das ji, to welcome the August gathering.
Jai Jagannath, distinguished guest on the dais, Dr. Tushar Kanti Behra, Honorable Director, ICAR IHR Bangalore, Dr. Ram Gopal Agarwal, Chairman Emeritus, Dhanuka Agritech Private Limited, Dr. Sai Das, former Director, ICAR DMR New Delhi, Dr. Dr. Gorok Kalunge, IAS, Honorable Director, Horticulture, Government of Odisha, eminent scientists, policymakers, fellow agriculturalists, and dear students and farmers, good morning to you all. It is a profound honor and a personal privilege to stand before such a distinguished gathering today. On behalf of the organizing committee, I extend a very warm and hearty welcome to each of you to AgriVision 2026, the 8th International Conference on Sustainable Agriculture for Food Security. As we convene here in 2026, the global agricultural landscape stands at a critical crossroad. We are no longer just talking about increasing yield. We are talking about the survival of our ecosystems and the resilience of our food system. The theme of this year's conference reflects our urgent reality, bridging the gap between traditional wisdom and cutting-edge technology to ensure no plate goes empty. This year, we are proud to host over 800 delegates from across India and abroad. We have with us visionary scientists, progressive farmers, industry leaders, young scholars from different institutes of India. Over the next 3 days, this conference will serve as a melting pot of ideas. We have curated a series of plenary sessions, technical workshops, expo, poster presentations, focusing on climate smart agriculture, precision farming, and circular bioeconomies. Our goal is simple yet ambitious: to move beyond academic discourse and generate actionable blueprints that can be implemented from the laboratory to the land. I'd like to express my deepest gratitude to our sponsors, collaborating universities, and the tireless numbers of organizing subcommittees. Your dedication is the reason we have gathered in this hall today. Most importantly, I thank our keynote speakers and delegates. You have traveled so far and wide, bringing with you the expertise that will make AgriVision 2026 a landmark event in the history of sustainable development. I would like to thank our sponsors and exhibitors, not in order: ICAR, IHR, Department of Biotechnology, NABARD, CRRI, ICR CRRI, CubeTech Scientific, Central Warehousing Corporation, Paradip Port Authority, Bayer, Dhanuka Agritech Private Limited, HURL, CRIPCO, OUAT, Central Institute of Women in Agriculture, Coconut Development Board, NFDB, PPVFRA, CIFA, National Seeds Corporation, ORMAS, CDA, BDA, CRUT, Odisha State Housing Board, Odisha Language, Literature and Culture Department, Indian Meteorological Department, NRC Lichi, Department of Veterinary and Animal Husbandry, Government of Odisha, Directorate of Horticulture, Government of Odisha, Housing and Urban Development Department, Government of Odisha. I hope the deliberation of this conference will be intellectual, stimulating, and personally rewarding. Let us use this platform to forge new partnership and renew our commitment to a food secure world. Once again, a very warm welcome to you all. I wish you a productive and memorable stay. Thank you.
Thank you so much, sir, for your warm welcome. I'll be requesting all of you to do a collective round of applause for all our sponsors and partners. And with that note, I'll move ahead with requesting Dr. Tushar Kanti Behra, Director ICAR IIHR Bangalore, to address this August gathering.
Jai Jagannath. Most respected the Chairman Emeritus Dhanuka Agritech, Dr. R.G. Agarwal Sahab, my teacher and very senior, I will say the former director of IC, Dr. Saida Sahab, my colleague and friend when we were studying together in IRI, Mr. Kalungi Sahab, Director Horticulture. The most important persons today, we have many are there, and also the organizing secretary for the AgriVision 2026, and the scientists, the stakeholders, and more Priya Krushaka Bhai or Bhoni. ମୁଁ ଜଷ୍ଟ ତାଙ୍କ ପାଇଁ ଫାଷ୍ଟ କହିବି, ସେମାନେ ହେଉଛନ୍ତି ଆମର ଯେଉଁ ଅନ୍ନଦାତା ଆମେ ଜାଣିଛୁ ଆଉ କହୁଛୁ, ସମସ୍ତଙ୍କ ପାଇଁ ମୋର ବହୁତ ବହୁତ ଧନ୍ୟବାଦ. The day we celebrate is AgriVision 2026, because last year also I was here. That time we have discussed many things about this program. I will not take so much of time. We are already 1 hour late. So we have to finish and in the time. So very important that our own many friends, many senior people are there. They should be stick to the time. Even they can cut it at least 10 minutes. If their speech, I should be more obliged and thankful to them, because they are seniors. So I cannot tell the things what we have. But as I mentioned, it is a wonderful to this Chase IHR. We are the organizations which is in Bangalore, but we are operating here with respect to horticulture and agriculture as a whole. And I know that many of the scientists, many of the deliberations towards the sustainability in agriculture. Sustainability science is very important because we have to sustain, not at the cost of ecology, environment, energy, water, and food. So these all things together, if you combine, this is reflected as a sustainability. Sustainability is not that to have that did the production, increase the productivity at the cost of the environment. You know that we are now facing a lot of problem. ଆମର ଚାଷୀ ଭାଇ ଭଉଣୀମାନେ ଏଠି ଅଛନ୍ତି, ସେମାନେ ବି ଜାଣିପାରୁନାହାନ୍ତି ଆମେ କ'ଣ ୟୁଜ୍ କରୁଛୁ, କ'ଣ ଆମେ ଏଇ କାମ ପାଇଁ ଏଗ୍ରିକଲଚର୍ ପାଇଁ ଆମେ ପ୍ରଡକ୍ସନ୍ ଖାଲି ଦେଖିବା କଥା ନୁହେଁ, ତା ସହିତ ଆମକୁ ଦେଖିବା ପାଇଁ ପଡ଼ିବ ଆମର ଏନ୍ଭାୟରମେଣ୍ଟ କେତେ ସୁନ୍ଦର ହୋଇଯାଇଛି. ଆମେ ଯେଉଁ ଆଗକୁ ଚିନ୍ତା କରିବା, ତାହେଲେ ଆମର ପିଲାମାନେ କ'ଣ କରିପାରିବେ, ତାଙ୍କ ପାଇଁ ବି ଚିନ୍ତା ଯଦି କରିବା, ଆମର ଯେଉଁ ମାଟିର ଉର୍ବରତା ଆମେ ଆସ୍ତେ ଆସ୍ତେ କ୍ଷୟ କରିଦେଉଛୁ, ତେଣୁ ତାକୁ କେମିତି ଆମେ ଉର୍ବରତାକୁ ବଢ଼ାଇ ପାରିବା, ସେଇଟା ହିଁ ମଧ୍ୟ ଆମ ପାଇଁ ବହୁତ ବହୁତ ଭଲ କାମ ହୋଇପାରିବ. କାହିଁ ଆମେ ନିଜ ପାଇଁ ବଞ୍ଚୁଛୁ, କିନ୍ତୁ ଆମେ ଚିନ୍ତା କରୁନେ ଆମର ପରବର୍ତ୍ତୀ ମୁହୂର୍ତ୍ତରେ ଯେଉଁମାନେ ଆସିବେ ତାଙ୍କ ପାଇଁ ଆମେ କ'ଣ କରିପାରୁଛୁ. So that is very good as a sustainability. Sustainability means ଆମେ ବଞ୍ଚିବୁ, ଅନ୍ୟମାନଙ୍କ ପାଇଁ ବଞ୍ଚିବା ପାଇଁ ଦେବୁ. ଆମ ସହିତ ଯେତିକି ଏନ୍ଭାୟରମେଣ୍ଟରେ କୀଟ କୀଡ଼ ପତଙ୍ଗ ଯେତିକି ଯେଉଁମାନେ ଅଛନ୍ତି, ଯାହା ବି ଅଛନ୍ତି, ସେମାନେ ପାଣି ବାୟୁ ସବୁ ସେହି ପ୍ରକାର ରହିବା ଦରକାର, ଯେଉଁଟା ଆମେ ଆଜି 50 ବର୍ଷ ପୂର୍ବରୁ ଦେଖି ଆସୁଥିଲୁ ବା କହି ଆସୁଥିଲୁ ବା କରି ଆସୁଥିଲୁ. ସେଇ ଜିନିଷଟାକୁ ଆମେ ସଷ୍ଟେନେବଲ୍ କରି ଯଦି ପାରିଲେ ତାହେଲେ ଆଗକୁ ଯାଇପାରିବା. ସୋ ଆଇ ୱିଲ୍ ନଟ୍ ଟେକ୍ ସୋ ମଚ୍ ଅଫ୍ ଟାଇମ୍. ଆଇ ଥିଙ୍କ we can have at least a few remarks from our co-chairperson. If you have just to have some just emails. So thank you very much. We'll start immediately because inauguration pore habo, so that will do.
Thank you so much, sir. Now we'll move ahead with the address of Director Horticulture, Government of Odisha, IS Dr. Gorak Kalungeji. Sir, can we hear some words from you?
Good morning to all. Our Director IIHR, Dr. Tushar Kanti Bera Mohoday, Sri R.G. Agrawal Chairman Dhanuka Group, Dr. Sain Das, ex-Director DMR, ICR India, and organizer, scientists, students, and farmers. So, this conference regarding the sustainability and food security. So, this sustainability word is a very buzzing word actually, everywhere that is being used. So, the question comes, why that word? Always sustainable. That means something is there, the system we are following, the production technology we are using, there is some issues. That's why we are using this sustainability. When Kharif comes, farmers start thinking what to sow. When it is harvested, again Rabi, again which variety, which crop, so every time. So that means the system of our food production is in question. Nutrition. So how they are giving the nutrition? So they will go to some agro service center. They will ask that fellow what to give. Whatever he recommends, he believes him. That fellow is believed. So accordingly, he applies that fertilizers. Liquid fertilizer, whatever it is. That may be urea, that may be potash. And how much is the requirement of the crop? Whatever the prescription given by universities, 150-50. It is very common in vegetable crops. Again for fruit crops. So farmers, those doing some experiments, they are following some standard practices. They keep on experimenting. You may be aware of Mr. Palekar. Subhash Palekar, when he saw the jungle growing, how these forest plants grow. No one is watering. No one is providing any fertilizer. No one taking any spray. So how come? And he questioned his professors in university, PDKV Akola. No one had answer. When productivity of his crop started declining, when he asked, why so? So that forced him to do some research. So that's how the farming, nowadays permaculture, natural farming, organic farming. And I will tell you, people are crazy actually for these organic produce. So authenticity is in question. So how to certify and all those things, it is organic and all those things. So suppose in the morning, whatever comes in our plate, if you analyze, suppose some leafy vegetable, some salad, if you just analyze what about its spraying, what about its fertilizers, then that raises alarming situation for us. So ultimately, always health is wealth. Health gone, everything gone actually. So that's how we are playing through this with our own health. When it comes to our health, then we become more conscious. So our food system, that should be good. Even before the green revolutions, we were growing. But after the advent of this green revolution and aftermath, and again recent era, this hydroponics and all those things, I just going through the paper from Netherlands just few days back regarding hydroponics and what are its repercussions. Because we are completely manipulating the system nutrient wise. So here comes the role of scientist and science. So if we are conscious enough what research is needed for the benefit of the farmers and how we can strengthen this our production system with scientific backing, and that should continue for the years together with least cost. Costing is the most important in production system. So with this word, I wish all the best for this conference. Thank you. Jai Hind.
Thank you, Mr. Kalungi sir. It's wonderful that you mentioned many things are there. So we'll start to go directly to the presentation and then self-introductions, as you mentioned that Dr. Saindes sir as a match breeder, जितने मक्का के काम किए हैं, particularly baby corn, which is a vegetable crops what we are using. Also full he has a dedication to the maize improvement programs in this country. I think he's really a farmer. If you see, when I was there as a student in IRI, I know him very well when he was in DMR. So that time how best he has done it in the particular maize improvement of the programs. And also feeding the whole nation the maize proteins, which is very important to all of us. So डॉक्टर साहब हिंदी में हो जाएगा तो थोड़ा अच्छा रहेगा तो हमारे थैंक यू सर थैंक यू सर प्लीज
प्रेजेंटेशन के लिए तो फिर या तो आप लोगों को थोड़ा सा ओके फाइन रिस्पेक्टेड किसानों की आज की दिन जो बहुत ध्यान रख रहे हैं और उनके लिए ये इस उम्र के अंदर जो घूम रहे हैं, आप लोगों के बीच में आए हैं। अग्रवाल साहब हमारे मित्र डॉक्टर बेहरा साहब हॉर्टिकल्चर के डायरेक्टर हैं, वो ये इंस्टीट्यूट भी उनके अंडर है। धन्यवाद, आपने हमको आने का मौका दिया। डायरेक्टर हॉर्टिकल्चर स्टेट गवर्नमेंट ऑफ उड़ीसा डॉक्टर साहू जी, दास साहब, डेलीगेट्स, लेडीज जेंटलमैन, स्टूडेंट, किसान भाइयों बहनों, आप सबको नमस्कार। मेरे को आदेश हुआ है कि हिंदी में बोला जाए, तुम नहीं बिल्कुल बोलेंगे संक्षिप्त में। देखिए उड़ीसा का क्यों बेबी कॉर्न और स्वीट कॉर्न के अंदर ज़्यादा महत्व है। सबसे बड़ी बात है देखो, उड़ीसा ऐसी प्लेस है नेक्स्ट अभी तक जो हमने हरियाणा से स्वीट कॉर्न बेबी कॉर्न को स्टार्ट किया था और 2 गांव भी डेवलप किए हैं और जो हिंदुस्तान के मैप पर है। अगर गूगल पर जाएंगे तो अटेरना बेबी कॉर्न आ जाएगा, मनोली स्वीट कॉर्न आएगा। उसके बाद इसका स्प्रेड हुआ, दक्षिण भारत में हुआ, महाराष्ट्र में हुआ, कर्नाटका में हुआ, तमिलनाडु में और स्टेट्स में हुआ। लेकिन ये होता कहाँ है? ये दोनों के दोनों पेरिशेबल है। अमेरिका के अंदर इसको हम सब्जियों में लेते हैं, इसलिए हमने इसको वेजिटेबल के अंदर डाला है। और बेहरा साहब को खुशी होगी कि 2007 में जब मैं डायरेक्टर मेज ज्वाइन किया तो ये सब्जी के अंदर नहीं आता था वेजिटेबल स्वीट कॉर्न को। हमने कहा इसको अमेरिका में वेजिटेबल स्वीट कॉर्न में आता तो इसको इस भी सब्जी में लो, ताकि जो कुछ स्कीम आई थी हॉर्टिकल्चर की तो किसानों को फ़ायदा हो, उसको किसानों को फ़ायदा भी हुआ। तो अब उड़ीसा के अंदर इसका क्यों महत्व है? उड़ीसा एक स्टेट है कि 70 किलोमीटर के रेडियस के अंदर ही इतने टेम्पल्स हैं, बुद्धिस्ट प्लेस है, कोणार्क है, चिल्का लेक है, और विदेशी लोग आते हैं, हिंदुस्तान के बाहर के लोग आते हैं। और जगन्नाथ जी महाराज भगवान है, जो कोई भी जो आएगा उड़ीसा के अंदर उसके बाद, और फिर भुवनेश्वर कैपिटल भी है। तो ये पेरी अर्बन के अंदर अगर इसका एरिया भी ऐसा है, पेरी अर्बन में इसको लगा सकते हैं। और उड़ीसा ऐसी राज्य है कि जब आप कभी भी लगा सकते हो बेबी कॉर्न को, स्वीट कॉर्न को, क्यों कि ये 70-80 दिन में, उड़ीसा जैसे स्टेट में 60 से 70 दिन 80 दिन के अंदर आपको क्रॉप तैयार हो सकती है। तो आप देख लिए कितने साइकिल लगा सकते हैं। तो आप डेयरी भी, दूध भी लो, किसानों को मिल्क भी मिलेगा, लाइवलीहुड सिक्योरिटी मिलेगी, हर घर को काम भी मिलेगा, 8 to 80 ईयर्स को काम मिलेगा। अब ये लगानी कहाँ है? आपको लगानी है शहरों के नज़दीक, ताकि इसकी मार्केटिंग आसान हो। आप हरियाणा के अंदर दोनों गांव क्यों पॉपुलर हुए? क्योंकि दिल्ली के पास थे। तो दिल्ली सबसे बड़ा मार्केट है और एक्सपोर्ट भी इजीली कर सकते हैं, और साथ में वहाँ से दूसरे राज्यों को भी हम एक्सपोर्ट कर सकते हैं। और दूध भी जाता है, बेबी कॉर्न भी जाती है, स्वीट कॉर्न भी जाता है, और वहाँ कोई माइग्रेशन नहीं है। अगर आप जाकर के देखिए, 2 गांव हैं जहाँ पर हर घर पक्का है, हर घर के आगे गाड़ी खड़ी है, और कोई किसान ऐसा नहीं है जो शहर में नौकरी करने जाता हो, ताकि उनको अपने घर में काम की फुर्सत नहीं है। अब इसकी क्या है, स्वीट कॉर्न जो होती है उसको ग्रीन भुट्टे में लेते हैं, उसके बाद जो चारा बचता है वो जानवर ले जाते हैं, फ्री में हो जाते हैं। बेबी कॉर्न भी क्या है, पीलिंग करो और उसके बाद उसकी छिलाई करो और जानवर को चारा जाता है। जिससे क्या होता है, किसान जिनके पास लैंडलेस फार्मर हैं उनको काम मिल गया, उनकी डेयरी का प्रमोशन मिल गया, साथ में उनको मिल्क प्रोडक्शन भी बढ़ गया, और साथ में मिल्क की भी मार्केटिंग हो गई। ये देखिए बेबी कॉर्न ये है, और सामने ऊपर स्वीट कॉर्न है, दोनों हरे हैं, दोनों स्टे ग्रीन है हर कटने के बाद। और आप आजकल साइलेज बन रहा है, तो वैसे बेस्ट क्वालिटी का साइलेज होता है ग्रेन के साथ, फॉलोड बाय बेबी कॉर्न, देन फॉलोड बाय हार्वेस्ट ऑफ ग्रीन कॉर्न। तो ये 3 जो है ना अलग अलग से वो हैं, देन फॉलोड बाय स्टे ग्रीन हाइब्रिड हार्वेस्टिंग के बाद, तो और एक विद ग्रेन के साथ होता है। तो 4 टाइप के आपका साइलेज भी बन गया, ऐसा डायरेक्टली ग्रीन फॉडर भी चला गया, और साथ में किसान को एडिशनल लैंड की जरूरत नहीं पड़ी डेयरी के लिए चारा लगाने के लिए। ये कुछ इनके जो रिक्वायरमेंट होती हैं, इनका बेबी कॉर्न, स्वीट कॉर्न नॉर्मल मक्का से थोड़ी सी डिफरेंट होती है, क्योंकि बेबी कॉर्न को डिस्टेंस हमको घटाना पड़ता है, क्योंकि भुट्टा नहीं चाहिए। स्वीट कॉर्न को डिस्टेंस बढ़ाना पड़ता है, और ग्रेन वाली में और डिस्टेंस बढ़ाना पड़ता है, फोडर वाली में डिस्टेंस और घटाना पड़ता है, और साइलेज के लिए तो डिस्टेंस। बाकी सीड रेट, जब पॉपुलेशन बढ़ती है तो आपको सीड की मात्रा भी ज़्यादा जरूरत होती है। जैसे बेबी कॉर्न के लिए हम 10 से 12 किलो रिकमेंड करते हैं। स्वीट कॉर्न छोटा दाना पिच का होता है, हल्का होता है, तो उसका हम 2 से 2.5 किलो पर एकड़ का रिकमेंड करते हैं। और साइलेज के लिए जो लगाते हैं उसके अंदर 15 से 20 किलो करते हैं, क्यों कि पौधों की संख्या बढ़ानी है, 40 से 44 होता है तो ग्रेन के साथ आएगा। और बाकी मैनेजमेंट वही है। स्वीट कॉर्न को कब काटते हैं, जब 70% ग्रेन में मॉइस्चर होता है, और 18 से 20% उसके अंदर। आजकल स्वीट कॉर्न का जो दाना पहले आता था 10-11 वाला, अब हमारे के पास 18 से 20% का TSS वैल्यू वाला स्वीट कॉर्न है। अब ये 2 जीन होते हैं जो हिंदी में बताऊं, एक तो होता है जिसका शुगर से स्टार्च जल्दी कन्वर्ट हो जाता है, वो हिंदुस्तान में कामयाब नहीं है, वो ठंडे कंट्री में चल सकता है। हमारे देश के अंदर गर्मी ज़्यादा पड़ती है, तो हमारे का SH2 जीन डाला हुआ है, एक इम्प्रूव्ड है, वो करते हैं। लेकिन उसका शुगर टू स्टार्च बहुत फास्ट है, लेकिन स्लो है स्लो है। लेकिन इसलिए हम कहते हैं शाम के टाइम स्वीट कॉर्न को हार्वेस्ट करें, और बेबी कॉर्न को भी अगर कोल्ड स्टोर नहीं है तो एकदम ट्रांसपोर्ट करिए और मंडी में ले जाओ, वहाँ पर उनके पास कोल्ड स्टोर बने हुए हैं आजकल। और बेबी कॉर्न की पीलिंग करिए, हवा छाया में रखिए, और शाम के टाइम उसकी भी छिलाई करके, आज की डेट में वो क्या करते हैं, पैकिंग करते हैं, पॉलीथीन के अंदर पैक करके सीधा दिल्ली में भेज देते हैं, या एक्सपोर्ट करते हैं। तो पहले तो प्रोसेसिंग यूनिट लगता था, कॉस्ट ज़्यादा लगती थी, आजकल ओपन जाती है। विदेशी लोग भी क्या करते हैं, वो ओपन लेना पसंद करते हैं, उनको दिखाई देती है कि क्वालिटी ऑफ बेबी कॉर्न है कि नहीं है अच्छी, स्वीट कॉर्न भी अच्छा है कि नहीं है, तो दाना जाता है आजकल स्वीट कॉर्न का दाना। हमारे हटेरना और मनौली के अंदर बहुत बड़े प्रोसेसिंग यूनिट लग गए, एक्सपोर्ट हो रहा है, साथ में दिल्ली को भी मार्केट हो और राज्य के दूसरे एरिया में भी जा रहा है। अब ये दोनों की दोनों जो है ना बहुत हेल्थ के वाइज से अच्छी है। स्वीट कॉर्न एंटीऑक्सीडेंट है और बेबी कॉर्न है रिच सोर्स ऑफ फास्फोरस है। हमारे देखकर बुजुर्गों की हड्डियां कमज़ोर हो जाती हैं, तो बेबी कॉर्न बहुत अच्छा है। और बाकी के इसके जो तत्व हैं दूसरी सब्जियों के बराबर हैं। और स्वीट कॉर्न के अंदर आयरन है, विटामिन सी है, B12 विटामिन है और विटामिन ए है और एंटीऑक्सीडेंट है। और जब खाते हैं तो काफ़ी समय तक पेट में रहता है और हर आदमी इसको ईजिली पचा सकता है। इवन डायबिटिक लोग भी खा सकते हैं, नेचुरल शुगर है इसमें। हमने इसके काफ़ी रेसिपी भी तैयार किए स्वीट कॉर्न की, बुलेटिन भी लिखे हैं। और स्वीट कॉर्न को क्या है, आप पिज़्ज़ा में डाल लेते हैं, एज सच खा सकते हैं, ग्रीन रोस्ट करके खा सकते हैं, बॉयल करके खा सकते हैं, कच्चा खा सकते हैं। आप इसका मिल्क चॉकोबार मिल्क के अंदर चॉकलेट डाल करके इसका दूध बनाकर पी सकते हैं। और इसको आजकल तो पिज़्ज़ा के अंदर बहुत चल रहा है, ये मिक्स वेजिटेबल में जा रहा है। मिक्स वेजिटेबल में आप बहुत स्वीट कॉर्न का सूप तो बहुत ही पॉपुलर है। पहले ज़माने में स्वीट कॉर्न सूप कहते थे और नॉर्मल मक्खी डाल देते थे, तो हम जाते थे बताइए स्वीट कॉर्न, तो हमने उनको बताना शुरू किया कि भैया स्वीट कॉर्न जो का सूप असली स्वीट कॉर्न होगा ना, जब दोनों जाड़ों में दबाएंगे तो मेल्ट हो जाएगा। अगर असली होगा, नकली होगा तो फाइबर चिपक जाएगा, वो स्वीट कॉर्न नहीं है। तो बाकी स्वीट कॉर्न जो उसके अंदर स्टार्च थोड़ा सा ज़्यादा होता है एज़ कम्पेयर टू कम होता है एज़ कम्पेयर टू नॉर्मल मक्का का स्टार्च। ये मैं आपको बता चुका हूँ कि किस किस तरीके से हम इसको कर सकते हैं। अब ये भी आपको बोल दिया है कि पॉपुलेशन के हिसाब से बेबी कॉर्न स्वीट कॉर्न को उसको हम 60x15 रखते हैं, इसको 60x25-30 रखते हैं ताकि भुट्टा बड़ा होगा। आजकल भुट्टा के साइज़ को देखकर, और जो भुट्टा बड़ा होगा, दाना बोल्ड होगा, तो उसकी ईजिली पीलिंग हो जाएगी, इसलिए हम डिस्टेंस। तीसरा ये है कि अगर भुट्टा मार्केट के अंदर बड़े साइज़ के भुट्टे की मार्केट है। और बेबी कॉर्न का जो है सीड हम ज़्यादा यूज़ करते हैं, ये पर हेक्टेयर में दिया हुआ है। लेकिन हम जो 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 SUSC है आजकल नहीं चलता, इसका दाना जस्ट लाइक स्वीट कॉर्न नॉर्मल ग्रेन की तरह होता है, लेकिन शुगर कंटेंट बहुत कम होता है। ये कुछ हमारे हिंदुस्तान के अंदर स्वीट कॉर्न के हाइब्रिड रिलीज हुए हैं और पब्लिक के हाइब्रिड है। लेकिन अच्छा हाइब्रिड जो मार्केट एक बार 2001 में मैंने आइडेंटिफाई किया था, शुगर 75, 3 हाइब्रिड दिए थे सिंजेंटा कंपनी ने, 74, 75, 76 and 75 was the best and it continued for 20 years growing. Nowadays the best hybrid जो कल्टीवेशन में पूरे कंट्री में है, नॉनगो मिठास है। मिठास इज द बेस्ट हाइब्रिड। तो आगे मैं तो अब फ्री मैन हूँ ना, आई कैन स्पीक द बेस्ट वेदर इट्स पब्लिक सेक्टर और प्राइवेट सेक्टर विच सूट्स टू द फार्मर। हम तो वही बात करेंगे ना जो किसान को फ़ायदा भी हो, किसान लगाए भी सही और साथ में उसकी मार्केटिंग भी हो। तो मिठास आज की डेट में बेस्ट है। एक अभी CP का भी हमने ओवर द कंट्री रिलीज़ किया, वो भी अच्छा है, CP100, CP2 भी है, लेकिन CP100 भी एक अच्छा हाइब्रिड है। ये सभी के सभी ऑलमोस्ट एक थाईलैंड का साइंटिस्ट था, उसने डेवलप किए थे तवाचाई, लेकिन वो पहले सिंजेंटा में था, उसके बाद अब वो थाईलैंड में है, थाईलैंड के बेस को। तो होता है जब कोई कंपनी से दूसरी कंपनी में जाता है तो कुछ मटेरियल भी ले जाता है तब उसको रिटेन करेंगे ना। अब हम तो इसलिए नहीं गए कि कहते भाई ये लेके सब मटेरियल लेके आ गया, सो दैट इज व्हाई आई डिड नॉट जॉइन एनी कंपनी, सो आई एम विद एवरीवन। ये जो स्वीट कॉर्न की जो खास करके पिकिंग होती है ना, सिल्क के पोलिनेशन के बाद डिपेंड करता है। हमारे हरियाणा पंजाब में जून से लेकर के अप्रैल से लेकर के जुलाई अगस्त तक तो 18 से 20 दिन पोलिनेशन के बाद। जैसे टेम्परेचर लो हो जाता है तो डेज बढ़ जाते हैं, टेम्परेचर कम होता है तो डेज बढ़ते हैं। जैसे टेम्परेचर हाई है तो 17 से 20-22 दिन के अंदर हम इसको तोड़ लेते हैं। और किसान के पास कोई रेफ्रेक्टोमीटर तो होता नहीं, वो देखता है, जाकर ऊपर का दाना चबा के देख लेता है, मीठा है, दाना निकलने के लायक हो गया है, तो एकदम तो हार्वेस्टिंग कर लेता है। लेकिन जनरली टाइम वर्कआउट किया गया है 17 से 22 दिन के अंदर। अब इसकी स्वीट कॉर्न की जो है ना, आज की डेट में जो पॉली बैग के अंदर भी जा रहे हैं, लेकिन उसके अंदर शुगर डालते हैं एज ए प्रिजर्वेटिव, और नेचुरल शुगर हार्वेस्टिंग ग्रेन निकाल के जा रहे हैं और कोल्ड स्टोर में जा रहे हैं, और नॉर्मल होता है, नेचुरल होता है, उसमें शुगर नहीं डालते। और दाना कैन में जा रहा है, उसके अंदर भी वी पुट सम शुगर, सिट्रिक एसिड एंड सम लिटिल साल्ट एज ए प्रिजर्वेटिव, इसको हम ब्राइन सोल्यूशन बोलते हैं। और उसके बाद हमारे जो स्वीट कॉर्न का सूप के रूप में भी जा रहा है। जितना जो जब पीलिंग करते हैं दाना, जो दाना इंजर्ड होता है, इट इज डजंट वेस्ट, तो वो सूप बना करके कैनिंग करके सूप बेच देते हैं, ताकि जाओ सिंपल बॉयल करो और सूप पी लो, तो कोई पार्ट इसका वेस्ट नहीं होता। वैसे ही मेज को मैं एग्रीकल्चर के अंदर ब्रह्म प्लांट कहता हूँ। क्यों कहता हूँ? कि इसका कोई पार्ट वेस्ट नहीं होता, जो भी पार्ट है यूजफुल है। अगर रूट्स हैं वेरी इजीली डिकम्पोजेबल है, सॉइल में डिकम्पोज हो जाते हैं। राइस और व्हीट का क्या है, सिलिकॉन कंटेंट होता है, वो इजीली डिकम्पोजेबल नहीं है। स्टॉक है पूरा का ड्राई भी स्टॉक, आप इसको एज ए फ्यूल यूज़ कर सकते हैं, और जहाँ स्केर्स जो फोडर है वो फोडर के रूप में भी यूज़ कर सकते हैं। और इसका तना है, लीफ है, टेसल है, कोई पार्ट इसका बेबी कॉर्न का, स्वीट कॉर्न का भी वेस्ट नहीं है, और नॉर्मल कॉर्न का वेस्ट नहीं है। और ये ऑक्सीजन रिलीज करता है, बाकी तो सब कार्बन डाइऑक्साइड। C4 प्लांट है, ये क्या करता है? ऑक्सीजन रिलीज करता है। ये कुछ बात बेबी कॉर्न के देखे हाई डेंसिटी के अंदर है, ये भी आपको इसके साइकिल ज़्यादा ले सकते हैं, लेकिन ऑलमोस्ट सेम है, क्योंकि इसको पिकिंग में टाइम लग जाता है थोड़ा सा, क्योंकि 4-5 बार पिकिंग करते हैं, तो हर 2-3 दिन इंटरवल में। तो जो स्वीट कॉर्न में टाइम लगता है फालतू का, एक 1 टाइम हार्वेस्ट होता है, इसको 4 टाइम हार्वेस्टिंग होती है। तो फ्री की लेबर जाती है, पिकिंग करके आती है, और पील करके बेबी कॉर्न दे देती है फार्मर को, जिसकी एग्रीकल्चर लैंड है, और चारा अपना लेके जाते हैं मिल्क के लिए, डेयरी के लिए। तो लैंडलेस लेबर को भी काम मिल जाता है। और बेबी कॉर्न, स्वीट कॉर्न, 2 फायदे हो रहे हैं। तो कंटिन्यूस आपको फोडर मिलता रहता है जानवरों को, तो फार्मर को भी फायदा है, लैंडलेस फार्मर को भी फायदा है। और कंटिन्यूस मिल्क सप्लाई में भी इंटरप्शन नहीं होती ग्रीन फोडर की। अदरवाइज ग्रीन फोडर की हमारी स्केर्सिटी है इंडिया के अंदर, लेकिन जो लोग ये लगाते हैं तो उनको घर बैठे उनकी डेयरी के अंदर प्रमोशन मिल जाता है, और साथ में उनका जो लेडीज़ हैं या जो घर के बच्चे हैं उनको काम भी मिल जाता है। ये इसकी को मैं क्वालिटी बताया था, देखिए तो फास्फोरस सबसे ज़्यादा होता है इसमें, और फास्फोरस जितना है किसी और क्रॉप में नहीं होता, किसी वेजिटेबल में नहीं होता। तो दिस इज रिचेस्ट सोर्स ऑफ फास्फोरस। ये कुछ पब्लिक सेक्टर में हाइब्रिड रिलीज हुए हैं, प्राइवेट में भी रिलीज हुए हैं, पर HM4 वाज द बेस्ट एंड फर्स्ट हाइब्रिड ऑफ बेबी कॉर्न, जो HE से हमने रिलीज किया, जहां मैं जिस संस्था में मैं काम करता था। वो एक ब्रांड हाइब्रिड के नाम से आता था। उस हाइब्रिड की वैल्यू क्या है? 0 रिजेक्शन, 0 रिजेक्शन। और उसकी तो चारा बहुत टेस्टी है, और फार्मर की रिपोर्ट खुद की है कि उसके चारे से 10 से 15% जानवरों का दूध बढ़ जाता है। और साथ में उसकी क्वालिटी ऐसी है कि नॉट इवन सिंगल बेबी कॉर्न रिजेक्टेड, और लंबी होने के बाद भी उसको हम बीच में कट करके 2 पैक कर सकते हैं। बाकियों के अंदर क्या थिकनेस बढ़ जाती है, जैसे आपने डिले की, ईल्ड तो बढ़ गई लेकिन थिक बढ़ गई, थिकनेस होगी, और वो रिजेक्शन ज़्यादा हो जाती है उसकी। तो वो रिजेक्शन में आता है, लेकिन इसमें कोई रिजेक्शन नहीं होता। और वो HM4 के से ही हमारा बेबी कॉर्न का स्टार्ट हुआ था 2001 में। ये देखिए, ये बेबी कॉर्न की क्वालिटी, जो भी कोई भेजता है ना प्रेजेंटेशन देते, तो HM4 की बेबी कॉर्न की प्रेजेंटेशन दे देता है। और एक्सपोर्ट भी इंडिया का HM4 से स्टार्ट हुआ था। लुधियाना के अंदर जो पहली इंडस्ट्री लगी थी, प्रोसेसिंग यूनिट, मित्तल की, एयरटेल मित्तल, उसने स्टार्ट किया था और उस बेबी कॉर्न के सैंपल से आज भी वो एक्सपोर्ट कर रहा है 20 कंट्रीज को बेबी कॉर्न। पहले हम बेबी कॉर्न भी इंपोर्ट करते थे, स्वीट कॉर्न भी। 2008 के बाद हम इंडिया जो है ना एक्सपोर्टर ऑफ बेबी कॉर्न, एक्सपोर्टर ऑफ स्वीट कॉर्न है। इसकी क्या है, जो फोडर और बेबी कॉर्न की 1:6 होती है। 6 टाइम चारा हो जाता है, 1 टाइम बेबी कॉर्न। जब बेबी कॉर्न को तोड़ेंगे और उसकी 2 से 3 सेंटीमीटर होता है, 3 सेंटीमीटर, उस टाइम बेबी कॉर्न तोड़ लो। अगर बढ़ जाएगी, ओवर ग्रोथ हो जाएगी तो थिकनेस हो जाती है। जो आजकल की जो बेबी कॉर्न वैरायटी लग रही है, सिंजेंटा की या सीपी की या दूसरों पब्लिक सेक्टर, जैसे साइज ड्यूरेशन बढ़ाया, किसान का मेरी 20 क्विंटल हील होगी है, और रिजेक्ट कितनी हुई है, लेबर कितनी लगी अननेसेसरी, और वो वेस्ट गया, वो एनिमल को जाएगा, इंसान उसको आदमी को नहीं जाएगा यूज़ में का, और ना प्रोसेसिंग यूनिट में जाएगा। तो प्रोसेसिंग यूनिट वाले को नुकसान होता है क्योंकि उसको लेबर लगानी पड़ती है सॉर्टिंग के अंदर। तो इसलिए इसको ईल्ड की तरफ ना जाए, क्वालिटी की तरफ जाए, तो आपकी भी लेबर बचेगी, रिजेक्शन भी नहीं होगी, ताकि आपको मार्केट में पैसा भी मिलेगा। और इसका जो डायमीटर 1 to 1.5 सेंटीमीटर थिकनेस होनी चाहिए और 7 to 13 सेंटीमीटर लेंथ होनी चाहिए। 7 to 10 तो पैकिंग में आ जाता है, पॉली पैक में करो, उसमें करो, और 13 सेंटीमीटर लोकल मार्केटिंग में होती है। जो HM4 था उसका 13 सेंटीमीटर तक हमने देखा था कि उसका कुछ इफेक्ट नहीं है, एज सच टेस्ट है, पर मार्केट में जाने के लिए एक्सपोर्ट करना है या पैकिंग करनी है, वो प्रॉपर पैकिंग डब्बे के अंदर जो बने हुए थे उसमें 7 से 10 सेंटीमीटर का आता है। ये 3 ग्रेड है, 1 2 3, 7 से 7 सेंटीमीटर, 7 से 10 सेंटीमीटर एंड 10 से 13 सेंटीमीटर। ऐसी आजकल ऐसी बेबी कॉर्न जो है ना विदेशों में जा रही है इंडिया से, ताकि उनको क्लीन दिखता है, क्वालिटी ठीक है, तो पैसा भी ज़्यादा लगता है, क्वालिटी भी ठीक है, और मार्केटिंग एक्सपोर्ट भी बहुत ज़्यादा हो रहा है। वो केन में जाता था, पता नहीं क्या दे दिया। तो ये इसकी पैकिंग हम केन में कर सकते हैं, ग्लास में कर सकते हैं, पॉली बैग में कर सकते हैं, थर्मोकोल शीट में कर सकते हैं। तो शेल्फ लाइफ डिपेंड करती है, क्योंकि ब्राइन सोल्यूशन डालते हैं तो अप टू ग्लास वाली में अप टू 36 मंथ, केन वाली में अप टू 18 मंथ, और पॉली बैग्स वाली में शॉर्ट लाइफ है। ये तो ग्रेड मैंने पहले बता दिया आपको, 7 से 10 एंड मोर देन। अब बेबी कॉर्न के हमने बहुत रेसिपी बनाई है, बेबी कॉर्न पिकल, बेबी कॉर्न मुरब्बा, बेबी कॉर्न की खीर, बेबी कॉर्न का रायता, बेबी कॉर्न की भुजिया, बेबी कॉर्न का हलवा, बेबी कॉर्न की बर्फी, बेबी कॉर्न का मंचूरियन, बेबी कॉर्न का चीला। ग्राइंड करो और उसको बेबी कॉर्न का पकोड़ा, वेरी पॉपुलर, जो कि सबसे पॉपुलर। ये इसकी रेसिपी बुक्स भी बनाई है और ये पॉपुलर भी हुए हैं। और आजकल तो बेबी को मिक्स वेजिटेबल में बहुत जा रहा है, और बेबी कॉर्न का ऐसा स्टीम करके भी खा सकते हैं, फील्ड में डायरेक्ट ले तो कच्ची भी खा सकते हैं, लेकिन कच्ची बाहर से आते हो नहीं खाते, क्योंकि हाथ लगे होते हैं, लेबर की मक्खियाँ बैठी होती हैं, उसको स्टीम कर लो या लिटिल बॉयल कर लो। बेबी कॉर्न को रोस्ट कर सकते हैं, बेबी कॉर्न को फ्रोजन कर सकते हैं। तो दिस डिफरेंट वे ऑफ यूजिंग, और अब स्टीम रोस्ट की हुई या फ्रोजन की हुई, थोड़ा सा गर्म पानी लो, उसमें डिप करो, इट विल बिकम जस्ट लाइक नॉर्मल बेबी कॉर्न। ये बेबी कॉर्न की डिफरेंट रेसिपी है। थैंक यू। एज पर दी उन्होंने कहा जल्दी ख़त्म करना है, मैंने जल्दी ख़त्म कर दिया। कोई क्वेश्चन, कुछ जवाब पर देने में अगर मैं समर्थ हूँ। नहीं, देखिए, पेस्ट डिजीज वीड मैनेजमेंट, इन्होंने कहा टाइम कम है तो इसलिए नहीं मैं बताया। तो खेती बेबी कॉर्न हो, स्वीट कॉर्न हो, पॉपकॉर्न हो, हाई स्टार्च हो, ऑयल हो या ग्रेन क्रॉप हो, सेम है। आपको वीड मैनेज करना पड़ेगा, डिजीज मैनेज करना पड़ेगा, पेस्ट मैनेज करना पड़ेगा। मैं ये कहता हूँ वीड जो है ना सबसे सीरियस है, दे आर मोर हार्मफुल देन पेस्ट एंड डिजीज, और डिजीज से और कीड़ों से भी ज़्यादा है। अगर हिंदुस्तान में अगर से या वर्ल्ड में देख रहे हैं तो 45% लॉस मक्का की क्रॉप को होता है, वो वीड्स के कारण होता है। जो खरपतवार है उनका सोल्यूशन है आज की डेट में हमारे पास, जैसे एट्राजीन, लोडीज, सेम्प्रा। सेम्प्रा एक बहुत अच्छा है, अग्रवाल साहब का ही कंपनी को हम थैंक्स भी करते हैं कि इसका इतना इफेक्टिव है कि साइप्रस को कंट्रोल करना हेल ऑफ जॉब था। जितनी बार काटो दोबारा हो जाता, और इतना कम्पीट करता था कि वो मक्की को उठने नहीं देता था। लेकिन वो एक नेट के ऊपर अटैक करता है, और नेट ख़त्म हो जाती आपका जो है जो खरपतवार, और बाकी 60 है ब्रॉड लीव्स हैं, उसके लिए हमारे पास एट्राजीन सिंस 64 से है। एट्राजीन एंड मेज इज फॉर द लास्ट मोर देन 60 ईयर्स। अब लोडीज भी आ गया है, तो वो उसको भी वो भी काफ़ी इफेक्टिव है। और सेम्प्रा, ये कुछ ही अलग अलग सिचुएशन है इनकी, कुछ कंपेटिबिलिटी अगर हो जाए तो एक स्प्रे हो सकता है पॉसिबिलिटी, कि वो एट्राजीन प्री एमरजेंस है। वीड्स तो ऑलरेडी एग्जिस्ट करते हैं खेत के अंदर, और मक्की को जाना है तो जो पानी आते ही एकदम वो तो उग जाएगा, मक्की को स्प्राउट करेगा, फिर जर्मिनेशन होगा, बढ़ेगा, वीड तो एकदम ग्रोथ में ले जाएंगे। तो इसलिए उसको निप द विलन द बर्ड, उसको सप्रेस करना इनिशियली बहुत ज़रूरी है। इसलिए उसको हम शुरू से कहते हैं प्री एमरजेंस एट्राजीन का स्प्रे कर दो, एक डब्बा लो पूरे खेत में कर दो। उसके बाद 30-25 दिन के बाद हम रिकमेंड करते हैं कि लोडीज का करो, बच्चे खुचे जो वो है। और लेकिन जो खरपतवार सेम्प्रा है, इसकी रिकमेंडेशन ये है कि जब आपका साइप्रस 2 या 3 पत्ती में आता हो तो इमर्जिंग है, तो उस टाइम करेंगे तो इफेक्टिव होगा। लेकिन बाद में भी होता है, लेकिन ग्रोथ को सप्रेस करता है, बट कंप्लीटली सोल्यूशन नहीं होता। तो अगर साइप्रस है तो करिए सेम्प्रा, नहीं है तो कोई जरूरत नहीं। अगर बच्चे खुचे लीव है तो लोडीज करिए, मत करिए। अगर प्री एमरजेंसी एट्राजीन से काम चलता करिए। सिचुएशन के हिसाब से हमको अटैक करना है। अब जो अच्छे किसान हैं और जो कहते हैं मैं 40 क्विंटल मक्का ले लो, 45 क्विंटल 50, वो पूरा मैनेजमेंट करते हैं, ठीक है ना, जो वो अपना पूरी क्रॉप की प्रैक्टिस को अडॉप्ट करते हैं, उनकी यील्ड भी आती है। और इन देखा जाए मैनेजमेंट प्रैक्टिसेस के साथ आपको प्रोडक्शन और प्रोडक्टिविटी बढ़ती है, और कॉस्ट ऑफ कल्टीवेशन के अंदर आता है, लेकिन वो कंपनसेट कर देती है। जैसे किसान हमारे क्या करते थे, वो सीड हाइब्रिड का नहीं लेते थे, हम कहते हाइब्रिड सीड जो है लोस्ट इनपुट कॉस्ट है। आप अपनी मक्की लगाते हो तो आपकी 4 क्विंटल होती है, ये लगाते हो 40 क्विंटल होती है। अब 4 क्विंटल भी नहीं होती थी, 2 क्विंटल भी नहीं होती थी, 2-4 मन होती थी। अब क्या है, आप 40 से 50 क्विंटल एक किसान और मक्की ले रहा है। इवन जहाँ उड़ीसा में बैठे हैं, जब मैं 2008 में कालाहांडी में ट्रायल लगवाए थे, 100 डेमोन्स्ट्रेशन दिए थे यहाँ के लोगों को, 100, उसके बाद आपके दूसरे डिस्ट्रिक्टों में भी लगवाए थे। तो ये कहने लगे जी अगर क्वालिटी प्रोटीन मक्का के लगवाने तो कालाहांडी में लगवाओ। मैं चलो लगवाते हैं। तो एक रामचंद्र रथ थे एपिकॉल के चेयरमैन, हमने 100 लगाए और उनके रिजल्ट बहुत अच्छे आए। अब फॉलो अप करना तो स्टेट गवर्नमेंट को है। अब आपके यूडी सिंह थे यहाँ सेक्रेटरी एग्रीकल्चर थे, तो मेरे पास आए, तो वो कहता जी हमको गुजरात में थे, तो फिर यहाँ भी थे वीके यूके सिंह, बहुत अच्छे थे। यहाँ पर हमने हाइब्रिड का प्रमोशन स्टार्ट किया। विंटर के अंदर हमने मेज लाए, राइस के बाद राइस लेते थे। इवन हमारे महापात्रा साहब डीजी थे यहाँ पर, जब मैंने कहा कटक में, मैंने यार यहाँ क्या करते हो आप, तो यहाँ का तो सीड प्रोडक्शन हो सकता है। विंटर के अंदर बंगाल उड़ीसा कैन बिकम सीड अप फॉर द नॉर्थ ईस्टर्न स्टेट। वंडरफुल लैंड है, पानी आपके पास है, सब कुछ है, लेकिन करने के लिए किसान यहाँ के सेटिस्फाई हुए। असल में यहाँ का किसान बहुत सेटिस्फाई है। 2-4 मन राइस हो गया, मछली ली, खाई, खुश है। आपको भाई अगर अच्छे कपड़े पहन कर सूटेड बूटेड रहना है तो पंजाब, हरियाणा, वेस्टर्न यूपी के किसान से सीखना पड़ेगा, महाराष्ट्र के किसान सीखना पड़ेगा, आंध्रा कर्नाटक के किसान से सीखना पड़ेगा। हमारे डायरेक्टर हॉर्टिकल्चर साहब बैठे हैं, महाराष्ट्र के हैं। महाराष्ट्र के अंदर पहले ही साल 37% मेज का ग्रोथ एरिया बढ़ गया था। मैंने वहाँ पर मेज हाइब्रिड को इंट्रोड्यूस किया। वहाँ पर क्या हुआ, तो शरद पवार जी ने कहा भाई मेरे एरिया में एक मीटिंग करवाओ। 5000 फार्मर की मीटिंग हुई। जब मैंने उनका शुगर कैन से मेज आ रहा है, कॉटन से मेज आ रहा है, वो उनको बड़ा झटका लगा, उनको ये लगा कि इट इज़ थ्रेट टू माई फूड सिक्योरिटी। मैं नो सर, नॉन प्रोडक्टिव बेल्ट जो शुगर केन के कॉटन की है, फार्मर को नुकसान हो रहा है, और मक्की लगाते हैं तो उनको फूड भी मिलता है, फीड भी मिलता है, फोडर भी मिलता है, फाइनेंस भी मिलता है। अब कॉटन से उनका फ्यूल मिल रहा है, शुगर केन स्टैंडर्ड ग्रोथ है तो ओनली फोडर मिल रहा है। कॉटन से फ्यूल और कॉटन शुगर केन से फोडर, फार्मर को ना मिलता है फाइनेंस, ना फीड, ना फूड। तो इसलिए वहाँ सुसाइड तो मैंने नहीं कहा, फार्मर सफ़र करता है। अब सुसाइड करता तो कुमार साहब नाराज हो जाते। तो और मेरे डीजी साहब भी बैठे थे, अप्पन साहब, मैं जी फार्म, तो कहता अच्छा ऐसे मीटिंग करो वहाँ पर। 5000 फार्मर की मोहित पाटिल डिप्टी चीफ से मीटिंग की हमने। और आज देख लिए महाराष्ट्र इंडिया का 3rd स्टेट है, 19 लाख हेक्टेयर मक्का का। मध्य प्रदेश में एक मीटिंग छोटी सी की हमने, उपाध्याय जी प्रोडक्शन कमिश्नर थे उस टाइम, शिवराज चौहान जी चीफ ने। तो आज 24 लाख हेक्टेयर एरिया है मध्य प्रदेश में, 6 लाख, एक 50,000 हेक्टेयर से मध्य महाराष्ट्र 19 लाख हेक्टेयर। और कर्नाटका के अंदर तो सबसे पहले होता था, लेकिन वहाँ भी 2 मिलियन हेक्टेयर है। लेकिन देखिए टेक्नोलॉजी में इम्पैक्ट होगा तो किसान को फ़ायदा भी होगा और पॉलिटिशियन को भी बड़ा फ़ायदा हो जाता है उसमें। तो ये मैं तो खैर मक्का पे बोलना शुरू कर तो बोलता ही रहूँगा। तो टाइम इन्होंने कहा कम, तो बैरा साहब तो मेरे ख्याल चले गए, उनको शायद जाना भी था ऐसे लगता है। तो अगर कोई आपका प्रश्न हो तो वीड मैनेजमेंट इम्पोर्टेंट है। फॉल आर्मी वर्म की हमने बहुत मीटिंग की और उसके अंदर कोराजन है, सिम्पेंटोरम है, लार्गो, अगेन अग्रवाल साहब बैठे, थैंक्स टू हिम। लार्गो का टाइम लॉन्ग सस्टेनेबिलिटी है। अगर लार्गो हम यूज़ करते हैं वो काफ़ी समय तक उसका इफेक्ट रहता है। मैं इनका स्पोक्समैन नहीं हूँ, जो रिजल्ट बता रहे हैं, कहीं कहीं कहें कि भाई अग्रवाल साहब बैठे तो मैं प्रशंसा करूँ, नहीं है। लार्गो जो है आज की डेट में उसकी सस्टेनेबिलिटी टू कंट्रोल द फॉल हमने ज़्यादा है, और इससे हम किसान को कहते हैं पहला ट्रीटमेंट्स उसका मैनेज हो गया है। नाउ फॉल आर्मी इज कम्प्लीटली मैनेज, जो कि एक हैवक था, ये 100 क्रॉप पे जा सकता था, 100 क्रॉप पे। तो थैंक्स टू द गवर्नमेंट ऑफ इंडिया एंड प्राइवेट सेक्टर आल्सो, दे ज्वाइन हैंड्स कि हमने इसको 100 मीटिंग तो मैंने की फॉल आर्मी पर, 100 मीटिंग फार्मर को क्लीन कल्टीवेशन सिखा दिया कंसेप्ट। एक तो क्लीन कल्टीवेशन का कंसेप्ट आ गया किसानों को, भाई फ्री रखो खेत को वीड से, क्यों इसे माइग्रेट कर जाएगा। तो फर्टिलाइज़र भी देने लगे, क्लीन कल्टीवेशन होने लगी, और पेस्ट एंड डिजीज़ भी कंट्रोल होने लगी, और फार्मर की इनकम बढ़ने लगी। तो थिंग्स आर आज की डेट में हर चीज़ मैनेजेबल है इंडिया के अंदर। हिंदुस्तान के अंदर टेक्नोलॉजी कमी नहीं है। इंडिया, इसलिए हमने कहा ना सीमेंट वालों को पीछे कर दिया, भाई इंडिया कैन सपोर्ट यू टेक्निकली टू इंक्रीज द इनकम प्रोडक्टिविटी एंड फार्मर लाइवलीहुड सिक्योरिटी ऑफ द अफ्रीकन कंट्री आल्सो। तो आपका कोई सवाल नहीं है तो मुझे चुप करना पड़ेगा, नहीं तो कहेंगे भाई ये बोली जा रहा है बोले जा रहा है। तो थैंक यू वेरी मच, थैंक यू वेरी मच।
बहुत बहुत धन्यवाद सर। एक बहुत ही इन्फॉर्मेटिव सेशन हमने अभी भी सुना डॉक्टर सायन दास से, और अब आगे बढ़ते हैं, लेकिन जिन्हें हम बुलाने वाले हैं उनके बारे में 2 शब्द ज़रूर कहना चाहूँगी। वेल, इट्स एन प्रिविलेज टू इंट्रोड्यूस डॉक्टर R.G. Agrawal, Chairman Emeritus, Dhanuka Agritech Limited, a visionary leader and the pioneer of India's agri-inputs industry. With over 5 decades of experience, he has played a transformative role in advancing Indian agriculture through innovation, research, and farmer-centric initiatives. His contributions extend beyond business into education, healthcare, and social development, reflecting his deep commitment for nation building. We are honored to have him with us today. He is also a recipient of numerous prestigious national honors. So I'll be requesting all of you to please give a good round of applause for Dr. R.G. Agrawal, who will be talking on role of quality pesticides input in increasing productivity.
जय जगन्नाथ, जय भारत माता की। आज के कार्यक्रम के हमारे बेहरा साहब शायद जा चुके हैं बैठे हैं, हाँ सॉरी। डॉक्टर साईदास भीषम पितामह ऑफ मक्का के, डॉक्टर तालुंके साहब डायरेक्टर हॉर्टिकल्चर, डॉक्टर दास, डॉक्टर आचार्य, बहुत से वैज्ञानिक बैठे हैं जिनको मैं नहीं सबके नाम जानता हूँ। हमारे सभी वैज्ञानिक गण, स्टूडेंट्स, फार्मर्स, सभी भाइयों और बहनों, मैं कृषि के क्षेत्र से करीबन 6 डिकेड से जुड़ा हूँ। जब प्राइवेट सेक्टर में खेती की शुरुआत हुई, खेती तो बहुत टाइम से होती थी, लेकिन जिसको हम कह सकते हैं कि ये एक सस्टेनेबल एग्रीकल्चर, जो सस्टेनेबल वर्ड की बात हो रही है, एक ज़माना था PL 480 में, 60s में हम भीख मांग कर गेहूँ आता था सड़ा हुआ, वो खाने को मजबूर थे क्योंकि बेगर्स की कोई चॉइस नहीं होती है। मेरा सब्जेक्ट है क्वालिटी एग्रोकेमिकल से क्या चमत्कार हो सकता है। एक तो चमत्कार अभी डॉक्टर साईदास ने बताया कि जो मतलब वीड से 45% तक का नुकसान होता है और वीड मैनेजमेंट हो तो उससे काफ़ी आपका नुकसान कम हो जाता है। वीड के साथ साथ फिर डिजीज़ और इंसेक्ट या फर्टिलाइजर सभी का मैनेजमेंट बहुत ज़रूरी है। आज 60s के बाद क्या ऐसा हुआ कि हम सेल्फ सफिशिएंट नहीं, आज हम एक्सपोर्ट भी कर रहे हैं। इसके लिए फर्टिलाइजर और सीड्स और क्रॉप प्रोटेक्शन केमिकल्स सभी का रोल है, लेकिन इसमें क्वालिटी वर्ड एक हमने जोड़ा, क्वालिटी। अभी बहुत से लोगों को एक भ्रम है कि हम खाना खाते हैं तो हमारे खाने में कितना पेस्टिसाइड है, कितना फर्टिलाइज़र है। अरे भाई, जितने न्यूट्रिएंट है सब हमारे ब्लड में है, जिंक भी है, आयरन भी है। आज हमारे ब्लड में जिंक और आयरन की कमी है तो डॉक्टर के पास जाओ तो शहरों में ब्लड टेस्ट करा के कहता है जिंक के कैप्सूल खाओ, आयरन के कैप्सूल खाओ। और इसलिए आज जिंक और आयरन फोर्टिफाइड राइस की वैरायटीज़ डेवलप हुई हैं कि आपको मतलब ऊपर से जिंक आयरन के कैप्सूल ना खाने पड़े, आपको अपने फूड आपका इतना न्यूट्रियस हो। जैसे अभी मक्का का इतना प्रचलन बढ़ा है, मैं समझता हूँ कि आज कोई ऐसा होटल नहीं होगा, जैसे बिना टमाटर प्याज के किचन नहीं चल सकती, तो आज के ज़माने में स्वीट कॉर्न और बेबी कॉर्न के बिना कोई होटल की किचन नहीं चल सकती। अब बंगाल फेमिन बहुत से जो बुजुर्ग लोग हैं वो जानते होंगे। हमारे यंग स्टूडेंट्स बैठे हैं तो शायद उन्होंने पढ़ा हो हिस्ट्री में बंगाल फेमिन क्या था। 42 में हमारे लाखों भाई बहन लोग भूख से मर गए। मतलब आज किसी को कहा जाए कि भूख से मर गए तो यकीन नहीं होगा, लेकिन सच्चाई है। और भूख से हिंदुस्तान में भी नहीं मरे, अकाल हमारे देश में नहीं हुआ, यूरोप में भी अकाल हुए, वहाँ पर भी लोग मरे, भूख से मरे। पैडी में एक बीमारी आई थी, लीफ स्पॉट डिजीज, और उससे सारे धान के खेत खत्म हो गए। और लिहाजा जो हुआ, जो हिस्ट्री में लिखा है, हम उस टाइम मौजूद तो नहीं थे, लेकिन जो हिस्ट्री है वो सच्चाई है। कोई मतलब पॉलिटिकल रीजन से हो तो हिस्ट्री को बदल के लिखा जाता है, लेकिन अदरवाइज हिस्ट्री सच्चाई है। अब जैसे पहले ग्रीन रेवोल्यूशन का बीज बोया लाल बहादुर शास्त्री जी ने 65 में, जय जवान जय किसान, और अपने हाथ से 2 बैलों की जोड़ी लेके PM हाउस में हल चला के गेहूं बोया। शायद बहुत से लोगों को नहीं पता होगा, लेकिन सच्चाई है।
आज भी लाल बहादुर शास्त्री मेमोरियम में जाएं तो आपको लाल बहादुर शास्त्री जी की फोटो 2 बैलों की हल्की जोड़ी चलाते हुए मिल जाएंगे। खैर, मैं राजनीति में नहीं जाना चाहता, मैं अब क्वालिटी एग्रो केमिकल्स के ऊपर बात करता हूँ। डॉक्टर बेहरा साहब से भी पीछे है। मुझे शौक है, मैं वैज्ञानिकों से मिलता रहता हूँ और जो भी मैंने कुछ सीखा है मैंने वो वैज्ञानिक लोगों से सीखा है। आज चिल्लीज के अंदर ब्लैक थ्रिप्स की रेसिस्टेंस की बहुत सीरियस प्रॉब्लम है, बहुत प्रॉब्लम। डॉक्टर बेहरा, मैं आपका ध्यान चाहूँगा और आपसे अभी भी चर्चा हो रही थी कि ये कंट्रोल नहीं हो रहा पेस्ट, और आज ये पेस्ट चिल्ली से बहुत सी दूसरी क्रॉप्स में फैल रहा है। और आपने भी कहा जब ये ब्लैक थ्रिप फ्लावर के अंदर चला जाता है तो उसको कंट्रोल करना बड़ा मुश्किल है। अब ये सबसे बड़ी बात आती है कि रेसिस्टेंस क्यों हुआ। भाई थ्रिप तो बहुत टाइम से हैं, लेकिन ये रेजिस्टेंस, ये भाई पेस्टिसाइड से नहीं मरते, ये पेस्टिसाइड की रेजिस्टेंस आ गई, तो उसके लिए क्वालिटी ऑफ पेस्टिसाइड जिम्मेदार है। जो कॉकटेल ऑफ पेस्टिसाइड्स कर्नाटक में, साउथ में यूज हुई, कर्नाटक सरकार ने 250 सैंपल बायो पेस्टिसाइड के टेस्ट किए और उनमें 12 पेस्टिसाइड की कॉकटेल, जो चाइना से स्मगल करके आए हुए पेस्टिसाइड। अब एज ए साइंटिस्ट आप जानते हैं कि एक कीड़े को मारने के लिए यू नीड ए LD50 वैल्यू डोज। अगर आप 12 पेस्टिसाइड की अलग अलग डोज डाल रहे हैं तो उसको आपने किसी एक पेस्टिसाइड की लीथल डोज तो दी नहीं। तो नतीजा क्या हुआ? आज जैसे डॉक्टर साहब ने, साईदास जी ने बताया कि भाई फॉल आर्मी वार्म भी हैवक आया, बहुत बड़ा हैवक था, लेकिन सरकार और वैज्ञानिकों ने मिलकर, डॉक्टर साईदास, डॉक्टर माई और डॉक्टर माई साहब की टीम, मैं भी, क्योंकि मैं मतलब अपनी कंपनी के कोई प्रमोशन के लिए बात नहीं है, लेकिन क्योंकि मैंने क्वालिटी की बात, आज धानु का एक बड़ा नाम है, किस वजह से है? आज क्वालिटी की वजह से है। फिर क्वालिटी के साथ साथ स्प्रे टेक्नोलॉजी भी बहुत इंपॉर्टेंट है। आज जो ब्लैक थ्रिप जिसकी रेजिस्टेंस है, या अभी कॉटन की क्रॉप हमारे देश से वाइप आउट हो रही है, वो पिंक बॉल वर्म की वजह से। आज पिंक बॉल वर्म का भी कंट्रोल नहीं होता, क्योंकि इनिशियल स्टेज पे जो हमारे देश में जो हमारे किसान भाई बैठे हैं, उनके साथ बहुत अन्याय हुआ है। उनको नकली पेस्टिसाइड, नकली बीज, नकली खाद, बहुत उनके साथ अन्याय हुआ, और इसलिए हमारे किसान जो उनको तरक्की करनी चाहिए थी वो गरीब रह गए। आज शहरों की आमदनी और किसानों की आमदनी देखें तो बहुत फर्क है, 6 गुने का फर्क है। शायद डॉक्टर साहब 6 गुने का फर्क है। डॉक्टर साईदास, शहर के लोगों की आमदनी और गांव के लोगों की आमदनी में 6 गुने का फर्क है। जैसे आपने कहा हमारे उड़ीसा के किसान भाई तो बहुत सेटिस्फाइड हैं, उनको खाने को मिल गया तो सेटिस्फाई हो जाते हैं। लेकिन आज डॉक्टर साहब जो यंग जनरेशन आ रही है वो सेटिस्फाई नहीं है। वो भी जो शहरों में जैसे आप और हम सूटेड बूटेड हैं, तो वो भी वही ज़िंदगी जीना चाहते हैं, और उनको भी हक है। उन्होंने क्या गुनाह किया कि उनको वो हक नहीं मिलना चाहिए? ये हक कैसे मिल सकता है? किसी भाषण से नहीं मिल सकता। राशन है हमारे पेट में, नाश्ता करा है तो भाषण दे रहे हैं, नाश्ता नहीं होता तो भाषण भी शायद नहीं होता। सच्चाई है। अब आज ये इनकम कैसे दूर हो, उसमें क्वालिटी क्रॉप प्रोटेक्शन केमिकल क्या रोल अदा कर सकते हैं? देखिए, हम अभी कल एक बहुत अच्छी बात हुई है कि हमने यूरोपियन यूनियन के साथ FTA साइन कर लिया फ्री ट्रेड एग्रीमेंट, जिससे यूरोप, पूरे यूरोपियन यूनियन और इंडिया का इंपोर्ट एक्सपोर्ट का बिजनेस बढ़ेगा, काफ़ी बढ़ेगा। और इसमें हमारा एग्रीकल्चर प्रोड्यूस का एक्सपोर्ट भी, आज जो ग्रेप्स हैं, हॉर्टिकल्चर में हम बैठे हैं, तो आज हमारे टेबल ग्रेप्स वन ऑफ द बेस्ट इन द वर्ल्ड, और जिनका बड़ा भारी महाराष्ट्र से नासिक और ये सांगली और ये सारी बेल्ट, मतलब कौशिक बनर्जी साहब वहाँ NRCGS के डायरेक्टर हैं, सुजोय शाह भी, और काफ़ी लोग हैं। आज ग्रेप्स के अंदर हमने बहुत उनके साथ मिलके बहुत अच्छा साइंटिफिक, हमने शुरू से लेके ग्रेप्स सोइंग की हर पोजीशन की वीडियोस बना दी उनके साथ मिलकर, और वो फार्मर्स को हम सर्कुलेट कर रहे हैं कि भाई आप इस प्रोसेस से अगर पेस्टिसाइड यूज करते हैं तो आपके कंसाइनमेंट रिजेक्ट नहीं होंगे, क्योंकि एक्सपोर्ट में 3 गुना दाम मिलता है। अगर वो रिजेक्ट हो जाते हैं तो लोकल मार्केट में उसको कम रेट मिलता है, उसका नुकसान होता है। तो आज जो क्रॉप प्रोटेक्शन केमिकल में एक ये भावना कि भाई हमारी थाली में हम बहुत जहर खा रहे हैं, हम बहुत ज़्यादा पेस्टिसाइड यूज़ कर रहे हैं। पहली बात तो मैं क्लेरिफाई करूं कि हम बहुत कम पेस्टिसाइड यूज़ करते हैं, डेवलप कंट्रीज़ के मुकाबले देखें हमारा पेस्टिसाइड का यूज है 360 ग्राम प्रति हेक्टर, जबकि आज यूरोप, अमेरिका, दूसरे देशों में देखें हैं तो हमसे 10 गुना यूज है। हम डेवलप यूरोप से मान लो अपनी एग्रीकल्चर प्रोडक्शन को कंपेयर नहीं करें ग्लोबल नॉर्थ से, आज ग्लोबल साउथ से तो हमको अपनी इनकम कंपेयर करना चाहिए या नहीं करना चाहिए। और ग्लोबल साउथ में भी हमारा पड़ोसी देश है चाइना। चाइना के मुकाबले हमारी जीडीपी प्रति हेक्टर 1/3 33%। यंग स्टूडेंट्स के लिए बहुत सोचने की बात है कि हमारी जीडीपी प्रति हेक्टर 1/3 है, तो कृषि में कितना पोटेंशियल मौजूद है। तो आज अच्छे, मैं समय मेरा हो रहा है तो मैं लास्ट बात करके अपनी बात को समाप्त करूंगा कि क्वालिटी, और क्वालिटी से पहले जो वहम है कि हमारे खाने में, भारत सरकार का कृषि मंत्रालय का एक डिवीजन है ऑल इंडिया रेजिड्यू नेटवर्क, जिनकी करीबन 28-30 NABL एक्रेडिटेड लेबोरेटरी हैं, और जो साल में करीबन 30,000 फूड सैंपल्स, इसमें पानी भी है, फिश भी है, फ्रूट्स है, वेजिटेबल्स हैं, सब कुछ टेस्ट करते हैं। और हार्डली 2% सैंपल में जो रेजिड्यू अबव MRL पाया गया, 2% सैंपल में। यूरोप में भी देखें तो 4-5% में भी उनके सैंपल्स, फूड सैंपल में रेजिड्यू अबव MRL मिलता है। अगर आज इसका साइंटिफिक यूज हो, और जो नकली का कारोबार है, नकली का धंधा है, उससे बचना है। और उससे बचने के लिए किसान भाइयों को हम ये बताते हैं, वरना हमारे पास एक वीडियो भी है, लेकिन मैं उस प्लानिंग से नहीं आया था, कि आज अच्छे पेस्टिसाइड सरकार ने बीज, उस पर QR कोड कंपलसरी कर दिया। तो आप स्मार्टफोन से QR कोड स्कैन करिए और तो आपको असली नकली का पता लग जाएगा। और आप जो सामान खरीदें बिल से खरीदें, कोई भी सामान बिना बिल, भाई 150 रुपये की चीज़ अलग है, लेकिन ख़ास तौर से एग्री इनपुट या कोई भी सामान है हम बिल से खरीदें। और मेड इन इंडिया, लास्ट बात जो मैं रख रहा हूँ, देश को विकसित भारत बनाने के लिए जो हमारे प्राइम मिनिस्टर साहब का विजन है 2047 तक विकसित भारत बनना, हमें वो बन सकते हैं। क्योंकि आज से संवत 2082 साल पहले जब महाराजा विक्रमादित्य का साम्राज्य था, तो वर्ल्ड की 1/3 GDP हमारे देश से होती थी, 1/3 GDP। आज हमारी GDP 7% चल रही है। अगर हमको विकसित भारत बनना है 2047 तक, तो हमारी GDP ग्रोथ कम से कम 10-11% होनी चाहिए, तब कहीं जाके हम 2047 में विकसित भारत बन सकते हैं। और खेती में, और ख़ासतर एग्रीकल्चर में, सीरियल में तो हमारे पास गेहूं धान रखने की जगह नहीं है, लेकिन आज फूड सिक्योरिटी की बात नहीं है, आज न्यूट्रिशन सिक्योरिटी की बात हो रही है, जिसमें हॉर्टिकल्चर, वेजिटेबल और दूसरे डेयरी, पोल्ट्री सभी का योगदान बहुत ज़्यादा है। तो आज हर क्षेत्र में कोई भी चीज़ हो, खास तौर से कृषि से कोई कंप्रोमाइज़ नहीं हो सकता और इसकी पैदावार बढ़ाने की गुंजाइश है। उसमें क्वालिटी बीज, क्वालिटी फर्टिलाइज़र, क्वालिटी सीड, हर चीज़, और साथ में स्प्रे टेक्नोलॉजी वो बहुत बहुत ज़रूरी है। किसान को पता होना चाहिए वीडीसाइड है तो कौन सा फ्लड फैन नोजल यूज़ करना है, या पेस्टिसाइड है तो होलोकोन नोजल यूज़ करना है। तो समय की सीमा है, कभी होगा तो इसमें डिटेल में हम अपने 60 साल के तजुर्बे से चर्चा कर सकते हैं कि पेस्टिसाइड, फर्टिलाइज़र अगर सही तरीके से इस्तेमाल किए जाएं तो इनका कोई नेगेटिव असर नहीं है। अगर कोई भी टूल जो हमें मिला है, आज एटॉमिक एनर्जी अगर बम से यूज़ करते हैं तो डिस्ट्रक्शन है, और बिजली करते हैं तो बहुत, आज सबको अगर लाइट है तो हम यहाँ बैठे हैं, लाइट नहीं होती तो शायद हम नहीं यहाँ बैठे होते। तो ये सब एटॉमिक एनर्जी साइंस की देन है। तो अंत में लाल बहादुर शास्त्री जी को जय जवान, जय किसान, और इसके अंदर जय विज्ञान और जय अनुसंधान। तो अगर बढ़ना है तो विज्ञान और अनुसंधान के साथ आगे बढ़ेंगे। आपने मुझे सुना, धन्यवाद। जय हिंद, जय भारत।
बहुत बहुत शुक्रिया सर। बाहर एक छोटा सा इन्फॉर्मेशन है आप सभी के लिए। द टी ब्रेक इज़ ऑलरेडी गोइंग ऑन। चूँकि हम थोड़ा सा टाइम से बहुत लेट चल रहे हैं तो हम सेशंस भी कंटिन्यू करेंगे, दैट विल ऑल्सो हैपन। अगर किसी को चाहिए तो आप वो भी ले सकते हैं। और अगले सेशन की तरफ बढ़ते हैं। लेट्स हैव प्रोफेसर कल्पना मोधा, कॉलेज ऑफ हॉर्टिकल्चर, वेंकटा रमना गुडम। She'll be talking on performance of yield and quality of cocoa grown under different cropping system in Andhra Pradesh. मैम जस्ट 1 मिनट। पछपटे जो भाई माने हमर बसचंती एटी सेशंस चलची, जदि आपण कथा हेबे तो सेटा टिके डिस्टर्ब हबो। इधर बाहर जाई की कथा होंतु नले टिके पार्टी कम करंतु। थैंक यू।
Respected dignitaries on the dais and off the dais, very good morning. My name is Dr. Kalpana. I am working as a professor in College of Horticulture, Venkataramanagudam. Okay, today I am going to talk about performance of cocoa with regard to the yield and quality, which is grown in different cropping systems in Andhra Pradesh. Before going to the actual work of this crop, I want to introduce this is one of the important breweries crop after coffee and tea. It is the important raw material for chocolate industry. It is the only raw material for chocolate industry. So actually this crop is grown near to the tropical. It is an understoried crop in Amazon River forest region. Mostly this crop is cultivated in the tropical region that is situated at 20 degrees north and 20 degrees south of the equator. So this crop was introduced in the 19th or 18th century, but its cultivation came into existence in India only in 19th century. The work on this crop has been started only after Cadbury's India Private Limited. They had given the feedback to the farmers, especially in the Kerala region. After that, the crop the area has been expanded, and this crop is mostly cultivated in the southern states only. That is mostly Andhra Pradesh, Karnataka, Kerala, and Tamil Nadu. Now this crop is also introduced into the Orissa, especially in the what we will call the coconut gardens. Okay, with regard to the area and production, Andhra Pradesh is the leading with both in area and production. Out of 1 lakh hectares, we got 32,000 tons in India. But Andhra is leading in both area and production. This crop in Andhra Pradesh is mostly grown only in the 7 districts, that is from West Godavari, East Godavari, Krishna, Visakhapatnam, Nellore, Vijayanagaram, and Srikakulam districts. Out of these 7 districts, only West Godavari is the leading one, where it occupies almost 75% of the area and also the production, followed by East Godavari districts. Only 20% of area and production is there. Meager is from other districts. Okay. So this, I already told, this crop is an understored crop. That's why it is mostly cultivated under coconut, areca nut, and oil palm gardens. But in Andhra Pradesh, the farmers are cultivating this crop under coconut and oil palm. So, one of the farmer in Andhra Pradesh in 2008, he started cultivating this crop as a solo crop. He has taken only less area, less than 1 hectare of land. In that, he planted 680 plants per hectare with a spacing of 3 into 3 meters spacing. After establishment of the crop, he got almost 3.5 kg per hectare with a population of 380 plants. He told that we are getting more than 3 lakhs of net profit income, but we don't have any scientific evidences for that. So, initially, what he did means after planting, he planted the shade plants, banana as a shade plants. Up to 2 to 3 years, he maintained that banana. After that, the banana crop was removed. This banana crop, which acted as a shade plant up to 2 to 3 years. Why means 2 to 3 years it will take for first jorketing. So, it's a height of almost 1 to 2 meters from the ground. The jorketing will take place means 1 meter from the ground. Okay. So, seeing all this, the other farmers also started cultivating this crop as a monocrop. Actually, this is a shade-loving crop and understored crop, but when one farmer started, means it is encouraged, it was encouragement to the other farmers. They also started this cultivating crop. Like that, the area under monocropping is slowly expanding in Andhra Pradesh. But we don't have any data on this crop. So one of our students started working on this crop, surveyed and gathered the information from the farmers. So what she did was, with a few objectives, he collected the data on the growth and yield of the crop in different cropping systems, and also the bean quality. Why means fat is very important in this crop, where the fat content in the bean is almost 50 to 55%. Okay. So, the bean quality and also the economics. How much returns the farmer will get if he cultivated this crop as a monocrop. So, here we have taken the 2 factors. One is the cropping system and the second one is the soil type. Why we have taken the soil also? Most of the area under this crop is in laterite soils, and few in East Godavari district is in alluvial soils. So we started collecting the data in both the type of the soils with 3 cropping systems, that is cocoa under coconut plantation and cocoa under oil palm and cocoa as a monocrop. The variety is forestro. So these are the geographical locations of the farmer's garden where we collected the data. So this is the cocoa under coconut in the laterite soils. This is the farmer who is cultivating the cocoa In oil palm, actually, oil palm in Andhra Pradesh is the leading crop under plantations. Maximum area and production of oil palm is from West Godavari district only. That in that too, they used to plant the crop in hexagonal type of system. That they call it as a triangular system. But this cocoa is planted when the oil palm is planted on the square system of planting, where the spacing of oil palm is around 9 into 9 meters, and also the cocoa is around 3 meters spacing. Only in the single line they used to plant. So this is the farmer who is cultivating cocoa as a monocrop in less than 2 acres of land. So this is coconut, cocoa under coconut in alluvial soils, and also under oil palm in alluvial soils, and cocoa in alluvial soils as a monocrop. So before going that, we started collecting the soil factors also, that is pH, EC, and organic carbon content, where the pH ranged from slightly acidic to the neutral, and also the organic carbon is also, organic carbon percent is also normal. So in that gardens also we started collecting the NPK content, which is ranging normal. Generally the recommendation of for cocoa crop is 100, 40 and 40, which is given by the CPCRI Vittal. But farmers, as the crop is grown as an intercrop in the coconut and oil palm gardens under irrigated conditions, the farmer supplies more fertilizers to the cocoa crop, that is almost 500 grams of urea and 500 grams of single superphosphate and 500 grams of muriate of potash. It is a little bit higher dose. So the growth parameters, when we started recording the growth parameters, generally in open conditions the growth parameters are little bit robust only, where in cocoa as a monocrop, they got more canopy volume, which reflects the yield indirectly. So then actually cocoa crop got one habit, means it will give the pods throughout the year under very good congenial conditions. But in AP, we will take only the harvest from the December to the Jan, which is the peak season. In western conditions, they will take harvest to 2 seasons. But here in AP, we will take only 1 season crop from December to May, June. By June, the crop will be completed, but still pots will be there. The Mondelez India Private Limited, they ask the farmers to remove the chirals, but farmers, because they will get little bit income, so they remain the pots like that on the during the rainy season also. They will harvest the crop. Means whatever the leftover pots will be there, they will harvest in the rainy season. So the number of the pots, if you see, the number of the pots ranged minimum in the cocoa under oil palm. Why? Means in oil palm we got more shade. And CPCRI Vittal, they are also trying for the variety which will give good yield under oil palm conditions. But in cocoa as an open field, we are getting more than 92 pots. 92 pots per plant, per tree. So this is the performance of, means number of pots on the tree in under coconut. This is oil palm, and in this one is cocoa. This is near to Vijayrai area. Then in alluvial soils. So generally the pod weight also, it ranged from 375 grams to peak of 655 grams. The maximum pod weight is only recorded in the cocoa as a monocrop. So this, like this, we have weigh the pod weight. And the fresh bean weight and dry bean weight also recorded more in cocoa under monocropping system only. So this is the fresh bean weight and the dry bean weight. And the nib recovery, why means the nib recovery is more in the cocoa as a monocrop, because the bold beans will get when it is grown under open conditions, where means the canopy is more. Because of the canopy, the photosynthesis will be more, so that it will divert to the pod yield, and also encourage the nib yields. So this is the dry bean weight. The minimum dry bean weight they observed in cocoa under the oil palm only 1.45 kgs, means less than 2 kgs. Less than 2 kgs yield will be is observed in cocoa under oil palm. More yields are recorded in cocoa as a monocrop, which is 5.3 kg per tree. So per ton, obviously the cocoa which is grown on the laterite soils, it is 3.64 tons per hectare, whereas in alluvial soils it is 3.1 ton per hectare. Even the phenols are very important in, pardon sir? Okay sir, okay. Phenols are important in cocoa because nowadays the chocolates, doctors are encouraging to take the diabetic patients or heart patients the dark chocolate. So the phenols are very high in cocoa as a monocrop, and also the fat content I already told, it is rich in very fat content. Nowadays it is using in the cosmetics also. The cocoa fat is, butter is using in the cosmetics also. The fat content is also recorded more in the cocoa as a monocrop. So cost benefit ratio, we got more in cocoa as a laterite soils, followed by the cocoa monocropping in alluvial soils. So in nutshell, what I want to say is, we can encourage this cocoa as a monocrop only in less acreage area. Why means, it is a partially shade loving plant. In long run, what will happen means, the cocoa, because the temperatures are very high, cocoa when it is exposed to high temperatures, there will be a breaking of the trunk where the pods will develop. So this can be encouraged only when the crop is taken in less acreage area and it is surrounded by any perennial plantation crops or agroforestry crops. And if there is any outbreak, at that time also, we cannot encourage the farmers to go as cocoa as a monocrop, but sustainable cropping system is very important. So cocoa can be grown as a intercrop in coconut gardens along with the other biennial crops like your pineapple and also banana, and also means multi-storied cropping system. That is that should be encouraged to the farmers. If they are interested to take as a monocropping, we should encourage only less acreage area, even though they get more yields. Thank you. Thank you for giving an opportunity.
Thank you, Dr. Kalpana. As you mentioned, the monocrop of this cocoa is very important, but again you can mention also the intercropping also fetch the good price to the farmers.
Yes.
So this is wonderful. So we'll discuss during some tea and coffee. Somebody have some questions because
Yes, sir.
already we are late and that way. So
Okay,
thank you
sir.
very much for
Thank
this
you,
one.
sir.
So yeah, please.
Sir, actually, we are doing this parallelly. चाय चल रहा है, ठीक है. We are running actually 45 minutes late, तो हम session शुरू करते हैं. So that was all for the plenary session. We will now start with the technical one. The first speaker for the technical session is Dr. Sumit Tripathi, IIT RAM, Ahmedabad. He'll be talking on the topic wettability studies of plant leaf services using an optical tensiometer with water, oil and emulsions. Just a request, these technical sessions, each session would be of 10 minutes and speakers are requested to please adhere to the time. 10 minutes. 7 minutes. Okay. So now it is 7 minutes, sir. Please adhere to the time.
Hello, good morning. My name is Sumit. I am working as assistant professor in the Mechanical and Aerospace Engineering Department at IIT RAM Ahmedabad. We had a wonderful talk by Agrawal sir, and you might wonder what an engineering guy is doing in this agriculture conference. So please forgive me if I say anything wrong about the agriculture things, but what we do, we work on fluid mechanics, and we work on different measurement techniques like contact angle measurements and dynamic interfacial surface tension measurements, which help in developing some better formulations for emulsions. So emulsions are used in agriculture in various ways: fertilizer applications, pesticide formulations, dust suppressions, soil amendments, drip irrigation system. So the main thing is the farmer faces challenges from limited resources and chemical overuse. Most of the things that we spray on the plant surface, they don't retain on the plant surface, they actually go to the soil, as whatever I have studied. What we are doing, we are trying to use this advanced emulsion technologies. Actually, emulsion technologies is quite advanced now, but they are quite limited to things like cosmetic products, paints, and not too much in agriculture. So why don't we use them in agriculture? We have this conventional emulsion, then picric emulsion, which are stabilized by particles. Then we have nano emulsions, solid lipid particles, and high internal phase emulsions, which are highly concentrated. The internal phase volume fraction is more than 74% in this case. So the existing agrochemicals do not offer tailored solutions for local crops. So I particularly like this image, which is I have taken from the literature, that to retain the agri inputs, you should have a specific droplet size. The problem, the good thing about small droplets, which are about 100 micron on a leaf surface, so these are the different droplet size on a leaf surface. If you have a small droplets, the evaporation is very fast. Although the coverage is very good, but the evaporation is very fast. If we have a bigger droplet like 600 micron, the evaporation is slow, but the coverage is not that good. The droplets rebound and they don't retain on the leaf surface. So for this, we can actually develop some emulsions. And if just a quick introduction of emulsions. Emulsions are mixtures of two immiscible liquids stabilized by some surfactant or emulsifier, and nowadays with particles also. So the emulsions can be water in oil or oil in water or multiple emulsions. Then there's HLB scale and stability issues. So HLB is the hydrophilic lipophilic balance number. It decides the type of emulsifier. If the HLB number is large, then it is water soluble emulsifier, and if it is low, then it is oil soluble emulsifier. These are the emulsion breakdown mechanisms. Emulsions can break, so it is very important to formulate a stable emulsion with having certain shelf life. Emulsion and particles, nowadays we don't only use the emulsifiers, we also use some particular particles to stabilize the emulsions. So these are some of the details of that. So because of the time limit, I'm just skipping these slides. These are some of the dimensionless numbers that we widely use in engineering in our fluid mechanics things. So Weber number, Reynolds number, and they decide that what type of forces are there during the emulsion formulations. So what we do in our lab, so this is an optical tensiometer. It's a very simple technique, and it is based on the Young-Laplace equation. So what it does, it relates the coverage, the curvature of the droplets with the surface tension and the pressure. So we just need to take the image and we can directly measure the surface tension. So these are some of the snaps of from my lab. So what we do, we take different types of leaves and we put certain type of water or oil or emulsifier, and then we measure the contact angle, as well as for in a different study we also measure the dynamic surface tension. So these are very few results that I could include in this. So this is a Tulsi leaf, which is, although we don't need to use pesticides on Tulsi leaf in general, but these are some of the results with water drop and oil drop and emulsion drop. Oil also we can choose different types. So what we have, so these results are for the groundnut oil, and the emulsion also we have prepared using certain certain composition that we have taken from the literature. And these are on a oleander leaf. So similar results are there. So this is the dynamic contact angle with respect to time. So we can see that the black one is for the for the 5 microliter droplet, and this blue one is for the 5 microliter emulsion droplet, and the red one is for the oil. So actually the black one is the top, which is water, and red one is the oil. And in between we can tailor any formulation, and we can decide that what exactly we want on the leaf surface. So this is just on this leaf. We have done it for various other leaves also. So this is another result for the same. This is how we prepare emulsion. So there is a specific method with which we can prepare the emulsion. So we just add the oil and water and mix it in particular speed, and then we get this kind of emulsion. In commercial formulations in our agriculture also, we have neem based emulsifiers which we need to dilute. They are at certain ppm values. The pesticide is there in 3000 ppm or 5000 ppm or 10,000 ppm. So they are also concentrated emulsion that we need to dilute before using. This is something that we are doing contact angle measurements. So we have prepared different samples, and these are some of the contact angle measurements. So concluding is that the droplet spreading on leaf shows a strong dependence on types and the other constituents like surfactant, what type of surfactant we use. The time-dependent evolution of contact angle demonstrate good agreement with established wetting models. So we have also, we do some theoretical study also, we do experiments, and we try to relate it with the theory. So we have observed some good results. So that's it. I would like to thank my institute and this funded project and some of my students. So thank you.
Thank you so much, Dr. Sumit Tripathi, for finishing this on time. We'll do the questions later. So moving ahead with the next speaker, we will request Dr. Dulal Chakraborty, Indian Meteorological Department, Ministry of Earth Sciences, Government of India, to talk on the topic empowering Gen Z with a new digital modeling framework for climate resilient agriculture.
Please.
Thank you, Mr. Chairman. Really, it was a very learning type of information systems, but what I am going to tell you now is a very latest development for the farmers. I mean, you can use, today the farmer is not only a recipient of advisories and then which cannot be proved, they are due to various climate risks. Like say, for example, there will be sudden thunderstorm, wind lodging. So all these things are affecting the farmers. So the farmers need not be only at the receiving end, but he can be a data steward using his mobile smartphone. How it is to be done, so that is what I have tried to project here. Now, first thing in any agricultural cultivation is geotagging of crop image. Today, the first dispute will come that ये तो आपका फील्ड नहीं है, ये कोई और का फोटो आपने भेज दिया. But you know, one photo is worth thousands of words. So if you can geotag an image, this is available as a feature in a smartphone. And then two things. One is prompt and response. This an artificial intelligence model, like say here I'll be showing the Gemini's performance. If you prompt it in a proper way, it will respond to it. It will identify the crop. It will tell you the stage of the crop growth and also the health of the crop. It will give you various types of suggestions and recommendations, including insurance claim dispute. It will help you to preserve a sustainable, to preserve a actionable evidence in the form of the image. And most important thing is the smartphone holder, he need not be an expert in English language or in any language. Today, this Gemini AI model is available in almost all the Indian languages. So let us see how it works. So this is the artificial intelligence framework. What it does is, a farmer has to capture his image whenever he thinks. One time, 2 times, 3 times. Actually, the data is with him. He need not go to anywhere else and ask for the response. And then the interface, which is the prompt which he will be making, namely, in this mobile phone, he will be prompting it with like, I will show you, and then response will come again from the Gemini, and this evidence is already recorded, so it can be uploaded to the insurance authorities. So here I have shown the first this is the field which is I mean, here there is an embedded video. I don't know, this may not be working here, but the response is coming from the okay. Now here, the geotagging process has been spoken in detail by the AI model itself. And you see, here the location is shown as, once it is geotagged, the location comes right on your screen, and you can save it. and upload also to the insurance authorities. Now, how you install and set up the Gemini Live, that is the logo of Google Gemini AI, and the latest is 3.0. And here you see there is a, I don't know, I mean, it is not, okay. And the most important thing is, as I told you earlier, the prompt and response, they can be both generated in the local, all the Indian local languages, it is available. Say, for example, if the embedded video would have worked, I could have shown you how it is prompted in Oriya and how it is prompted in Telugu. And so all the Indian languages, Tamil, Bengali, all languages are available. And here you see the actions for the insurance claim that is in detail. Here is a case of a lodged crop. So there will be crop loss, and if you are not taking very quick action, then it will lead to your insurance claim dispute. So the recorded evidence is here, and okay, so that's all. So with this facility in the hand of a farmer, he becomes the data steward himself. And if it is adopted, then the agricultural details will be available in a hyper-local mode. So that's all. If you have any question,
Thank
I will
you.
put this video in my social media links. And if you have any questions,
Thank you. Yeah, please. Very nice topic, Dr. Saab. Thank you very much. A wonderful topic, as you mentioned. Any questions if you have, because we have time 1 minute.
Yeah.
So please, if you have any one questions, because it is a model which is giving by the AI-based model, which is Google Gemini AI model. I think these are some of the new things when you are talking about this digital era of the work of the agriculture, and also climate resilience and the agriculture. So if anybody have any questions for one questions, please, or we can have a discussion with some other time also. Anybody?
Anyone have any question? We can propose one question as of now.
So okay, sir. That's up. Thank you very much.
Okay,
I think
okay,
many of the
thank you.
things people have understood. That's a good question. Yeah, thanks. Thank you, Dr. Sir.
Thank you so much, sir. Well, we move ahead with Dr. Harshata Pal from Amity University, Kolkata. She'll be talking on the topic untangling regulated metabolic and molecular features in plant cells for surviving under salt stress by speed priming and mangrove-derived bacterial biostimulant.
Jai Jagannath. Very good morning, one and all dignitaries present over here, and mainly the farmers, those who came over here for supporting us, for whom we are standing over here right now. So my journey is a very small story, although it's an extensive last 3 years research work of my PhD student. And it is a kind of alternative proposal by employing our own native resources, invisible resources towards the sustainable agriculture. To understand this thought, I am working on the story of using beneficial microorganisms towards sustainable agriculture. I'll move on with my story. So we know we have huge approaches towards the resilience and biotic abiotic stress of the plants. We have many technologies which are having its bottlenecks as well as the advantages in terms of the policy issues and whatever the responses we get from the research work. I have moved forward towards the story of the plant biostimulants, which is very emerging, and last 5 years it is extensively progressing throughout the world towards the employment of this biostimulant research in terms of agricultural resilience, import, as well as the sustainability. So you know it's not only the microbes, it also the non-microbial biostimulants, which can restore the plant system itself in terms of resilience, and it promote the plant health, it promote the plant growth, as well as its production. I will just try to prove it. So the story of biostimulants, including the non-microbial sources and along with the microbial sources, which could be of endophytic or might be of rhizospheric. Definitely the Made in India story goes with research approach which we are targeting utilizing our own resources. So my story started with the hypothesis that we think of our own Sundarban mangrove delta, which is having huge resource of halotolerant plants, and we tried to understand whether the endophytes, root endophytes, which are harboring in those mangrove plants, whether they do have the capacity to serve the host plant, non-host plant in terms of the food crops. So they are not the food crops. So we tried it in the food crops. So we collected the background story. We started with the application story and tried to reach to the understanding in mechanistic view of metabolomics as well as transcriptomics approach. As we know, the story of biostimulant is definitely interacting with the plant systems through its different signaling molecules and volatile molecules or the signaling cascades, which is restoring the plant system through its defense mechanisms, plant metabolites, nutritional components, and the growth inducing factors. So our objective is to isolate and identify root endophytes from the mangrove plants growing naturally in the saline soils and to perform, in terms of judge their performance with respect to alleviate major stress in our country as well as internationally, that is salt stress. So I have kept some questions in my scientific background, that is whether these strains are actually having the characters or they do have the capacity to coexist together or co-act together, not a single strain, it is a multiple strain, it is a coactive microbial organization. And then we try to model crop tomato, which is a food crop and a non-host crop. And also we studied a nice and wide story of mechanistic understanding for that purpose. So my students, actually they started the PhD program last 3 years back, and she is supposed to complete this work. It is already almost completed, collected the samples from these 2 regions of Sundarban regions. They have collected 12 species of the mangrove plants, and those mangrove plants are identified properly with the Botanical Survey of India. They have identified. Then we started the isolation of the endophytes from those plants, and we started with the media-specific isolations to focus on the specific genus growth. And after isolating, we have selected more or less 31 culturable isolates, which are bacterial endophytic isolates, and gradually we go through the process of screening. And through the process of initial screening, we selected 7 isolates from the 31 isolates by putting stress to a specific 250 millimolar concentrations and gradually checking whether which strain is showing the direct plant growth impact. First, we checked for the application in the plant. Then we gradually took our specific growth function towards all the strains, means in terms of 7 strains which we selected, and we have seen uninoculated plant is showing the extensively loss and reduction of the health, and the plants which we have selected 7 strains, they were going very nicely in this regime of salt stress. Then we move on with the normal studies of our bacteria, colony morphology, then biochemistry, then molecular identifications. We have put our sequences in NCBI through the 16S rRNA analysis. We have then checked the all qualitative and quantitative analysis of the plant growth promoting factors in relation to zinc solubilization, siderophore production, SSC deaminase, HCN production, ammonia production, IA production, then your biofilm production, as well as many analyses. I am not putting it over here. So we have tried to do it qualitatively as well as quantitatively, and then we tried to check whether it is colonizing in the plant system internally or not. So we go for a scanning electron microscopy. We have a very nice result that it is nicely colonizing with respect to the remaining microbial population within the plant systems. Then we checked for the compatibility analysis of these 7 strains. We have checked the compatibility group 4. Finally, we selected this 4th strain to check their coexistence as a microbial consortia. So we formulated the consortia with these 4 strains, and then it was actually liquid carrier-based microbial formulations. We just seed primed tomato seeds we considered for priming, and we go through the process of seed priming, and then we checked the pot experiment as well as the field experiments. And gradually when we moved on to the pot experiment and field experiments, we have kept 5 groups to check our parameters. And these 5 groups, either treated, non-treated, stressed, non-stressed. Keeping in this group, we have carried out the morphophysiological analysis, plant height, and all other morphological features. Along with this, we tried for the root biomass, shoot biomass. Here you can see group 1 and group 3, which is treated, stressed, and only treated, are performing very nicely in terms of your pot experiment, and we have a very excellent yield with respect to group 1 and group 3 for our field experiment. So we have chosen in a Sundarban region 1 field, and also we have imposed the stress over there, and this was the result of the pot experiment where we have very nicely outcome of this all 4 groups and differential growth patterns. So then we move to the biochemical analysis. Extensive work has been done on this, about 15 characters. So in short, your membrane transaction in terms of sodium-potassium uptake, which is very important in salt stress. And then next, your proline content, glycine betaine, which is a very important signature molecule in the salt stress resilience. Then total phenolics. Then we moved to our molecular understanding towards the metabolomics through the gas chromatography, HPLC, and SIMS analysis. And gradually we moved on to the understanding the metabolite signatures which are available through this biochemical analysis. Phenolic acids, which is also showed a very nice result with respect to the stress treated and stress non-treated. And gradually we moved on with all the plant hormone study, as well as your specific polyamines, pyrimidine, which is having a very nice result with respect to our treatment groups. And accordingly, we propose this hypothesis that this green color showing the signature molecules, which is representing the pathway, which is linking towards the resilience of the plants through metabolomic signature modulations. So then we move to the transcriptomic study through the help of the RNA sequencing analysis, and we progressed with the RNA-seq data. In short, I am just presenting in a quick view that gene ontology proposed us that antioxidant defense genes, secondary metabolic system genes, which are related to the plant system, as well as the transporter genes, is highly regulating the plant resilience. So we have coordinated our raw data as well as our high throughput data in an integral system through the WGCN analysis. Gradually we have the signature good expression and linkage with the signatory changes with the different color groups and all the biochemical parameters which we studied with our transcriptomics data. This is actually the amalgamation of the transcriptomics data and all our parameters. So then we proposed a concept that this kind of ion homeostasis balance, abiotic stressor balance, and as well as the regulatory component in the plants for more and more photosynthetic production is proposing through our experimental outcome. And this journey tells you the genes which might be significantly linked with the salt stress resilience, the green one. So this was in short my story, started with the isolation from endophytes of Sundarban mangroves, and ending to the journey of proposing a molecular insight towards its functionality. Our next work is already published in BAB as well as Plant Cell Reports. this was my PhD scholars work. So I would like to thank my scholars, definitely without whom it would have not been possible. And my funding body, IGSTC, which is the Indo-German bilateral research program. I would like to thank all my co-colleagues and the institutes who helped me to carry out my experiment. Thank you so much for listening.
Thank you very much. Wonderful talk. And as you mentioned, the biostimulants, particularly for the mangrove region. My question is that what about the registrations of this under the CBIRC? Region biosafety issues are already done?
Yeah,
because
so
now biostimulants also come under CBIRC registration.
Yeah.
So for that, did we have any of the information?
We are actually, sir, in the process, because this was my PhD scholar's research work, and she is going to submit the thesis right now.
Yeah.
So after her submission of the thesis,
Yeah.
I'll be progressing. Dr. Pranjeep sir, who is the ICAR, means Indian Council, he is highly linked. I am highly linked with him. So Pranjeep sir is actually guiding me to coordinate how to make it commercialize and the safety issues. He is dealing with the biostimulants.
That's good because it is required. Otherwise, the registration if it's not there, particular biosafety issues are not
Yes,
there.
yes,
Well
yes.
test. You would
Yes.
have all the genomic level you would have it.
Yes, yes.
Metabolomics and the genomics level. But
Yes.
again, we have to think of.
Yes, we also have given this product to the farmers who are growing beetle vines in the saline
It's okay,
prawn
but
zones.
that is again, don't put it in the field itself
Yes.
because there will be again issues of the biosafety concern
Yes,
will come.
yes, yes.
So for that, have some closed experimentation is required.
Yes, yes.
Containment facility should be required for that one. Otherwise, people will not have taken.
Yes,
this one.
definitely, sir. We are
So have to
trying
take
best.
care. We have to
Yeah.
take care. Thank you very much.
Thank you so much.
Thank you. Thank you.
Thank you so much, Dr. Harshita. We'll move ahead with Dr. Hare Krishna Swami, Botanical Survey of India, ERC, Shillong. He'll be talking on the topic, the bright side of parasitic plants. What are they good for? I don't think that he's here. So I'll be requesting Dr. R. Arthuselvan, ICAR CT CRI Bhubaneswar, to come and talk about comparative genomics analysis of rhizosphere and endophytic trichoderma isolates from tropical tuber crops.
Good afternoon, everyone. I'm here to present my research salient achievement on the topic of comparative genomic analysis of rhizosporic and endophytic trichoderma isolates from tropical tuber crops. So we all know that tropical tuber crops plays a vital role in food security and livelihood, mainly in Odisha. So, but it is severely affected by fungal diseases, especially by the pathogenic fungus like Sclerotium, Fusarium, Phytophthora, Coltotrichum. At severe cases, it may lead to 30 to 70% of yield loss. Keeping in this consideration, we thought to take trichoderma, a promising indigenous biocontrol agent, to overcome this pathogenic fungus. So what we have done, we have taken up a survey, a periodical survey from 2022 to 2024. We have collected around 120 samples consisting of rhizosporic soil and endophytic plant parts. And we have isolated around 88 isolates totally, comprising of 61 rhizosporic trichoderma isolate and 27 endophytic trichoderma. And these isolates we have subjected to two studies, growth rate assay and dual culture assay. We have studied the growth pattern under in vitro condition, and we have noted the fast growing trichoderma isolates. Parallelly, we have tested its antagonistic activity against one soil-borne pathogen, Sclerotium, and one foliar-borne pathogen, Coltotrichum, and we have tested. And we have found that the isolate 29 and 73 found to be highly effective. Based on this, we want to identify at molecular level. So we have subjected to PCR, the traditional method for detecting the trichoderma species. We have prepared the pure culture broth. We have extracted the DNA, and we have designed a specific primer for identification of different trichoderma species, and we have run the PCR. And we found that around 4 clade of trichoderma is found to be under this 88 isolates that has been sequenced through Sanger sequencing, and we have submitted in the NCBI. Following that, we have documented the morphological picture by under the scanning electron microscopy of different strains, important strains, that also documented. And we also inoculated in the soil where the severe infestation of sclerotium is noted, and we have found the clear colonization ability, you can see in the picture, against the pathogen. It found that it is highly antagonistic activity potential trichoderma strain. Following that, we also want to see how it can be produced under cost-effective condition. So we have taken 3 different types of media: culture media, tuber substrate, and tuber peel. And we have taken 10 different tuber media, and we have inoculated the potential trichoderma strain. And we found that sweet potato in culture media, tuber substrate, and tuber peel found to be highly producing mass volume of trichoderma within 7 days. So not only mass production, we also should know its compatibility with agrochemical. So in this study, we have taken around 15 botanicals and 29 agrochemicals with one control. We have tested under in vitro condition to test this compatibility. And we found that in botanicals, betel extract found to be least compatible, whereas in case of agrochemical, out of 29, carbendazim and combination of mango sap and thiophanate methyl were found to be least suitable for trichoderma. This is the overall ANOVA table what we have found. And this is so these 2 important strains we have taken up to the field condition because we have tested this mass production ability, we have tested its compatibility, and we have detected 2 PCR. So field level study we have taken. Same we have taken 2 diseases, soil-borne diseases through cholera disease of elephant foot yam. And we found that this strain found to be highly effective in reducing the disease incidence, recording the lowest disease incidence of 3.6 with producing the highest tuber yield of 28.6 tons per hectare. Similarly, we have tried for greater yam also for foliar pathogen where we have sprayed. Here also we got the similar kind of result. We got the lowest disease incidence of 8.5 and followed by 19.5 highest yield in the trichoderma inoculated fields. So considering all this lab study and field study, we thought to study the differentiation between the rhizosporic and endophytic trichoderma. So we have subjected the both isolates to Illumina platform. We have done the whole genome sequencing. Once we got the sequencing result, we have submitted to the NCBI under this bio project, biosample and SRA number, and we got it. So this is the overall differentiation of this endophytic and rhizosporic trichoderma isolate. And if you see overall, the raw reads range from 31.6 million to 53.4, whereas in case of total bases in the GB size, 9.56 GB to 16 GB roughly. But in case of number of chromosomes, it is 7, and total predicted genes is around 12,800, which is very common for trichoderma isolate. When we study the gene ontology under different classes like biological process, cellular component, and molecular function, which has more or less similar identical between the both genome, which clearly shows that the whole genomic framework of both isolates are similar. Then we want to know the functional biological process of this both genome. Here we also tested in the KEGG category. We have tested around 27 category. We found that 25 categories are almost similar, identical. It shows that they both are similar in biological functional process. Here comes the main point, because trichoderma is known for its degradation of plant polymers in the soil. So we deeply look into this Cas enzyme analysis so that we will get a clear picture differentiation between the rhizosporic and endophytic trichoderma. As we expected, we found the difference in the carbohydrate binding molecule, which is very much essential for sticking of trichoderma in the root system and multiplication. So when we found the clear rhizosporic trichoderma isolate has an upper hand over the endophytic, it clearly shows that in the compatibility in the soil application, this rhizosporic trichoderma has a higher advantage over endophytic trichoderma. So we also studied the secondary metabolite profiling also. Here also we didn't find much difference because both these isolates has been categorized into 47 biosynthetic gene clusters only, which shows that the both isolates has the high potential of producing the antibiotics, siderophore production, and other secondary metabolites. It found to be that both are indicated. So overall, the rhizosporic and endophytic trichoderma, what we have studied, this rhizosporic trichoderma harzianum found to be little superior over endophytic in adhesion in the root zone, and also it is highly suited for soil application. This is the overall conclusion what we have studied. We started with isolation and characterization of the trichoderma isolate, around 88 isolates, both rhizosporic and endophytic, and we have selected based on the strong antagonistic activity through dual culture assay. We also used the SEM analysis for documentation of his mycoparrot stem and direct hyphal interaction, and also we have evaluated the compatibility and with the agrochemicals, and also we have studied the integrated disease management for 2 different diseases. And we also successfully detected through PCR and Sanger sequencing. And finally, we have completed the whole genome sequencing for the 2 isolates. So I want to thank RKVY, my director, staff member, and lab member of CTCRI.
Okay,
So
thank you, Dr. Selvan. One important, as I mentioned, is trichoderma, particularly isolates in the different crop species, also rhizosphere, we have different type of activities. So what way you have to, again, because you have done all these analysis with respect to their genomic analysis, then whether you have done it or not, the metabolomics in this particular trichoderma. But one important is that in that particular phase, we have a lot of trichoderma isolates are there in this India. Okay.
Yes, sir.
How best your trichoderma, the efficiency and the efficacy of this particular type species?
Sir, our objective of the study is to, because some people are claiming endophytic is superior than rhizosporic trichoderma, I just want to clarify which one is highly suitable. So that's why we have taken multiple assays to prove that rhizosporic trichoderma has a superior soil adhesion and soil application capability over trichoderma, sir.
Yeah, what is the efficacy level? Because that is also most important thing, no? Whether you have to go the efficacy of that particular species.
Efficacy, sir, it is, sir, within 96 hours it will grow very quick, sir, compared to other trichoderma. Usually trichoderma, it will take 7 days to grow full plate, whereas this culture will grow in 96 hours, sir, which means 5 days it will go full plate with pigmentation, sir. So which means that it reached the lock phase and it will start producing the pigment and spores also.
Okay, okay, thank you. So I think good to learn these things. Thank you very much.
Thank
good
you,
work.
sir. Thank you, sir. Thank you.
Okay. So next is, I think, Mamidi Sujan from Alpha Institute of Biotechnology. What's an Alpha Institute of Biotechnology?
Good afternoon. I'm Sujan. I work at Hudson Alpha Institute for Biotechnology US, and I'm scientist affiliate at Missouri, Columbia as well, and an adjunct professor at Angro, India. I'm here to talk briefly about a technological advance on identifying genes, because end of the day, susceptibility is a thing that has to be attained through multiple ways, and reducing chemical usage is one. And for that, we know fungicides are short-term costly, but at the same time, it doesn't meet our sustainability goals. So to counteract this, we need to have resistance varieties, which are generally long-term, eco-friendly, and farmer-friendly. So for that, we have designed a pipeline over the years, wherein we have your QTL Jiva studies, where we identify some regions of the chromosome. But from that, we do identify some diagnostic markers for molecular breeding purposes. But not necessarily given the time durations, it takes much time to develop a new variety. So we want to go with further identifying candidate genes, validating them, and using gene editing technologies to get a required variety faster and easier. So the first thing is why white mold matters. It's in common bean Phaseolus vulgaris. It's a major protein source for Latin American and African countries. And in general, biotech stresses cost about 20 to 30% losses. And this white mold particularly causes 30 to 60% yield loss, and sometimes up to 100% as well. And this is caused by Sclerotinia sclerotium. It has a broad host range and it can survive in soil for years, favored by cool and humid conditions. So for us to develop a hybrid variety in general, we are prone to breeding complexity because this trait is polygenic and we have QTLs spread over all the chromosomes. And it also has a strong genotype by environmental interactions. And we need both disease avoidance and physiological resistance to develop our new complex varieties. So it's difficult to do a classical breeding approach. And one other problem with developing a variety easily is the genetic diversity of common bean. We have 2 domestication centers, Mesoamerican and Andean. It has strong population structure and has many market classes. On your right, you can see some of the kinds of the market classes: blacks, navies, kidney beans. Each one is different. Each belongs to a different land race, different cultivar, kinds of multiple things. And at the same time, it has high allelic diversity. And for this population structure to establish, both the gene pools, the wild gene pools, diverged approximately 165,000 years ago in Mexico region, and followed by domestication independently in each one of them at approximately 8,000 years ago. It took approximately 1500 years for the domesticated, and further within these land races we have many gene pools as well. And we have been using QTL mapping from a long while, where we use biparental populations. We identified multiple QTLs, but they're prone to low resolution and they're difficult to deploy in breeding. So we were looking for integrative genomics. So to start with, we went with meta QTLs, where we combined QTLs from multiple studies, and we were able to identify some stable genomic regions. And with this, we were able to narrow down the confidence intervals. And this is a table that lists all the QTLs from 37 QTL studies over 14 real populations. As you could see, like most of the chromosomes, at least 7 of the chromosomes of the 11 have QTLs. Of this, the ones we are interested right now are 2.2, that has an R square of 31%, followed by 8.3 and 7.1. These are the 3 we started with. On the right, you can see there are many genes, 100, 200. So whenever we want to do some candidate gene analysis or downstream using CRISPR to identify, to confirm the gene or any other thing, working with 200 genes is really hard. So we need to narrow it down to maybe a couple of genes. So for that, the first approach we started is called as a bulk-seq approach. So basically, we have a distribution of our real population. We take the highest bulk and the lowest bulk. And using sequencing, bulk sequencing method, we identify the variance among both the pools and narrow down the QTL positions. So as an example, with 7.1 and 8.3, first we did like fine mapping to narrow down the QTLs, and the final table shows the things. Looks a simple table, but a lot of work goes into it. Basically, we had about 40 million reads that went with this, and we had approximately 2x coverage for 10 samples each roughly, and out of which we have 175,000 SNPs and about 280 are fixed SNPs, or 300,000. Fixed SNPs are only we have one allele in resistance pool and the other allele in the susceptible pool, and using the distribution of them, we are able to narrow down the QTLs. Say for example, with 7.1, we are close to roughly 1 MB, whereas 8.3, we are close to 3 MB, which was previously regular QTL analysis, meta QTLs, it was like 5 to 10 MB. This is a pictorial representation of the same. And in the same way, we had a 2.2 QTL. This kind of initial classic QTL mapping, we had in one of the population roughly 7 MB, and we reduced it to 1 MB. So this is the first stage of doing things. And whenever we combine multiple approaches, some approaches can detect a few things, whereas the other approaches can't detect the other things. So this is regarding WM 2.2. So we got the 3 major QTLs pinpointed to 1 MB with just a few handful of genes. And with this approach, we were able to even identify candidate genes based on the non-synonymous substitutions. Given limited time, I just don't want to go with them. So what's next? So we got some pangenomic data right now, which we are working with. Our idea is to identify the structural variations, presence or absence of genes, core genes, shell genes, and all those things. And within these regions, are there any genes that are missing or that are causing resistance or susceptibility? And that's better for allele mining. And our next steps would be like functional validation using CRISPR and gene editing. And we want to edit the susceptibility genes, and if that works out, that would be an easy way. Of course, not as easy as I mentioned it. It leads to accelerated trait development. So with this approach, our idea is to reduce the fungicide use, stabilize the yields, lower production costs, and develop climate resilient cultivars. Thank you. And I would like to acknowledge my 3 groups at Washington State University, Hudson Alpha, and North Dakota State University for all the work. Thank you.
Thank you. Thank you. Yeah, thank you very much, Dr. Mamadi. As I mentioned, a good point, particularly with respect to the pangenomic studies. Do you have any lead on that, what the resistance genes and the susceptible genes and in different type of this?
So we're still working on that. We just have sequence out of the sequencer. So we have about 40 samples that we sequenced to depth. So it takes a bit for assembly and trying to get to that.
Okay. Okay, thank you very much. Wonderful talk. Yeah. So next we have Dr. Bhavani Shankar Satpathy, the millet-based cropping system under ANFED.
Hello. Good afternoon to all. Respected chairman, co-chairman, reporters, distinguished delegates, और मोर प्रिय कृषक भाई और भौणी माने. Today I will just present a small work done by us, just the impact of Odisha Millet Mission on sustainability of poor and marginal farmers of Odisha. In Odisha, rice, maize and millets are the major cereal crops. We see millets gradually in 1883, millets area is decreasing due to the comparison of the maize. Among the millets in Odisha, finger millet is the major crop, followed by small millets, sargam and bajra. Odia re kaile apangar mandia, suwa, kangu and jana. Most of the millets, particularly mandia, that is the finger millet in Odisha, grow under rain-fed condition, irrigation also limited. See, this is the decline area under finger millet mainly due to the crop diversification to the values, that is high value crops like maize and cotton. In 2017 and 18, Odisha government feel that we have to revive our traditional crop millets. For that, Odisha government has implemented Odisha Millet Mission. Now, these three Anna Abhijan to just revive the millet cultivation in Odisha. Now, at present, Odisha government has covered all districts under millet missions. Just we have done a small study the impact of the Odisha Millet Mission on the sustainability of the agriculture in Odisha. We collected primary data covering more than 11 districts, 26 blocks and contacted more than 1,000 farmers. This is our field with interaction with the Ganjam farmers. This is interaction with our Rayagada and Gajapati farmers. This is Malkanagiri and Navarampur farmers. This is Koraput farmers. This is Sundaragad farmers, Balangir farmers, and during the summer crop Mayurabhanj and Ganjam farmers. And the millets in Odisha, we collected sample from the sea level to up to 900, more than 960 meters sea levels, and the soils are mostly sandy clay and sandy loam soils. The impact of the millet mission, you see, just in 2006, earlier gradually decreased up to 2016. On the implementation of millet mission, the area under finger millet is increasing, area production and productivity increasing. Similarly, rabi crops also increasing. Similarly, in the small millets, particularly little millet and foxtail millet, area production and productivity is increasing due to millet mission. Similarly, sorghum slightly increasing, but that is not so much impact on sorghum crops. Similarly, no much impact on the bajra crop, but productivity is increasing both the crops. We collected the data from the farmer. This is the farmer's perception why productivity of millets increase in Odisha. That their perception is mainly due to crop diversification from direct seeding to transplanting among the transplanting, that is millet system of millet intensification can help in the increased productivity. Next to use of the quality seed, third one use the organic. We also estimated based on the primary data and second data the resilience. We have quantified the resilience of millets. The resilience of we have considered that is the 4 agglomeratic climate zones, 7 districts, and we have taken the data with 26 years of yield data and 26 of rainfall data, and we have considered, we estimated that the drought year, we compare the yield loss in drought year and normal years. This is the heat map. We found that is the rice is highly susceptible to drought, followed by maize, as compared to whereas the millets are more resilient across the all district as compared to rice. So rice-based crop production system is highly vulnerable to drought as compared to millet-based cropping systems. We also, when doing our visit, we found that in Ganjam district, Sergad and Sankamunde and Patrapur, 2024 was normal year, whereas 2023 was drought year. We collected data from the more than 300 farmers regarding the yield, everything, and we found that is significant during the drought. There is a significant reduction in rice yield, whereas in millet there is a very less reduction, and climate resilience index is high in case of the finger millet. Similarly, significant reduction in the maize, but horse gram and green gram. Among all the food crops, finger millet highest resilient index. We just see also the comparison resilience index of this crop, very maximum swing, maximum susceptibility with the rice crop, and maximum resistance is the finger millet, followed by horse gram and green gram. You just see during our field with Sundargarh, you just take one field, just adjust into 2 fields. That is a drought year in case of Rajgampur block 2024. And you just see rice crop completely failed. In adjustment to that plot, finger millet is in very good condition. There is no loss in the finger millet condition. It clearly evidence that the finger millet is more resilient, more tolerant to droughts. We also collected the farmers' perception, how farmers' perception relate to resilience of the cereal crops, rice, maize, and finger millets. We just see, and in case of finger millet, but the tolerance to drought is high. Similarly, input requirement is low, that the cost of cultivation is low. We have taken the return per unit cost low. So overall, the finger millet is resilient to farmers. Hello, as compared to maize and rice. We also, the crop diversification, we also studied the impact of planting material among the yield. That is, we found the system of millet intensification, the highest yield, followed by the line transplanting, followed by the random transplanting. It gives significantly higher yield than the direct seeding. Similarly, also we estimated from the primary data the physical water productivity, that the crop established method has a significant impact on the water productivity in case of physical water productivity, economic water productivity. Also the grain, similarly also Rabi season also crop establishment has significant impact on water productivity, water use efficiency. We just also the crop establishment has significant impact also the gross income, net income, and BCA ratios. We also studied the different cropping systems prevailing in the Odisha, particularly Ganjam, Gajapati, Rayagada, Koraput, and that are highly diversified with the millet-based cropping systems. And we studied the cropping system of the Ganjam state, particularly finger millet followed by horse gram and finger millet followed by the green gram. We found that the cropping system based, suppose the Ganjam district farmer, they are harvesting this finger millet by September, and with the available of winter rainfall and residual soil moisture, they are taking another crop of horse gram and or that is the green gram, so that there is increasing in the water use efficiency, water productivity, sustainability. By that also they are maintaining the soil health and this. Okay. On that basis, we are recommending the different millet-based cropping system for different districts. These are the just multiple and multi-tie cropping of the farmers. This is the live finger millet value chain. Also, we found that the millet-based cropping system, women are more involved than the men, sir. This is our conclusion. But our recommendation is in millet mission, we have to go for the cropping system more instead of considered by single crop only. Thank you, sir.
Okay, thank you, thank you. So, Dr. Co-chairman, have any question related to this one? Because as you are many of these things, yeah, please.
Sir, we also have done the waterproof. We will submit recently. We are going to submit project. Sorry, portal.
It was a good presentation, and I have a query about it. Whether you have calculated the cost of production?
Yes, sir. Yes, sir. We have calculated the cost of production in both Rabi crops and Kharif crops. This is small presentation. That's in my report. I am giving everything, sir.
How much it comes actually per hectare right now?
Sir, it is varies from the farmer to farm, district to district also, sir. Actually, we have collected data from the three.
It is completely government sponsored, starting from sowing to procurement.
Yeah.
So how much it? Because recently one team from Maharashtra, they visited Koraput and those area, and they are studying actually that cost, and they said ki this cost is very high actually. So per quintal, how much it comes
Actually, sir, when we will take the cost of the labor, then it exceeds 30,000, 40,000 per hectare. But normally, tribal people, they are using their own family labors. So Odisha government, when they are giving 4,000 per acre, only by the operational cost, excluding the level cost.
We have to include this procurement cost also.
Yeah. Very high that I have taken care of. Our economist is also calculated.
Okay, thank you.
And we are submitting report within this month. By February, we are submitting report. Okay, thank you, sir.
Thank you.
Next presentation by, I think, Dr. Preeti Singh in the online. Somebody there? Dr. Preeti
Sir,
from
yes,
IHR?
yeah. Good afternoon, sir.
You're there? Okay. So make it okay, go ahead. So evaluation of bioactive constituents of Garcinia. Quickly for 7 minutes only, Dr. Preeti. Hello.
Yes, sir, I'm trying.
Okay. Share the slides, yeah.
Is it visible?
Yeah, go to the slide presentation.
Yeah. Respected chair,
No, it is not visible. It is not visible fully. You go to the slide presentation mode.
Sir, I have already done
We are
that.
seeing your desktop only, but not that presentation. So you have to click in the presentation and share. Somebody.
Dr. Preeti, I'll share and again share.
I'll share now. Is it visible, sir?
Not yet. So you have any problems here itself? Somebody is working here? कहाँ है भाई कोई अच्छा नहीं क्या ये पटे प्रॉब्लम कौन होची तो शेयर किसे तो यार कोर्ची तारा स्क्रीन तो आसची हाँ yes go to the yeah it's okay go ahead.
Okay, sir. So I am senior scientist working at IHR Bangalore, and this work is on Garcinia indica, that is the evaluation of bioactive constituents of Garcinia as a potential source of hydroceptic acid. anthocyanin, and phenolic compounds. So this Garcinia indica, it is native to Western Ghats, though it is medicinally very important, but it is a underexploited food species, and not much work has been done in this plant. So here we try to evaluate different bioactive constituents in this plant. In Ayurveda, it is characterized to have antibiotic properties, antihistaminic properties, and so many can cure so many other diseases also. And this fruit can be used as a spices, and it is very famous in especially in Maharashtra region. Here they use a famous drink called as cold filling, and this impart a sour taste to food. So this sour taste is mainly due to presence of HCA, which is an important antioxidant agent. And because it is having this component, it is widely used in anti-obesity medicine. This fruit of this plant is purple red color, and it is mainly due to presence of these anthocyanin compounds. So in this work, we have collected different extractions from Karnataka and Maharashtra region, and we have evaluated them for different bioactive compounds using normal standard Woodcock method, SPC, and LCMS method. So we have analyzed for HCl content, anthocyanin, phenolic acid, and flavonoids. So when we analyzed HCl content, it was found that HCl content varied from 15 to 22%. Usually, Garcinia composia, which is another source, which is the prime source for HCA, and industrially, this is the plant which is used for HCA extraction, has 10 to 30%. But here, in our study, we found this HCA content up to 22%. So our extraction can also be used for commercial HCA extraction. We also did total phenol, flavonoid, anthocyanin, and antioxidant activity. And some of the experiments they were very rich with phenolics, and they showed very good antioxidant activities. When phenolic profiling was done, we have identified three phenolic acids, and major compound was fumarate acid, followed by orthophosphatic acid, protopatric acid, etc. We know this that trans was commercially exploited in food and pharma industry because they have various biological activities. And here, fumarate acid, it is the well-known isokinase inhibitor. And because these fumarate acid has this property, it can inhibit the melanin biosynthesis. So these tyrosine inhibitors, they can suppress the browning of fruits and vegetables, and also melanin production in mammals. Experimental evidence suggests that this p-coumaric acid is more potent than other conventional tyrosinase inhibitor, like arbutin and kojic acid. Usually in cosmetic industry, this arbutin and kojic acid is used for skin lightening agent. But here p-coumaric acid is more potent, so that can be also used in cosmetic. So here in Garcinia indica, p-coumaric acid content is quite high, which is 3.3 to 8.1 mg per gram. It is one of the richest source, so it can be used for commercial extraction. Then further flavonoid profiling was also done, and 12 flavonoid compounds were identified. These flavonoid compounds also, they have different biological activities. So one of the major compound in Garcinia was naringenin, followed by epigenin and quercetin. Naringenin, it is a well-known, it has an antimicrobial activity. So it can inhibit cell wall synthesis in bacteria and fungi. Because it has antimicrobial property, it can be used as a potent and safe natural preservative for food and pharma industry. So when we compare our results with USDA databases, then we found that the richest source as per USDA database was in grapefruit, pomelo juice, Mexican. In our result also, we got very good content for naringenin, which is quite comparable with other crops like grapefruit, pomelo juice, Mexican oregano. So they can be also serve as a good source. Further, we also did the anthocyanin analysis. So the purple red color of Garcinia is mainly due to anthocyanin. And because it has a very dark red color, so it can be used as a replacement for synthetic colors. And in our all the accession, it showed very high amount of anthocyanin content. So it exceeds the reported in most of the fruits and vegetables. So it can be also used as a promising source for natural color. Anthocyanin profiling was also done, and 38 anthocyanin compounds were identified. They belong to 4 major group, and major anthocyanin was cyanidin 3 stambuboside and cyanidin 3 glucosides. Further, we did cluster analysis and we generated heat map. This heat map, it helped in choosing the best accession with optimal quality parameter. Here we found that IAHR2 was the richest source for anthocyanin, comedic acid and naringenin, which is clearly visible from this heat map. So now coming to conclusion, in the present study, principal bioactive compound present in the fruit of Garcinia indica were analyzed, and data on the biochemical composition of Garcinia indica will help in promoting this fruit as a promising source of hydrocitric acid, anthocyanin, comedic acid, and naringenin. And overall, the present study highlights Garcinia indica as a rich, new, and sustainable source of bioactive substances with food, pharma, and other industrial applications. Thank you.
Thank you so much, ma'am. There's no questions. So we move ahead with Dr. B. Varalakshmi from ICAR IIHR Bangalore. She will also be presenting on online, and she's already there.
Yeah.
Can we connect?
Yes, madam. Yeah. Good afternoon, sir. Please allow me to share the screen.
Also, ma'am, it's just 7 minutes.
Yes, I do. I'll share my, no. You have to allow me to share. No, other participant. Dr. Preeti, please unshare. Dr. Preeti?
I think she's not there.
Or it can be done from there.
Yeah, I have unshare.
Yeah, please unshare. Okay. I'll go to slide mode. Good afternoon, everybody. Respected chairperson, co-chairperson, rapporteurs, and all the delegates. I'll be presenting my paper on identification and evaluation of advanced guidelines for powdery mildew resistance. Coming to a brief introduction, bitter gourd is an important cucurbitaceous crop economically as well as nutritionally. It also has got medicinal and dietary value, but its production is severely constrained by the presence of powdery mildew, which is caused by Podosporium xanthe. As high as 50% of yield losses have been reported. So this disease occurs like powdery colonies on the leaves, leading to premature senescence and also reduced photosynthetic efficiency. But we have control measures to control, keep disease under control, but they all will have a negative impact. Like they will provide temporary relief and also raise concerns regarding environmental safety, fungicide residue, and also in the fruits, and increased input costs and pathogen resistance also will be a problem in future. So better to tackle this particular problem with the genetic resistance, which is most sustainable, cost effective, and environmental friendly approach. So with an objective to develop one powder mildew resistant variety or hybrid in bitter gourd, we started the breeding program with screening lot of germ blossom, and we identified one IHR 144-1, which belongs to Momadica charantia variety muricata, which has got good resistance for this powdery mildew. Using this particular resistant source, we have made crosses between advanced breeding lines and this resistant source, and the further populations were advanced till F7, and then in the F7 generation we made selections, and out of which 26 shortlisted advanced breeding lines were screened under natural epiphytic conditions for powdery mildew resistance. Out of this study, we found three best lines, that is PMR 24-6, 24-7, 24-14, which exhibited very high level of resistance. And this resistance we wanted to confirm under artificial inoculation conditions also. So for that, we have raised all these lines along with the susceptible lines, Arka Harith, 156, 146, two other lines under the polyhouse conditions. And we sprayed the inoculum at the concentration of 2 into 10 power 6 spores per ml. After 1 month after sowing, all these plants, including susceptible, resistant, all the lines were sprayed with the inoculum. And also a second spray we have taken after 2 months of the first spray. Then once once the symptoms are started, we started recording the disease score every week. That is weekly intervals we have recorded, and we worked out PDI as well as area under disease progress curve. Under natural epiphytic conditions, there were no symptoms found on PMR 24-6, 7, and 24-14. But under artificial inoculation tests, PMR 24-6 and 7 maintain the resistance, each recording a percent disease index of just 11.1 and a AUDPC value of 302 and 325 respectively. In contrast, the one which we found as 24-14 under natural conditions as resistance showed moderate resistance under the control conditions with a PDI of 22.9 and AUDPC of 465. Whereas the susceptible check, Arka Harith recorded a PDI of 89 and AUDPC of 2871 under natural conditions, and a PD of 100, almost 100, entire crop has been affected by the disease with an AUDPC value of 2103. So these findings, under this, we could shortlist this PMR 24-6 and 7 as a durable source of resistance, which has shown consistent resistance under natural as well as artificial inoculation conditions. So these findings are expected to strengthen the Bitogo breeding program, especially for the powdery mildew resistance. And this will also facilitate the development of durable resistant cultivars and thereby it contributes to sustainable production systems. So if it's coming to the table, the scores of these particular lines, Sarkaharit is the highly susceptible line. You can see, you can better appreciate this here by this data. 24-6, 7, 14 have recorded 0 incidents under the field conditions, whereas they were resistant under artificially inoculated conditions regarding PDA of 11. And this 22 is 24-14 has recorded 22.96 PDI. If you see the pictorial representation of all these resistant lines, A represents 24-6, this is 24-7. These two are the highly resistant lines shortlisted, and C is the BTG 144-1. That is the resistance source used in developing these advanced breeding lines. Then this is Arka Harith is the E, which is highly susceptible. And this is also another advanced breeding line, which is very highly susceptible. You can see the damage caused by the powdery mildew under controlled conditions, whereas the resistance lines sustained the disease and they were free from the disease. Thank you. I would like to thank all my collaborators and also our dear director, Dr. Behra, for his constant support and encouragement. Thank you.
Dr. B. Varalakshmi?
Yeah.
I think there won't be any question as of now. We can discuss things later. Thank you so much.
Thank you, ma'am.
So we move ahead with Dr. Sauren Das, Indian Statistical Institute, Kolkata. I'll be talking on mangrove ecosystems as blue carbon reservoir, mechanism and impact on climate change mitigations.
Good afternoon, everyone. Actually, climate change mitigation is a robust word, but we all knows we cannot define as the single word the climate change. Climatic condition is very much hazardous to the present situation day by day. Only carbon is not the only party. There are several gases are there, but plant can reduce carbon content from the atmosphere. It is almost easier to say that during photosynthesis they can consume huge amount of carbon as any vegetation type of vegetation. But why the coastal vegetation is important? That is the question. Coastal vegetation comprises within the line of meridian 23 degrees south and 23 degrees north, and they consume much more sunlight. That means PAR, photosynthetically active radiation. They consume that much one, and freely they can do photosynthesis, and in that process they can mitigate carbon dioxide to some extent. But there is a problem. Too much power is not required for the photosynthesis, but the coastal ecosystem has already received maximum sunlight throughout the tropical world. That's why the photosynthesis system itself, it is a thermodynamic system, it's exothermic process. That's why the continuous process of the photosynthesis is not done there. But the amount of canopy, dense canopy rather, it has significant amount of carbon dioxide absorbent and mitigate the atmospheric carbon dioxide. Let's go. The blue carbon. Why it is blue? Okay. Blue carbon is the term for carbon dioxide captured and stored by the world's ocean and coastal ecosystem. The main coastal ecosystem responsible for their mangrove forest, tidal marsh and seagrass, collectively known as coastal blue carbon ecosystem. Mangrove vegetation and blue carbon. Mangrove forests are exceptionally significant for blue carbon sink, playing a critical role in the climatic change mitigation. Why? High carbon sequestration rate, one thing. Second is long-term carbon storage, how it is possible. Carbon sequestration rate, easily you can define that dense vegetation can always capture higher percentage of carbon dioxide from the atmosphere, but how can they store in long term? The majority of the carbon captured in mangrove is stored by below the ground in their dense, waterlogged and often oxygen deprived. That means anaerobic condition. Carbon is also stored in the above ground biomass such as trunk, leaf and roots, but soil accounted for 50 to 99% of the total carbon sink. This storage can last for hundreds or thousands of years, making a high effective long-term carbon sink. What is the difference between this mangrove and other terrestrial vegetation? See, other terrestrial vegetations grow on a sediment that is not the dynamic condition, but mangrove forest thrives on dynamic condition as because there are 2 inundation in a day and 1 maximum inundation maximum inundation per month. This dynamic flow of water and go away, they produce newer sediment every day. And mangrove root system is unique for capturing those carbon. But in the any terrestrial forest, their root, leaves, plant parts fallen in the substrate, their decomposition occurs there, and carbon is backing back to the atmosphere again. Please help me. Massive carbon stocks, mangrove ecosystem accumulates significant total amount of carbon. Estimated suggested that mangrove soils store large quantities of carbon per hectare. Mechanism of sequestration. Mangrove trees photosynthesize, absorbing from the air and convert it into the organic matter. The complex root system like prop root, pneumatophore, trap and accumulate organic material much more than any terrestrial forests. And sediment carried by tides and the rivers. The flooded, low oxygenated condition in the soil prevents the organic matter from decomposing quickly, which is process that would otherwise release back to the atmosphere. Now, our mangrove cover within India, mangrove has shown a slightly net positive trend, but not yet satisfactory. Though recent data indicates this slight dip in specific regions, mangrove forests are critical for coastal protection and carbon sequestration. Two organizations works on this carbon sequestration, mainly climatic mitigation. One is MISTI and one is SAME. Mangrove Initiative for Sustainable Habitats, Tangible Income. This is Government of India. And SAME is a private organization, Sustainable Aquaculture in Mangrove Ecosystem Initiative. Mangroves cover in India more or less 4,992 square kilometer, constituting 0.5 of the country's total geographic area. Major mangrove ecosystem, the Odisha in Bhitarkanika, Andhra Pradesh, Godavari, Krishna Delta, Gujarat, Kerala, Andaman, and the Sundarban. Mainly Sundarban is the largest single block, as we consider in the Asian, largest mangrove block in the world. The state-wise area like this, somehow all the places are showing somehow as 2013 to 2023-24, area is somehow increasing, but somewhere also decreasing because of the industrial revolution, industrial encroachment, in man intervention, all is fatal to the mangrove vegetation is now. Now, the mangrove formation average sequestration rate in Sundarban is 30 to 50 million tons per hectare, and highest in the all mangrove formation in coastal. Total Kerala is 139.28 hectare carbon per hectare stock is there. Largest contiguous block is Sundarban. High soil organic carbon is there. Second largest dominated by Abyssinia and Exocaria. There are two mangrove species are there. As because mangrove species are different from each other by their physical structure or their some photosynthetic affinity or some anatomical or some genetic factor. They are all mangrove are mangrove plants, but they are from different family and different species. they have some specific adaptive rate. That's why in Sundarban mangrove, Sundarban is named as Sundarban by a plant, Sundari, Heritiera fomis. But if you go now, hardly you see a healthy Sundari plant. Why? The Sundari plant, we observed in my laboratory that Sundari plant somehow anatomically, physiologically, and also genetically weaker than other mangrove plants. That's why Sundari and other some mangroves, Heritiera, Xylocarpus, Nipa, they should take care separately to regenerate. Otherwise, it cannot be possible. Carbon pool in Indian mangroves. Carbon is sequestered across the primary pools. In Indian mangroves, the soil organic carbon, the largest reservoir, often accounting 70 to 80% of the total carbon ecosystem, above the ground biomass and below the ground biomass. Above the ground biomass, it is from the stem, branches, and leaves. But more important is the below ground biomass, the root, taproot, proproot, etc., which contribute rhizophora maximum. Soil organic carbon. Soil organic carbon buried in anaerobic mud sediment, dead wood and litters, smallest but active pools. Species-specific sequestration potential difference have varying efficient capturing of carbon. Abyssinia in Kerala and Odisha, rhizophora, notable extension of their root and enhanced below ground carbon burial. It is dominated species in Andaman. Sonaraceae is rapid used in forest formation in younger plantation. Current trends in conservation rate. Conserved natural mangroves in India sequester 2 to 3 times more carbon than rehabilitated or anthropogenic interfered patches. Why? See, the older formation of the forest always have some dense patches than the rehabilitated or anthropogenic interfered patches. In West Bengal, mangrove area 2.144 square kilometer, and Gujarat, there is just a comparison to the West Bengal and Gujarat to mangrove patches. Total carbon stock 26.62 teragrams of carbon, and it in Gujarat. This is because of their huge density and area. Now, sequestration rate annual description. See, the tropical forest, Indian tropical forest, where annual rate of sequestration is 1.2 to 2.5 tons per hectare per year, whereas any mangrove forest 6 to 8 ton per hectare. That means it is higher than 3 to 4 times of the any tropical forest formation. In a typical dry forest, when leaves or branches fall, they decompose quickly because oxygen is painful. This release of carbon dioxide back into the atmosphere, but mangrove thrives in the waterlogged anaerobic condition where oxygen is less. Their microbial action is much reduced. Because of this oxygen fuel decomposition, no oxygen to fuel decomposition, that carbon stays locked into the ground for countries over millennia. Mangrove does not just store carbon in their leaves and roots. They are unique because deep peat, they build up thick layer of carbon-rich peat. Sediment trapping, their complex root system act like as a comb, trapping organic debris following from the riverside, effectively stealing carbon from the other ecosystem and burying it within. Thank you.
Thank you, Dr. Very nicely that he has mentioned about this mangrove in that way. But again, you have to see that the carbon sequestration, the different fruit plants, because you have a different orchards are coming in the big way.
Right.
So how is the difference will be there? That is one of the things. Then
Actually-
second thing, just one other thing is that you have mentioned that in the West Bengal, we have a more carbon sequestration than the Gujarat per hectare basis.
Per
Okay,
hectare.
you have put it this one.
That is
So
only because of that the dense patches of the mangrove plants
Okay.
is higher in Sundarban region than the
Okay.
Gujarat. That is one point.
Okay.
Second, species specification is there.
Hmm.
In Sundarban region, more rhizophora species whose root
Okay.
system is much more thicker and
Okay.
comb-like.
Okay.
That's why.
So that depends also the canopy and the
Right.
planting
Yeah.
density and all the things also
on
depends on this.
terrestrial plant, their root system not much more widespread.
Okay.
In any terrestrial forest.
Hmm.
And second thing, any terrestrial forest has a
Use the mic. Mic.
any terrestrial forest has is the is the older one rather than the mangrove one. As because mangrove forest regenerate every day through its not mangrove forest but mangrove forest
Mangrove
sediments
plants, yeah.
regenerate every day due to their inundation process. That's why it is a newer one.
Okay. Thank you, Dr. Sir. Very good presentation. Thank you very much. Go ahead, next, please. Yeah.
Thank you so much, sir. Now can we have Dr. Anandita Karmakar from Bidhan Chandra Krishi Vishwavidyalaya, Kolkata, over the online presentation she'll be presenting. If she is there, please connect to her. Yes, she is.
Yeah, please go ahead, Dr. Anindita.
Yes, sir.
Share your slides.
Also, ma'am, you're not audible.
Yes, sir.
All right.
Am I audible, sir?
Yes, you are, ma'am.
Okay. Then, good afternoon to all.
Make it a slide show.
Yes, sir. Sir, is it visible, sir?
Yes, please.
Yes, sir. Okay. So, good afternoon to all. Today, I am going to talk on the topic of mathematical modeling of drying kinetics and dehydration behavior of green peas. Green pea is an excellent source of nutrients as it contains a great amount of protein, fiber, different types of vitamins and minerals. But green pea is highly perishable as it contains a huge amount of moisture. That's why preservation is needed after their harvest. And different preservation methods like canning, drying, etc. are used to increase their shelf life. So among all the different preservation methods, drying is the most important and common one as it reduces moisture to prevent spoilage. It also reduces post-harvest losses and enables long-term storage. Apart from them, it also adds value to the product. So there are different types of drying. The most common one is convective drying, in which drying is carried out with the hot air. And in this case, the study of drying kinetics and model fitting is very important as they are required to optimize the different drying factors like temperature, time, airflow velocity, etc. And they also help in design of different types of dryers. Now, dehydration becomes important quality parameters for dehydrated products because we need to rehydrate the dehydrated vegetables before their consumption. Then dehydration capacity depends on different factors like drying conditions, product type, drying temperature, and ratio of the rehydration water to that of the dried product. And modeling of dehydration kinetics is important as it relates experimental results to the physical laws of water absorption behavior. So with that perspective, this study was taken to study the different drying kinetics of green peas at 3 different temperatures and also to study the dehydration characteristics of green peas. So the experiment was carried out following the steps presented in this flowchart. First of all, raw green peas were procured, and after cleaning and sorting, they were blanched at 85 degrees centigrade for 5 minutes to inactivate the enzymes. And then drying was performed at 3 different temperatures of 50, 60 and 70 degrees centigrade, following which dehydration was carried out at two different temperatures of 40 and 50 degrees centigrade. So from the drying data, moisture ratio was calculated with the help of this equation, and then the drying data was fitted to these four different drying models, namely Bayes model, Newton model, Wang and Singh model, and two-term exponential model. All these model contains model parameters or model constants, one or more than two. Then rehydration ratio was calculated with the help of this equation, that is the ratio between weight of rehydrated sample to the weight of dehydrated sample. And then Pellet's equation was used to model the dehydration characteristics, and the equation for that model is presented here. Here two constants are there, K1 and K2, and which represents Pellet's rate constant and Pellet's capacity constant, respectively. Now coming to the results and discussion, there are three graphs presented in this slide. First one is moisture content versus time. The second one is drying rate versus time, and the other one is drying rate versus average moisture content. So the moisture content of green peas reduced from 240 to almost 10% within the time of more than 6 hours. So initially, the drying performed at a higher rate, and which slows down as it approaches towards the equilibrium moisture content. This is evident from the second graph also, where this is presented between drying rate versus time. Initially, the drying rate was higher, and then it becomes slow and almost constant as it approaches towards the equilibrium. So from the third graph, that is drying rate versus average moisture content, it can be seen that drying rate is constantly decreasing with moisture content. That means drying was accomplished at falling rate period. That means the moisture movement from the product was governed by the subsurface evaporation. Then during the mathematical modeling, all the drying data was fitted to the models, and it was observed that Pellet-Page model showed the highest correlation coefficient, that is R square, and lowest RMSE. That's why Page model was selected as the best fit model to describe the drying behavior of the green peas. Now, next, coming to the rehydration ratio, then this graphs represented the rehydration ratio with time, and it was observed that this dehydrated green peas took almost 2 hours to reach to the equilibrium moisture content during the dehydration process. And it was observed that at 70 degrees centigrade, that the moisture absorption rate took highest time. And these are the parameters for Pellet's equation, and it was observed that all the R square values is almost close to 0.90, which describes that the Pellet's model can suitably describe the rehydration behavior of green peas. So coming to the conclusion, the moisture content of green peas reduced from 72% to almost 5%. Then Page model was found to be the best fit model to describe the drying behavior of green peas, and rehydration of the dehydrated peas continued for nearly 2 hours, and Pellet's model effectively described the water absorption behavior of green peas. So thank you, sir. That's all, sir. Hello.
Hello?
Yes, sir.
So what will be the optimum temperature for the drying of this garden pea and duration?
And that is, sir, 60 degrees centigrade.
And duration?
And it's almost 6 hours.
Okay. So what is the significant difference is there? Because you cannot find that any statistical parameters for that one. Any significant difference with respect to temperature and the rate of drying? Suppose you have 50 or 60 or 70, what will be the actual difference in the statistical way? Any significance are there with respect to temperature?
Of course, sir, for 70 degrees centigrade, time taken will be less, but it was not statistically defined. Actually, we did it in terms of
Sure, okay. So
drying
thank you
water.
very much. Thank you, madam. Thank you.
Thank
Go
you.
ahead, next presentation, please.
All right, now can we have Dr. Jyotirmay Lenka, ICAR IISWC, Sunabeda? As he talks on the topic seed spices for rural livelihoods in Odisha, evidence from on-farm demonstration.
Respected chairman, co-chairman, now I am presenting seed spices for the rural livelihood in Koraput district of Odisha. Actually, this is the need of the hour because means India is known for the land of the spices, and also the largest exporter of the seed spices is in our country. And the seed spices versus seed, if you see, said seed spices is the just subset of the seed spices, and in broader terms, if you see, the seed are the seed spices are the dried part of the all roots parts and leaves, bark of all the seed types. Because it is only confined to the mainly Rajasthan northern part of the country. So, but the climate, the cool and dry climatic condition is also in our state also. And we are now trying to, it is whether it is feasible to our condition also. According to the, so we have taken two objectives: to assess the feasibility study and to demonstrate and capacity building on seed production and organic cultivation and post-harvest technology in different parts of the Koraput district of Odisha. We have taken, sir, 8 crops in 2022-24. After that, we were in 20 coriander, fennel, fenugreek, azoine, nigella, cumin, celery, and dill. Cumin, celery, and dill is not conducive to our condition. So, in 2020, 24 and 25, we had taken 5 crops: coriander, fennel, fenugreek in different blocks, that is in 8 blocks of Koraput district: Koraput, Nandpur, Simliguda, Lakshmipur, Potangi, Boiparigoda, and Narayanpatna, with different villages. Number of farmers, so we have taken on-farm demonstration in 719 farmers. Now, also we have selected the site selection before. We have taken baseline survey. We have taken gender categories and economic status. We have taken all the site selection, and we have covered training to different stakeholder, farmers, FPO, and other stakeholder, with different package of practices, organic cultivation, post-harvest, and demonstration of different farm fields. We have given total number of 912 farmers we have covered, along with different 7 number of training programs we have covered on the crop production technology and demonstration. And also in 2025 also we have taken 35 number of farmers from the seed spices operation area, and also from the TSP, SSP, tribal subplan and scheduled caste plan. We have taken to NRC, ICR, NRC Ajmer to expose some of the number of demonstration and also improve crop production technology. These are the some of the different crop growth stages in 2024-25. This is the in Koraput region only, the land preparation and different stages of coriander, fennel, fenugreek, and different stages of harvesting stages of the different produce. These are the some of the results. So we have found that in different villages, we have found plant height, plant breadth, number of branches, seeds, and yield also we have calculated, so that we found a significant yield in different parts of the village of the Koraput district. In Gilliguma and Durkaguda area, we have also found a maximum number of that is 850 kg per hectare coriander seeds. And also we have taken different villages in Koraput district, that is for the different fennel crop also we have found that in Durkaguda and Gilliguma area we have found significant yield as compared to the other villages. In Durkaguda and Gilliguma area, the yield is also high in the Durkaguda and Gilliguma area, and also observed different parameters of the different crops. These are some of the publication we have, and also we have on-farm technology demonstration in ISET side also, sir. These are 6 number of leaflet we have already published in ODI language to disseminate to the farmers, sir. Thank you, sir.
Yes, thank you, thank you.
These are some of the conclusions.
Conclusions, what conclusion?
These are, sir, actually, sir, the growth of coriander, fennel, fenugreek, azwine and nigella was very good in different condition of the Koraput district, sir. And also we have on-farm demonstration already, sir, in 719 farmers, sir. And for scaling up, we have to 4 to 5 year or more training we need, sir.
Okay. One important thing is that also, as you mentioned, fenugreek and the coriander. This is the flavor is very important. Many of the things we are not getting the flavor as we got it in the early, before 30 years or 20 years.
Yes, sir.
So do you have any climatic or microclimate differences with respect to Koraput regions and what you have done it?
Yes, sir.
Because that is also very important. Microclimate is very, very important for this particular spices, seed spices particularly,
Yes, sir.
coriander and then you have fenugreek. So do you have any difference you find it?
Yes, sir. Actually, we have also standardized in our farm, also research farm. Also, we have standardized, and also we see that, sir, in green fennel, sir, our quality is much more good, sir, than Ajmer condition, means Rajasthan or that condition. But we have to revalidate again.
That's
sir.
why I'm telling now. You take it that data of the microclimates, okay?
Yes, sir.
Even the weather also, you have to very significantly changes will be there based on that. Okay? So
Yes,
you take it that particular data and correlate with this type of Ajmer condition. Then you'll find a lot of difference will be there.
Yes, sir. Yes,
okay, you are taking the same variety or you are taking the different varieties from us?
we have taken sir, same variety with Ajmer, sir.
Okay, thank
Okay.
you. So take it
Okay.
this one. Very nice.
Okay, sir.
Good
Thanks.
presentation. Yeah.
Thank you
Next,
so
please.
much, ma'am. So as of now, the last one for this technical session, Dr. Snehashis Rautrai, ICAR CISH, Lucknow. So please.
Yeah. So finish within 5 minutes?
Sir?
Within 5 minutes?
Definitely, sir.
Yeah, please. Thank you. Go ahead.
So, good afternoon, one and all, respected chair, co-chair, and all the invited delegates, academicians, scientists. So, I am Dr. Snehashis Rautrai from CISS Lucknow. Here I am presenting this topic. A native strain of Heterodaptitis indica from Indo-Gangetic plains, isolation, characterization, and field efficacy against bark-eating caterpillars in guava ecosystem. Bark-eating caterpillar is one of the major biotic hindrance in major of the fruit cultivations, and particularly these two species, Indrabella quadrinotata and Indrabella tetronis, they infest mostly around 13 fruit crops, multiple forestry species as well as some ornamental plants. So over a decade, if you see the major top 10 Indian states, the infestation is very much prominent in multiple fruit growing states of India. And the major infestation symptoms pertains to the tunneling and reducing the viability of the plant, overall plants, and ultimately affecting the plant vigors. So if we see the current management practices of this particular pest, so currently there is no level clump insecticides for this, to recommend for this management of this pest. And very common recommendation is the mechanical removal using wires or hooks, which is also very cumbersome to be adopted by the farmers in multiple times. And certain entomopathogenic microbes has been recommended, but certainly it has not gained the trust of the farmers because of this low efficacy. And cultural and botanical recommendation also, somehow it has been slower being in terms of its adaptability. So in terms of the thinking about the alternate pest management tactics, looking into the habitats of this particular pest, I thought this entomopathogenic nematode can be one of the better alternatives designing management practices for this pest. So I'm unable to change the slide. Can you help me? Yes. So over these 5 objectives, this investigation was planned initially to collect the soil samples over multiple locations of the Indo-Gangetic plains, and using Galleria, this wax moth larvae, to get certain strains of this native entomopathogenic nematodes, followed by molecular characterization using multiple markers. In vitro bioassays looking into what are the lethal concentrations of these nematodes, and taking into the field level, looking into its field efficacy, and to try to get certain formulations of these nematodes. So from western UPs, certain locations like Meerut, Saharanpur in central part of UP, from Unnao, Lucknow, Barabanki, and in eastern part of UP, Gorakhpur, as well as other locations, I try to collect certain soil samples, and utilizing this wax moth larvae, I try to collect certain strains of these entomopathogenic nematodes. So simultaneously, the culture of this wax moth larvae was also maintained within a certain artificial media to gain this continuous supply of this larvae. And in the utilizing the white trap methods, the infected juveniles were harnessed, and ultimately the pathogenicity of these nematodes were also tested. So I found total 3 strains of Heterorhabditis indica. Initially, based on the symptoms, it created this brickish brownish color. I found they are the species of Heterorhabditis indica, and with the initial bioassay, I found the strain 3 of Heterorhabditis indica. It was with low LC50 concentration, around 22.45 number of infective juveniles per larvae, so which I took to the next level. So initially pathogenicity I found 40 to 60 infective juveniles per 100 microliter. It was causing around 100% mortality in 2 to 3 days in wax moth larvae. And utilizing this ITS and 16S RNA, it was confirmed that the species was Heterorhabditis indica. And I tried to evaluate in the 3 different systems of this guava plantations in high density planting, elderly orchards, as well as the espalier systems. So I took chlorophypus as a standard check, and with the 3 different concentrations I found, around 500 infective juveniles per ml when I applied at the spot application. Over 2 to 3 days, I found the mortality percentage is ranged from 87.72 to 88%. Where in chemical treatment, the mortality percentage, it was 92 to around 93%. So based on this economical values of 500 infective juveniles per ml, I found the spot application, it was comparatively better as compared to others. So these are the certain host cadavers recovered from the field after these treatments. So typical symptoms pertains to the coming out of these tunnels, and the mortality I found around 2 to 3 days of the applications. And to confirm whether the mortality was exactly caused by these nematodes, it was again kept in white traps, and the infective juveniles also emerged out from these dead cadavers. So certain formulation initially I have tried to make a gel-based or talc-based, wood-based formulations, but initially I got a liquid formulation was comparatively better, but still I'm working more on this. And apart from these bark-eating caterpillars, I evaluated on certain other pests of these fruit ecosystems. I found certain better efficacy against mango stem borers, and in a simulated conditions also, fruit flies, against fruit flies also it has initially given something, but I need to take it to the field level. And against guava shoot borers also it has given a better result. So I'm currently designing a semi-automated EPN harvesting system, because harnessing this infective juveniles is certainly it is cumbersome and collecting. So a design is currently in the process so that the infective juveniles can be harnessed in a bulk, so that in a particular fruit ecosystem, the orchard ecosystem, it can be utilized in a better way. So it is a fast evidence of this effectiveness of certain this entomopathogenic nematodes against this bark-eating caterpillars. So further in a semi-synthetic media also, I have targeted to mass produce it, and automation also can be included in our plant. So I acknowledge the support and cooperation of the director CISH and our teams. Thank you so much.
Yeah, thank you, Dr. Sinasis. Dr. Maheshwari, do you have any, because it is the first phase of that one, particularly for entomopathogenic lab, and this one in nematode. So, is any?
Ma'am, the liquid formulation, the suspension itself, I counted and it was applied spot applications only.
But you have to take care that it is highly susceptible to UV and temperature.
Yes.
So time of application has to be seen. Either it can be in the early morning or in the evening hours for having maximum efficacy.
Absolutely right, you're right, ma'am. All the treatments I tried to apply in the evening hours so that they escape from the heat, the peak heat scorching time. So in the second half, nearly 4:30 or 5 p.m. that time I applied.
So along with the formulations, you can add some UV stabilizers and anti-desiccants, so to improve the formulation efficacy.
Right.
And you can also see the compatibility with other chemical pesticides, because in agro ecosystems, especially in the orchards, you will be spraying with other chemicals also.
Right.
So integrated pest IPM modules, as such, you can see how it is compatible with the other measures.
Right.
Okay.
Thank you, sir.
Thank you, ma'am.
Thank you. So suggestions you have taken, because you are the first time you are working, no? You are scientist there?
Yes, sir, I'm
When
scientist.
when did you join there?
In 2023.
So you contact Dr. Maheshwari. She is having a lot of experience in that way. So have some good programs with you. Okay.
Yes, sir. Thank you, sir.
So it's okay. Done it is, I think.
Yes, sir.
Yeah.
We will take a short break for the lunch and we'll resume the session at sharp 2:20.
So who will be the chairing, co-chairing that session, Dr.
I will just inform.
So just have who is there? So 1 minute. I think this I must thank to the organizer, because it is a wonderful session, and many of the senior people also there, like Dr. Saindas and Dr. Aji Agarwal. He mentioned the things in the baby corn, how it is for the Odisha, and particularly for pesticides inputs in case of increasing productivity. So that is also by Aji Agarwal Sahab. I think wonderful experience with respect to the learning also, with respect to the very good observations and the data they have analyzed with respect to the different system, whether it is in microorganisms, whether it is in the chemicals, botanicals, even many of the things also has been discussed here, even the isolations of the different trichoderma species as well as the biostimulants, which is very important. And I think this session cover almost all. But the only thing is that we don't have interactive session because of the lack of time, and also we managed 1 hour with respect to this one, because only half an hour late with respect to lunch is concerned. So I thank to the co-chair is not there, but as I mentioned, very good session. I learned many things from the many people, many scientists and many workers, which is very important. I think this is a good session, and congratulate to the organizer as well as to all my participants till they are here. So thank you very much. We'll have a break for the lunch. Thank you very much.
Thank you so much, sir. And post session, we have this session would be chaired by Professor Prasanjit Mishra and co-chaired by Dr. Sangeeta Ganeshan. So we'll start by sharp 2:20. Thank you. So we will try to manage time for the post lunch sessions. Thank you.
Ma'am, they are calling. Just we will wait for 2 more minutes. Okay. I said to Dr. Naresh to call all inside. Dr. Naresh, you informed? Okay. Okay, we can start.
So good afternoon, everyone. I am Dr. Uma Maheshwari, Principal Scientist from ICR IIHR Bangalore. Earlier pre-lunch session ended with a presentation on beneficial nematodes. Now post-lunch we will start with plant parasitic nematodes. And post-lunch I know there is a challenge of being awake. We will try our best to overcome that challenge, and the sessions will be interesting, I hope, and you will all enjoy the sessions post-lunch. And my topic is on biopesticides-based sustainable nematode management in horticultural crops. You all know plant parasitic nematodes are very minute, and they are, even though they are tiny, they are mighty pests, and they are called the hidden enemies of the crops, or the unseen enemies, because of their microscopic size and also the cryptic habitat. They are present in the soil, which they are not seen through the naked eyes usually. And so because of this, farmers often get confused with the nutrient deficiency symptom, and this problem goes unnoticed. And the yield loss estimated through All India Coordinated Research Projects on Nematodes, it is to the tune of 21.3% on the average yield loss caused by plant parasitic nematodes. And you can see the monetary loss is around 50,000 to 25 million rupees because of plant parasitic nematodes alone. That too in mainly 19 major horticultural crops, including the important fruit crops, vegetables and ornamental crops. And these nematodes, you can see, this is in all plant parasitic nematodes, you can see this stylet. This is the main armature that is needed for puncturing the roots. So through that, they directly damage the roots, and also indirectly they invite other plant pathogenic fungi and bacteria present inside the soil. So that is called as the nematode disease complex. So nematodes, both as the primary pathogens and also as the secondary predisposing agents, they cause huge crop loss to the agricultural and horticultural crops. And these are the major symptoms. You know, the root knot nematode is the major pest, nematode pest in horticultural crops. And the symptom is, the typical symptom is the root galls or the swellings, like cancerous growth in the roots. That is because of the formation of giant cells. You can see, so here you can see, this is a cross section of the root, and these are the normal cells, but this is the nematode. So the vermiform nematode now, after sucking all the nutrients and water, it has become swollen in the shape, and this is the female nematode. And around the neck portion, you can see these are the giant cells. 100,000 times they are enlarged in size as well as in numbers. So this is the normal cells and these are the giant cells. So this host cell modification happens because of the root knot nematode damage. So because of this giant cells only, you can see the galls or the swellings in the root. That is because of these giant cells. And now, especially in guava, you are seeing a serious problem because of the nematodes, especially the melodagain, incognita, melodagain, enterlobiae, melodagain, javanica, indica. So many species are associated, but the most virulent one, the damaging one, is the melodagain enterlobiae species. This is causing, it is increasing at an alarming rate all over India. And you can see the above ground symptoms because of these nematodes. It is yellowing, stunting, wilted leaves and drying of the branches. So this is often confused with the nutrient deficiency symptom, because below the ground, the nematodes, they are sucking all the nutrients and water. So because of the deficiency in the nutrients and insufficiency in the water supply to the above ground, you can see in general there will be the stunting in the patches. That too, it starts with the patches, followed by the yellowing of the leaves and then wilting of the branches. So these are all the typical nematode symptoms, but you can confirm only through the below ground symptoms, especially the nodules. If the this root galls are present or the root knots are present, then you can confirm that it is present because of the root knot nematodes. And so mainly it is being spread through the nursery seedlings. So when then through the seedlings from the young stages onwards, it is getting spread to the main fields in the orchards. And so weak seedlings cannot establish well, and so you can see the yield loss year after year it comes. Yield decline is noticed in the farmer's field. And not only that, these nematode infected nursery seedlings, they facilitate the spread of the nematodes to the main fields also, and together in with association with the fusarium, that is fusarium speedy, that is it is causing the guava decline disease. So this disease complex is causing complete crop failure in guava. And this is the damage symptoms in guava nurseries. You can see this is some few commercial nurseries in Karnataka and Andhra Pradesh, where you can see 70 to 100% incidence of nematodes. And so from the nursery stage onwards, this nematode is getting spread. And once it is planted in the main field, you can see these are all the symptoms. They are the photos sent by the farmers themselves. This is the type of damage the farmers are experiencing in guava. And recently we conducted a survey in Odisha also here, some in farmers field in Bhapur, in Brahmagiri, in KVK also, in Denkanal. We could see 100%. Everywhere the farmers fields are all, and also in our research plots, nematodes are there, because there is no awareness about this nematode problem. And here you can see in KVK, Denkanal, Ankul, and the varieties are mainly VNR, Thai Gova. All varieties are highly susceptible to these nematodes. And this in our experimental plot also, now we have started validation trials, and our biopesticides, new products we are trying now against these nematodes. And mainly these nematode, you can see wherever it comes in that planting material, wherever it comes in contact with the infested soil, right, root stalks or grafts or ground layers, you can see the nematode infestation. Okay, infested soil, wherever it comes in contact, so the nematodes can easily enter and cause the damage. You can see this, these are all the stained roots with the egg masses. These are the nematode females you can find inside the damaged roots. And nematode plus fusarium, as I said, nematode disease complex is, this occurs, both nematodes as well as other soil-borne pathogenic fungi and bacteria, they co-inhabit in the agro ecosystems. So here with the nematode you can see the association of the wilt, fusarium wilt, that is fusarium speedy and fusarium species. So you can see only because of nematodes it will be yellow in color, yellow or brown in color, but once that fungus comes inside, you can see the rotting. Okay, it turns black. So that is a disease complex. Both nematode as well as fusarium occurs together in the field conditions. And the yield loss incidence is up to 100%, you can see. And yield loss is around 25 to 30%, and year after year it increases. And there won't be any yield at all after 3 to 4 years. And infection mainly, it is through the planting material, and source is from the infested soil. That is, soil only is the main source of inoculum, and spread is through the agricultural implements, through the tractors or through the small equipment, whichever farm laborers are using. Through that also the soil can spread, from infested soil can spread from one area to another, and through the irrigation water, drainage water, nematodes can be easily spread from one area to another. And this is a scene at Kadiyam nursery. This is one of the major nursery hub. And like this we have in Rajasthan, UP, many nursery hubs are there. But here 100% all, you can see this is Gova, Thai Gova, VNR varieties, and even Arka, our Arka Kiran, Arka Purna, these varieties are all multiplied in many nurseries, and they are getting transported to different parts of our different states in India. And they are not aware of this problem, and they are fully 100% infected materials are getting transported all over India through these nurseries. And not only in guava, other fruit crops, major fruit crops like pomegranate, it is also susceptible to nematodes, and many areas are infected with nematodes. And in banana, the nematode is different. That is the burrowing nematode, Rhadophilus similis we say. And here you can see that nematode, because of that necrotic lesions, that wherever nematode feeds, that cell will die. Cell death is caused because of this nematode. If you longitudinally, if you cut open the roots, you can see all these necrotic spots, and that if you dissect it and keep under the microscope, you can see a lot of nematodes coming out. So they are all the migratory endoparasites. They continuously feed, and wherever the nematode feeds, you can see the necrotic tissues. So that because of this, it occurs not only in the root but also in the rhizomes also. So there will be poor anchorage. So even with the slightest wind, the plants can topple. This is the toppling disease, and together it occurs with the Fusarium oxysporum FSP cubens, that is the Panama wilt pathogen. The disease complex often occurs in the banana ecosystem. And citrus, we have citrus nematode. This also causes a severe root damage. You can see that root system is getting damaged because of, and in vegetables, 100% all the vegetable crops are susceptible to root nematodes. And in below ground, if you see in potato, carrot, beetroot, sweet potato, so the quality is also affected because the market value is down. If there are presence of these galls, if they are present in the produce, then market value also goes down. So quality as well as quantity is affected because of nematodes. So nematode damage, here you can see this is a patchiness, stunting in patchiness. So here and there you can see the in patchiness it starts, and as the time goes, when no control measure is taken for nematodes, you can see the entire field is affected, and it will completely turn yellow, and there won't be any yield. Year after year, you can see the decline, and these are the main symptoms because of root knot nematodes. And polyhouses are completely devastated because of nematodes. Whatever crop we grow inside the polyhouse, like capsicum, cucumber, or melons, or gerbera, carnations, all commercially important crops, they are all susceptible to nematodes. And here you can see these are the condition in the farmer's field, poorly maintained. Not even 30% crop germination is there because of nematodes. And here, and not only that, even so in flower crops also you can see the damage in Crosandra, in tuberose. Here you can see this, even the flower emergence is affected when the crop is severely affected by nematodes. And in chrysanthemum we have foliar nematodes. Now it is discovered in Andhra Pradesh and also in some Bengal areas. It is a foliar nematode. It is not affecting the roots, it is affecting the leaves, and also the flower production is affected when these nematodes occur. And in gerbera and carnation flower crops, commercial flower crops also, there is serious problem inside the polyhouses. So inside the polyhouses, why you have more nematode problem compared to open fields is because of the microclimatic conditions. There is more humidity, more temperature, and more agricultural inputs, and longer duration of the highly susceptible crops. So you will be growing tomato, capsicum, cucumber, all are highly susceptible. So continuously if you grow the crop, one nematode in 1 month, it can complete the life cycle and multiply 500 times. So in one egg mass, around 300 to 500 eggs are there. So in 1 month, one nematode can multiply many folds, and that too inside the polyhouse condition, its fecundity is more, and also its life cycle is shortened. So if outside it takes 28 to 38 days to complete the life cycle, inside the protected conditions within 21 days it can complete. So the nematode multiplication rate is very high inside the polyhouses. That's why you have more severe problems inside the protected conditions. And so how to produce this nematode free planting material, that is very, very important. Clean planting program is also going on now. It is an initiative taken by the government, because nematodes or other viruses or any soil borne pathogens should not be transmitted in the initial stages itself. So there is always a need for production of clean planting material. And for that, as I said, for nematodes, easy entry is through the infected soil. So you can avoid the soil and go for soilless medium, and that too it should be sterilized. It should be sterilized vermiculite or coco peat medium can be used, and that too air layering is recommended. Instead of ground layers, air layering is recommended for guava crop in order to avoid the direct contact with the infested soil and further spread. And you can use the soil from virgin land or solarization, even wherever you can trap the solar heat, sunlight. In hot summer months, you can even use that, and you can sterilize the soil. So for that, the potting mixture you have to spread over that some cement floor, and then you have to cover with the linear low density polythene sheet, LDP sheet. You can trap the sunlight, okay, 100% all the sunlight. This should be carried out only in the hot summer months. This is not feasible in rainy days or winter days, and only in the hot summer months. That too for 1 month you have to do. So it will be completely sterilized. Not only nematodes, even other soil-borne pathogens, weed seeds are also controlled by the solarization. And what is this double solarization is that solarized soil you can fill in the pots. You can, that potting mixture you can fill in the pots, and that again can be solarized again. That again you can cover with a linear low density polythene, transparent polythene sheet, so that it can trap the sunlight, and you can control the nematodes by total sterilization. So that is double solarization. This is very, this is a physical method of control wherein you use the temperature. So the temperature will increase 10 to 15 degrees more when you are trapping the sunlight inside. And this can be carried out in small area. It is not feasible in large areas. Solarization can be done for the nursery areas or under the protected conditions or for the potting mixtures. This is feasible. and use of PGPR in soil. Biopesticides you can enrich, that I will be telling you detailedly in the further slides. Air layering and nematode resistant rootstocks. We have found that Pseudium cattleyanum is having total immunity to these nematodes. And now we have come out with some promising interspecific hybrids also. We are evaluating for the horticultural traits, and soon we will be coming out with the technology. And this is how you have to treat the biopesticides in farmyard manure that is used for the potting mixtures, and the same can be applied in the soil also. So from IAHR we have found Trichoderma, Pseudomonas, Specilomyces, Poconia. So these can be mixed with the farmyard manure or neem cake in the potting mixture. Usually in the potting mixture we will be having 1:1:1 ratio of red soil, sand and farmyard manure. That farmyard manure can be enriched with these biopesticides. So instead of having the plain farmyard manure, you can enrich and add some good microbes, so that you will, from the initial stages onwards, you can see nematode free, disease free seedlings. And in main field, as I said, you can, these are the integrated measures you can follow for avoiding the nematodes. One is follow strict domestic and international quarantine measures. As I said, we have seen the pictures also how it is getting transmitted through the nursery seedlings, through the major nursery hubs. So we have to avoid, there should be strict quarantine, and even interstate also there should be strict quarantine in the borders, not to move the already infested nematode material, planting materials to other new states. So this guava nematode was first reported in 2016 in Tamil Nadu, and now in another 10 years it has spread to more than 17-18 states in India. So that is increasing at an alarming rate. But if we have strict quarantine measures in the state borders, then we can avoid these problems. And farmers also should have the awareness about these problems, so that from the initial stages they can avoid these nematodes. And use of nematode free planting material, it is very, very important. Once we purchase a lot from any nursery hub, just you cut open the roots, cut open the packets, poly bags, and see if any nematode damage is there. If any galls are there, you can reject that. Okay, you have to advise the farmers that whenever they purchase a fresh lot of these planting materials, they should inspect for these nematodes. Okay. And solarization for 15 to, even you can go for 1 month also in the hot summer months of April to June months in India. Then crop rotation, this is also very, very important. It is easily practicable cultural method, wherein because nematodes, these root knot nematodes are more than 2,000, 3,000 hosts are present for this Melodagain species. So you can avoid the multiplication by introducing, just breaking the cropping system by introducing one antagonistic crop or one non-host crop. The best ones are these marigold or velvet bean. Marigold you already know, Tagetes, it has in that root exudates, it has this alpha terphenyl, that is a alkaloid having nematicidal properties. And this velvet bean also we have found, this velvet bean is one medicinal crop actually. It is Mucuna pruriens. It is a medicinal crop. It grown as a green manure crop also. So it has multiple advantages, but we have seen that it is also having nematicidal potential. The root exudates are having nematicidal properties, so that can be grown in between, even in protected conditions also. We have evaluated this velvet bean can be grown inside the protected conditions or in the open fields in between two susceptible crops, so that the initial population will come down. Up to 60-70% initial nematode population is coming down just by growing this velvet bean, and then you can trample it. You can just plow the crop residues inside the field itself. Soil health is also improving, and then it has the nitrogen fixing ability. It is a green manure crop, so nitrogen, NP, phosphorus, even phosphorus availability also is more in the soil. And these are some of the chemical nematicides that is available in the market. Earlier we had only carbofuran or furadon. That was also one insecticide used for the soil pests. But now because of the growing nematode problem, many companies, multinational companies are coming with new products, nematicidal products. And now whatever is popular is this Valum Prime from or Flupiram. This is from the Bayer company. Its recommendation is 500 ml per acre. 10 to 15 days after transplanting in the protected conditions or under the open fields. This is the rate, 500 ml per acre. Then nimids or fluensulfone, this is from Adama company. This is a granular formulation. The recommendation is 1 gram per plant. And already available furadon or carbofuran. For perennial crops, it is around 30 to 40 grams. So for guava or for banana, it is 30 to 40 grams per plant. And nursery rootstocks, In poly bags, 3 grams you can apply per plant. On vegetables, it is around 33 kg per hectare or 12 to 13 kg per acre. This is the recommendation. And these are the chemical nematicides available, and we recommend this only need based. So if you are we will be dealing about the biopesticides, how to deliver that. If properly we are following, that is enough. But if it is a preventive, that is all prophylactic. But as curative, we can go for 1 dose of these chemicals, and we have to have the safe waiting period. It is around 18 days. So after 18 days only the harvest has to be consumed. So we have to wait, otherwise there will be residues you can see in the produce. And IAHR biopesticides, these are our products from IAHR. They are these Purpureosylium lilacinum, old name it is Specilomyces lacinus, and Poconia chlamydosporium, old name it is Verticillium chlamydosporium. These two are exclusive bionematicides. So they will go and target the nematode eggs. So the eggs are laid in the egg mass, so they will target it, and through that they will go to they go and infect the females also. And in the soil juveniles will be moving. So even the juveniles also are parasitized by these nematophagous fungus. So they are the beneficial fungus that parasitizes the nematode, eggs and juveniles. Then we have Trichoderma harzianum, Pseudomonas fluorescens, Trichoderma viridi. So already you know they are all very effective against soil-borne pathogenic fungi and bacteria. But our strains are having additionally nematicidal potential also. So they can control the nematodes also. So our strains Trichoderma Pseudomonas, and we have one more formulation, Arca plant growth enhancer and yield promoter, that is actually a consortia formulation of Trichoderma plus Pseudomonas. It can be used for drip application also at the rate of 10 ml per liter. And so how to deliver is very, very important. So the biopesticide, this talc powder just you take and if you apply in the root zone, you can't see the miracle. You have to go properly, you have to follow all these strategies. So the proper mode of delivery is very, very important to see the success with biopesticides. So you can go from seed treatment, substrate treatment. So seeds, vegetable seeds or flower seeds, you can treat at the rate of 15 to 20 grams per kg. Substrate, wherever nursery is needed, in the pro trace, you can mix the coco peat with this biopesticide, Trichoderma, Pseudomonas, any of it, with 10 grams per kg, you can mix it. So from the initial stages onwards, you can see very good germination and nematode disease-free seedlings. And this already we have, through many popularization programs, we have given training to the nursery people also how to enrich these biopesticides in the coco peat. And the third and most important step, first we saw seed treatment, next substrate treatment, third and most important is the soil application. Soil application, that too, we should not apply the biopesticides as such, we should give one substrate. So that we have this topic of this conference is sustainable agriculture. So there should be, for sustaining these microbe growth in the soil, you have to see one medium, that is best and cheap material is this well decomposed farmyard manure. That is sufficient. Well decomposed farmyard manure or vermicompost or neem cake or pongame cake, you can enrich with these biopesticides. For that the rate is, for 1 ton of this compost, well decomposed farmyard manure, you can apply 2 to 3 kg of Trichoderma, 2 to 3 kg of Pseudomonas and Specilomyces. So nematode occurs together with other soil-borne pathogenic fungi bacteria. So always we have to target, along with the nematodes, other pathogenic fungus and bacteria present in the soil. So one biobactericide, one biofungicide, one bionematicide, it is compatible. So we can mix all these 2 to 2 kg each in 1 ton of farmyard manure, and this has to be applied. See, this is the how to, you have to mix it, and you should be kept under shade. It should not be in the direct sun, because all these microbes have to grow, they have to sustain. So moisture is needed, shade is needed. Okay, so moisture, just you sprinkle water, 25 to 30% moisture. You hold in, after mixing thoroughly, just you hold in your hand, it should be like a ball. It should not be too much sticky, or it should not be dry also. It should be like a ball. That is the optimum moisture. This, how much time, how long it is needed is up to 3 weeks. 15 to 21 days, 2 to 3 weeks you have to leave under the shade for this enrichment. You have to cover it like this, even with leaf fronds or red soil, or you have to keep the shade. Okay. And so this has to be applied in the soil 10-15 days before planting, and you have to give 1 or 2 irrigations, so it will thoroughly decompose and good microbes will be in the soil. So the rate is 2 tons per acre. And for long duration crops, this has to be again repeated. Okay, so you have to continuously recharge this rhizosphere with these biopesticide microbes. So for that, neem cake is the best component. You enrich the neem cake, and the same enriched neem cake can be mixed with water, 1:10 ratio, and that suspension can be given as soil drench. Okay, it can be given as soil drench 2 liters per plant for guava, or 2 liters per meter square for protected conditions or open fields. So this has to be regularly done. Only then you will have a good load of microbes. Along with this compost, the medium also you have to supply so that it will sustain for a longer time. And this we have many farmers are adopting this technology both in open fields as well as in the perennial crops. This is the mode of action of biopesticides, you all very well know. It is a very good plant root colonizer, and they, this Trichoderma pseudomonas, they produce the secondary metabolites also. Here you can see the nematode. Actually the main mode of action of these nematophagous fungus is they secrete the chitinase enzyme that will degrade the chitin layer which is present in the juveniles and egg shell. So that chitin is degraded, this fungus can easily go inside and parasitize easily. You can see how they have, actually J1 stage will be inside the eggs. So they are getting prematurely aborted, you can see, because of the secondary metabolite production under the in vitro conditions. And induced systemic resistance. So ISR mechanisms, other plant defense enzymes are also more produced because of treatment with these biopesticides. So it is increasing the immunity of the plants against the nematode attack. And advantages... okay, sir. And these are the patents we have received for these formulations, and we have registered this with CABRC, and these technologies we have transferred to almost now 700 industries. We have transferred all over the India. You can see we have reached, our IHR strains have reached all over India, north to south and east to west. And success stories also we have recorded in many farmers' fields. They are able to reduce the chemical pesticide load, and also they are able to reduce the cost of cultivation and increase the yield, and they are getting more profits. And you can see many farmers, he is Dr. M.S. Rao sir, he is the primary innovator, principal innovator of these products, and he is now retired, but I am continuing for this transfer of technology to the industries also. And these are some of the success stories recorded under protected conditions. And this is one important farmer. He is a tuberose farmer. He is growing Arca prejual variety, that is high yielding flower variety from IAHR, but that is susceptible to nematodes. He is applying, we intervene with our biopesticide interventions, and this farmer is very clever. He is also selling this enriched farmyard menu to other farmers, so that is also an additional income for him. And he is, per year he is getting in 1 acre 25 lakhs. That means in 1 month he is getting around 2 lakhs. So we have honored him. He is a very progressive farmer. We gave him best farmer award during our foundation day, and IAHR honored him. And these also success stories we have popularized through many TV channels, and this is one success story under protected conditions also, and wherein he has got around, he saved 40 to 50,000 per acre, and he could get additional yield of 7 to 8 tons of cucumber. And we are through many extension activities and DST DBT programs we have.
Okay, ma'am, thank you. Thank you so much, Dr. Uma Megeshwari, for
thank you so much.
Excellent presentation on biopesticide-based sustainable nematode management. And the session is open for discussion. 1 or 2 questions, please. Sir. Yes.
Thank you, sir. Yes, sir. I'm there. Yes, sir. Fine, sir. Thank you, sir.
Thank you so much, ma'am. Now we will request Dr. Sangeeta Ganeshan, ICR, IIHR. So, sir, these two are the plenary session. We are doing it for 15 minutes. Next, every session would be 7 minutes. Ji, sir.
Good afternoon all. I am myself Dr. G. Sangeeta. I am working as principal scientist in IAHR Bangalore, and previously for 9 years I have worked at CHES Bhubaneswar So a major part of the work what I am doing a presentation today has been done at CHES Bhubaneswar, and this is about the dragon fruit, and it's a very catchy crop, fruit crop, and it is an exotic fruit crop which has been introduced a decade before to India, and as it has a lot of health benefits, and there are a lot of diseases as well which are very challenging to manage in dragon fruit, and my presentation is focusing on the some new diseases which we are encountering under Indian condition, as well as some other diseases which are present in across all dragon fruit growing countries, and it is very difficult to actually identify the disease itself. It is a big task because there are many overlapping symptoms of these diseases. So let me focus on that directly. I will go to the diseases. Though there are 14 major dragon fruit species, only 3 are majorly growing across India, and as well as in other countries as well. And here knowledge on the disease is very, very important before going for a proper management strategies. And these are the 4 major diseases which are infecting dragon fruit. One is stem rot, anthracnose, stem and fruit canker, and grey blight. Apart from that, there is an ant damage related to that. Some fruit spotting also we have recorded. And I will go one by one. And this is a stem rot disease which I have reported first time in India. That is actually, though it has been reported from Philippines and Bangladesh, some Asian countries, but the species is Lasiodiplodia theobrome, which is not very severely causing stem rot disease. But in our condition, we have reported Lasiodiplodia iraniensis, which is very, very causing severe symptom on the dragon fruit, especially on the white pulp varieties. And it causes a severe decline in the vigor of the plant, as eventually the plant become very much less productive, and subsequently, and sometimes the farmers are forced to remove the crop as well. So these are all the symptoms you can see. These are the very initial symptoms, and so much we studied for almost 3 years to come to a conclusion. These all are same disease. Means the symptoms are very different. But after the continuous observation and continuous isolation of pathogen and proving the pathogenicity, we could conclude that these are all the symptoms one or other way, it is all are same, and initially it can start in the margin, tip, or in the middle without any visible injury, and yellowing. Suddenly it will stem will become softened and followed by rotting, and complete or partial the stem will collapse. And this is the early stage symptom, and this is the advanced stage symptom where the stem completely rotten. Inside that the fungal fruiting body starts develop, and it will dry. Eventually it will have lot of fungal fructifications like black colored pycnidial structures, which may be circular or irregular. Then again what will happen, those will dry and it will fall off, leaving the characteristic semicircular hole pattern on the stem. So this stage symptom first I observed, then later I went back and checked one by one the symptoms, then all are same fungi we could isolate, and finally all host epidermis will completely dry, shred, and that eventually the tree become the plant become very weak, and it will give very poor yield. So this is the technical aspect related to pathology. We have with a multigene assay and with Koch postulate the pathogenicity assay, we could conclusively prove this is caused by Lasiodiplodia iraniensis, and that Lasiodiplodia theobrome also there, which has been reported from other countries, but those are very mildly virulent. This strain Lasiodiplodia iraniensis, what I said, it causes this rotting within 7 days of inoculation. And you can say here, this is what surprising us, though dragon fruit is a cactus, it is a temperature loving crop. But the temperature here, what we are getting during summer, makes the crop so vulnerable, and you can see that response of the pathogen to the different temperature. We have inoculated this pathogen in the stems, and we have incubated in the BOD condition, 15, 20, 25, 30, 35, 40. When temperature reaches 40, that complete stem collapsed. So this is how we can conclude that temperature has a lot of influence on the symptom development. And of course, the other thing it is surprised us, this has an endophytic association with the plant as well. Because from the site of inoculation we could isolate the pathogen, but apart from that, different place where 15, 20, 30 centimeter, 45 centimeter apart from the site of infection, we could isolate the pathogen. Not alone from that, from the healthy stem as well, we could isolate the pathogen. So it posed a great challenge for the management. As it is a new disease, we have conducted laboratory studies where that fungicide tebuconazole and hexaconazole has given promising result, which we are going to evaluate under field condition shortly. So this is with respect to the stem rot, where you can see the different symptoms all put together made as a stem rot disease. Then coming to the anthracnose disease, this also initially it was so similar and mimic to stem rot disease. And so far 8 different Colitotrichum species has been reported across the country. But from Odisha, I have proved that Colitotrichum tropical is a more severe strain which is causing the symptoms. Apart from Colitotrichum tropical, Siamansi and Colitotrichum truncatum also reported from India. Here also loss of stem portions due to the severe infection leads to severe yield loss. And of course, the rainfall between July to September, which happens in Odisha condition, is highly favorable for this symptom development. You can see the stages of stem damage by Colitotrichum tropical. Initially, the brown color symptoms, circular, irregular symptoms will develop on the stem. Slowly, it will develop, and you can see there will be a prominent Colitotrichum. In the pathology, we used to say black-headed aceruli. It will see on the inside the spot, and once it progresses without any further management, if you allow, it will slowly eat the total stem will get digested by the fungi. Only it will, if you just shake the plant, that stem tissues will be shredded, and only that central core will intact. Though the plant will not die because it has a strong central core, but all other stem tissues will be shredded away. So this is a very severe symptom we got in our experimental station. Though we are taking, initially we have we expected that this dragon fruit is it is not much prone to biotic constraints, but of course slowly when the crop is settled here, that time for initial 3 years we don't observe much disease, but over the period of year where we have all fruit crops in the particular orchard, and the Colitotrichum being infecting many fruit crops like mango, on all guava, every fruit crop, and one or other way the fungal inoculum is prevalent throughout the year in the fruit crop system. So it makes the crop more vulnerable. And so of course we have identified thoroughly this is the Colitotrichum tropical, which of course we have recorded in Odisha, but in other places in India it is Colitotrichum siamansi and truncatum. And this is severe symptoms on the plant where it weakens the stem, and here main thing is reducing the spore load is the main thing. And of course here azoxystrobin and difenoconazole has been suggested to manage the anthracnose, but this is working very well against the stem canker, which I am going to tell about after few slides. Both these fungicides are giving very good result, of course, for dragon fruit anthracnose as well as stem canker, which both are major disease. And in other countries where the dragon fruit is being grown for many more years, they have come up with some management strategies with biocontrol agents as well as with like a bacillus. Of course, we have to go long way. Now only we have started some work on that management aspects. Coming to the third important disease, this is a grey blight, which is caused by another fungi called Diaporthe species. And you can see, of course, that symptoms all are little overlapping, but here the Diaporthe, you can see the symptom. Initial symptom will be very grey, dry and papery, and that it will elongate as a it will go as in an elongated way like a necrotic patches, and slowly in the advanced stages that tissues initially only the upper layer of the stem has got the infection, will receive the infection. Slowly it will go down and it will eat away. Means that enzymes whatever produced by the fungi, it will digest and it will make a big hole on the stem. Though this is not, as of now, it's not alarming like a stem rot or anthracnose, but this disease also slowly gaining the momentum. We could observe in many orchards this disease, though of course it's not a major concern now. Of course, we have identified as Diaporthe fasciolarum. Other countries reported different species. And here also this hexaconazole is giving, azole group of fungicides are giving good results under laboratory condition. We need to evaluate under field conditions. We are going to do it shortly. And the last important and most threatening disease across the dragon fruit growing countries is stem and fruit canker. in many other countries where dragon fruits are growing, no, they are growing for over 5-6 decades. There also many orchards they are removing only because of this stem canker problem. So this is a destructive disease. Of course, it has been documented in India as well, and it is there in every place where dragon fruit is being grown. And you can see a typical symptom actually of this. It will follow a different pattern of symptoms. Starting from initially, if you see, there will be a small depressed chlorotic specks where there will be a red fleck will be there. Then after that, the lesion become little bit elevated. Second, if you see the second number, the symptom will be little bit elevated. Then it will turn to necrotic and have a clear yellow halo around that elevation. Then again it will go to the rotting mode, then it will form a scabby lesion. Finally it will all get detached off from the stem and it will fall off, leaving some holes here and there. So this is, and that first what I said, the stem rot is very much confused symptom, but there will be extensive rotting here. Only around the scabby lesion there will be a rotting. So we can differentiate over over a period of months and weeks. If you work in the field, you can identify which is due to what, because it will cause a small chlorotic specks. That is a very identical symptom of this stem canker. The same pattern comes on the fruit as well, and where the fruit, if you don't take any spray, then of course it will lead to cracking and it will invite other post-harvest pathogens as well and makes the fruits unfit for marketing. Of course, this is it has a long history, this stem canker. So other countries came up with the best management practices. Best thing is to remove the infected stem from the field to keep the inoculum level low under the field condition. That is the first and foremost one, to reduce the inoculum level. Then applying the fungicides with a different mode of action, not going with the same fungicide. Here protective fungicides with mancozeb and copper, and curative fungicides like difenoconazole and azoxystrobin. This combination is giving very good result, which has been worked out in other countries. And though of course it's an exotic crop, we are also following, it is giving good result. Even azoxystrobin also giving very good result. But in other countries like especially in Florida, where extensively this crop is being grown, where they are limiting only 4 applications per year, because it is an annual crop. We cannot go on, keep on spraying, and one side we are promoting as a healthy crop, another side we cannot dump the fungicides or any agrochemicals on the crop. So they are restricting only with 4 spray per year. And the final one, I said, this I suspected initially as a fungal infection, but I could observe some ant movement as well. There is some correlation between ant and fungi that I have studied at Bhubaneswar only. Then later, when I joined IHR, I joined with one more scientist, Dr. Jayanti Mala. So we could identify if ant is coming first and it will make a boreholes. That is also only one specific ant species called Trichomyrmex destructor. This is a ant species which is causing the boreholes on the fruits. Following that, in the second row of, if you see the fruit, there will be a fungal infection, Carvularia lanata, that comes second. So both jointly it will deteriorate the fruit quality. 10 to 30% of the fruits in a field is getting this kind of damage as well. So not we can't say all ants are causing this damage. Only specifically this ant. Rest are beneficial ants as well are there in the ecosystem. So here what is the management, best management? Encouraging the beneficial ant population. It will help to maintain the ecological balance, and it will reduce the pesticide dependency. As well as we have to keep the orchard or the dragon fruit field free of weeds and other we have to manage the habitat clean so that it will not invite the other pests as well, because when hoppers or when sap sucking insects come, it will excrete the sugary excretion, so that to eat that ant will come again. So we have to maintain the habitat cleanly, and using any kind of ant repellent like banding on the stem like that, because we cannot kill the ant. One way we are promoting the ant are beneficial, another way we cannot kill all ant. Farmers will not know which ant is beneficial, which ant is not beneficial. So we cannot indiscriminately use any pesticides to kill the ants. So we have to repel the ants from the orchard ecosystem, mainly from the dragon fruit. So the further works are progress in this regard by Dr. Jayanti Mala. So this is the overall my understanding about this crop, what I have worked for 5 years. And major disease is stem rot, which is being an endophyte, it giving a big challenge for the management. And rest all anthracnose or stem canker, where well-established management protocols are in place. So if a farmer is able to identify the symptom, then we can he can apply the management strategy proactively to avoid the number of large number of spray on the dragon fruit. Thank you.
Disease also coming, particularly for dragon fruit growing areas. And which are the major area where we have that type of diseases are there, and how much the percentage of infections and actually the loss of the crop if it is considered?
Yes, sir. Of course, actually, every orchard is not having all problems, sir. Even a few weeks before from Sambalpur, one horticulture officer sent the photo where that orchard is almost 70% loss with a stem rot. Some orchards are having the anthracnose problem where they have to remove the crop. It is like that depends. It depends on the management starting from the early stage of the crops, sir.
So
actually.
just start with some of the diseases because these are the new crop and new area also be covering. So which are the major diseases? You have some map of that one, na? Because these are very limited area we are growing in the dragon fruit.
Yes,
so make it that so that the growing area and what are the conditions of the growing area. Because in other crops it will be very, very difficult to manage where it is there, which area is there. But these are the some of these areas already new areas are there. You make it at some point that growing and then infestations, particularly etiology and all things. No, you study that particular crops is concerned.
Yes, sir.
Thank
yes,
you.
Sir, we are. Under COE Kamlam project also we are doing, sir. Thank you.
But this is a crop belonging to cactus family, which is most drought tolerant.
Yes, yes.
Was it only temperature or along with some humidity?
Along with the humidity, sir. Along with the humidity. Yeah, of course.
Then you
Definitely
should have-
along with the humidity.
Then you should have mentioned.
Yeah, yeah, sir. I
high,
missed it.
because high temperature is not going to affect.
No, definitely
Only
along
it
with
is
the
when high humidity coupled. Triangle is there, na? You are a pathologist.
But sir, at IHR, we have seen without the humidity, here at Bhubaneswar condition, I have seen along with the humidity, the severity was very high. But we are in the progress. At this stage, I cannot able to, I will not be able to conclude only this factor or that factor, where that is not a coastal belt, where that temperature of 40 degree makes the crop more vulnerable. So, without that, that it's a point of study, sir, actually. Yeah. Yes.
All right, ma'am. Thank you so much. So now we'll begin with the technical sessions of post-lunch sessions, and I'll request all the speakers to finish this in 7 minutes. We would now like to invite Dr. Banashri Saikia from CSIR NIST Zorhat to please present her lecture.
Good afternoon, everyone present here. I'm Dr. Banashri Saikia, a postdoctoral researcher and a woman scientist at CSIR Northeast Institute of Science and Technology, Jorhat, Assam. So today I would like to offer my gratitude for AgriVision to invite me to present my talk. Today I'll be presenting my 4 years of PhD, and 6 months back I have received a postdoctoral grant which I'll be giving a concept. Here I have my research area focuses on CRISPR-Cas-based genome editing for genetic improvement of Solanum lycopersicum, that is tomato. So before going into genomics, I have gone through a couple of molecular biology, cell biology, and genomic approaches, for which I will be giving in today's presentation a comprehensive overview of my past 7 to 8 years of work. So here I would like to add a point that being in Northeast, away from mainland, it was always difficult to start a new genre of research from the scratch. So with the constant dedication of my PhD supervisor, myself, now he is my postdoc mentor as well, and with the few dedicated research scholars, we were able to establish the CRISPR-Cas-based genome editing in my host lab, CSIR NEST, back in 2018. So now coming to the introduction, as we all know, global climate has been changing day by day. Now the combined occurrence of abiotic and biotic stress leads to severe yield loss in crops. So now it is a high time to develop multi-stress tolerance in crops. So now multi-stress tolerance itself is a complex phenomena. So to study at the genetic level, I have chosen tomato, Solanum glycopersicum, as my target crop, where there is a multiple stress susceptible cultivar Acavicas, which I have chosen for my study. Again, in addition, it is always reported that tomato is facing yield loss by various stress factors, which accounts up to around 70% of yield loss. So now it is reported in the literature that knocking down or silencing negative multi-stress regulatory gene can contribute in developing multi-stress tolerance in plants. So H5PRP and DR1 are two putative negative regulators identified from tomato from literature review. To execute my hypothesis, I have chosen CRISPR-Cas-based genome editing for which it has served as a best platform as it is a non-GM approach. So now to give a brief overview of the function of these two genes, these two are the loosely characterized genes, and this is the schematic diagram showing their function. So I will not go to the detail, but the key point is they both are known to be directly or indirectly involved in the bind with the significant stress regulatory genes in the multi-stress regulatory pathway in plants, and thereby blocking their expression and negatively regulate the process. So that is why there is a lot to be known about their expression, cell biology, and function in multiple stress responses in plants. Now, before going into genomics, it was important to study them at the cell biology level. So I did the in vivo study where I have performed the subcellular localization and interaction assays. So here I'm not going to the details, but we have used the YFP fusion and split YFP constructs prepared in my lab by myself. Then the significant finding was both PRP1 and DR1 showed to be strongly interacting with each other in the cell cytoplasm and towards the plasma membrane, which give an indication that they might function together in imparting multi-stress responses. Now to move towards proving the concept genetically, we have moved towards the genome editing and to functionally characterize them. So here is the brief methodology. I will not go to the detail, but briefly to highlight that we have designed the guide RNAs at the functional domains of the two genes, H5PRP and DR1. Then we have prepared the CRISPR constructs using Gibson assembly cloning method. Then molecular conformation were done. Then after that, they were transformed into agrobacterium competent cells. Then those agrobacterium cells were then transiently transformed into 4-week-old tomato seedlings. Then we have performed the Sanger sequencing where we were able to get the deletion and insertion mutation, and under the abiotic stress, as you can see, I'm not going to the details of the study, but to highlight that phenotypic, physiological and biochemical analysis showed that H5PRP and DR1 unedited transient plants were tolerant to both drought and salt stress as compared to wild type. So under the biotic stress, under the biotic stress also, I was able to get the indel and substitution mutation in both PRP1 and DR1 CRISPR-edited plants. So the phenotypic analysis visibly showed the bacterial leaf spot and bacterial wilt tolerance in the CRISPR-edited line as compared to wild type. And the histochemical analysis where ROS accumulation and cell death study were done, which indicated that they were tolerant to bacterial leaf and bacterial speck spot as compared to wild type. So that was in a transient system. Now I was also successful in doing the stable genetic transformation where I have standardized the tissue culture regeneration method of the Archivicas cultivar in my lab. Then we have on the genome edited first generation, that is the G0 lines. We did the molecular confirmation, and the Sanger sequencing revealed that here also we were able to get the indel substitution mutation. So we have taken care of the off-target effects as well for both transient and stable line. So that was whole about my PhD work. Then the progenies of the G0 lines were then taken up for the extensive molecular evaluation to select the homozygous line of HYPRP1 and DR1. Then there also phenotypic analysis were done, which showed the tolerance to abiotic and biotic stresses, that is drought and bacterial wilt stress tolerance. In addition to other parameters, the SEM micrograph shows that there is a disruption in the xylem vessel cells in the wild type line as compared to the CRISPR-edited lines, where there were the walls of the xylem vessels were very intact. So here, in addition, I would like to add a significant point, and we are happy to inform you that we are one of the few labs who are able to get a Cas9-free and transient-free genome-edited target line, which actually showed the tolerance response to drought and bacterial wilt. And the further confirmation are still ongoing, but this is a significant achievement from our lab, which I wanted to show you here. So now coming to the summary, that two genes, HYPRP1 and DR1 has been chosen, and their expression were studied where they showed that they are highly expressed in the stress-susceptible cultivar Archivicas. So then the cell biological study has revealed that HYPRP and DR1 both were strongly interacting with each other in cell cytoplasm. Then we did the CRISPR-Cas9 genome editing on them, and the transient genome editing revealed that we were able to get the indel and substitution mutation, and both in dual and single gene plants. So then transient stress assays showed significant multiple stress tolerance, and in the stable genome editing, when revealed that CRISPR-edited editing events of single and dual gene edited HYPRP and DR1 have been replicated with efficiency. Then CRISPR-edited HYPRP and DR1 showed significant tolerance to biotic and abiotic stress, and the further in the homozygous lines, and they are being evaluated for heritability and stability study. So with this work leads and the confidence of my lab setup and my PhD work, and also 2 years of my senior project associateship, I have formulated one project where I was successful in getting a UN scientist project from the ANRF government of India, which I have recently started 6 months back. So here also I am giving the concept, not showing the details of my data so far, but here also I have taken the Archivicas, that is the highly susceptible cultivar, again for late blight and early blight. And there is very less genetic information available regarding the genetics, regarding the disease susceptibility gene for early blight in tomato, and there's a huge scope for genomic studies. But I was able to identify a gene, MI482, which is reported to be a key potential disease susceptible and negative regulator to late blight. So again, here I have taken CRISPR-Cas genome editing.
I think you can conclude your presentation over. This is your project only, so we can discuss later.
Okay, ma'am. So here again, CRISPR-Cas has been taken. So the aim is to do the transcriptome-wide identification of early blight and late blight to identify the genes and study for the tolerance. Then here is my publication summary.
Okay, thank you. Very good presentation.
Thank you.
So any questions? The session is open. One question we can invite.
Yeah, Archivicas has been in India. It is known as a multiple stress tolerant cultivar. So
No,
there
not
are reports.
multiple stress tolerant.
Sorry,
it is susceptible cultivar.
Yes. So that's why I have chosen. So to work on a susceptible cultivar to prove my hypothesis was very important. So Archivicas is well known. So that's why I have chosen.
But additionally, you can take one more leading variety which farmers are growing at present, which is having susceptibility. It will have direct application in the field.
That will be the Pusa ruby probably.
Or any private popular variety. You can take additionally along with the Archivicas.
Okay, for my next project, I'll be then adding up that point. So far,
thank you.
Thank you.
Thank you so much, Dr. Banashri. Now, can we please invite Mr. Dibakar Malik from HCL Foundation.
Mr. Dibakar, you have to restrict for 5 minutes because we have 9 more presentation before 4 o'clock. 4:15 we have to conclude.
Hello. Hello. Jai Jagannath. Respected chair, dignitaries, students, professors, and farmers. I am Divakar Malik, along with my colleague Subham Sukla. I'm from HCL Foundation, the CSR arm of HCL Tech. Along with Forest Uttar Pradesh Forest Department, we are working in Sandy Bird Sanctuary, which is located in Hardoi district of Uttar Pradesh.
You can directly go to the study.
So I will be talking about climate-driven agroecological vulnerability. It is much more like social study, like not that scientific. So before I begin, I would like to ask, what do you think is the most affected by waterlogging? It is food production, or is it farmers' income, or is it pesticide usage, or is it biodiversity health? Actually, it is all of them. So I will be talking about कि ये सारे जो चीज़ें हैं वो कैसे अपने आप में इंटरलिंक्ड है, मतलब एक जगह की स्टडी करते वक्त. तो हमारा जो स्टडी एरिया है वो है एक सांडी बर्ड सेंचुरी जो लोकेटेड है हरदोई डिस्ट्रिक्ट में, जो एक रामसर साइट है, which is exactly 308.5 hectares area, and it is ये इंडो गैंगेटिक प्लेन में आता है. एंड ये सब अपना जो it receives most of its water from monsoon, and it is the home to 1% of southern Africans as a population of common teal or any Ferguson duck, and more than other bird species in that migrated from the Siberian areas. So the main problem arises from how we are going to link different things like food production, income, livelihood, and agrochemicals, and that things. So before we go for this, I would like to understand what our study area. So east to our study area, there is a river flowing, which is known as Gara River, and that is the main reason for flooding in area. And we can see that the study area has some flood-prone areas. That is the main that is mostly affecting the agriculture economy of that area. So, like that thing, the flood creates a hydrological stress among communities in the study area, along with cross-regional crop failure and staple food deficits. Like if, for example, if the yield loss is 0 to 20%, the household food availability is 5 to 6 months. Or similarly, for 40 to 60%, the household food availability will be 3 to 4 months of food. And similarly like that, and that affects the income and liquidity stock of the community or the farmer. And similarly, that creates the food insecurity and livelihood partiality amongst the farmer, and that collapse the food and income buffer and alters the behavior. And farmers tend to prioritize immediacy of return over risk mitigation, thereby increasing the exposure to high risk input and production of systems. For that, they are distressed and shift toward monocropping, or that is the vegetable monocropping, because as I explained earlier, the area is flood prone and they cannot grow traditional crops like rice. That's why they are tends toward monocropping vegetables, and that increase rapid transition from diversified cropping system to monocropping. to short cycle crops like 30 to 60 days to get high input with high input dependency like fertilizer, pesticide, and irrigation systems. Intent that increases the fertilizer area, and that um, to meet the market oriented productions. In another way, we can see like to gain short term gain the high production they do high production risk, and with greater dependency on agrochemicals, and that affects the environmental and health externalities, and that creates the monocropping offers a speed also often that monocropping offers a speed case. It simultaneously increases the vulnerability, setting the stage for intensive agriculture use.
Deepakar,
yes.
You can conclude.
Okay. So, like this, there is an explosion of high agriculture use, like 22 spray around agriculture, 30 to 60 day agriculture cycle, or 3 to 5 spray per week. And that intensified the ignorance, but due to constant risk, it's
जय जगन्नाथ। जैसे कि समय की सीमा को कम रखा गया है और अभी तो हमारे पास बहुत ज़्यादा मटेरियल था, इसलिए थोड़ा सा मैं अब ये बताना चाहूँगा कि जैसे हम लोगों ने जो काम किया है सांडी बर्ड सेंचुरी के अराउंड, वैसे तो HCL फाउंडेशन जो है 22 स्टेट में काम कर रहा है, और हम लोग जो HCL समुदाय करके हमारा परियोजना है वो उत्तर प्रदेश के 10 जिलों में और साथ में तमिलनाडु के थूथुकुड़ी में हम लोग काम कर रहे हैं। इसके साथ साथ यहाँ पे जब सर्वे किया गया फ्लड की वजह से वहाँ के जो किसान हैं वो लोग पेस्टिसाइड्स पे ज़्यादा डिपेंड हुए। उसका कारण ये था कि उनको अपनी आजीविका पालन के लिए वेजिटेबल्स पे स्विच करना पड़ा, जिसमें ज़्यादा वेजिटेबल्स ज़्यादा पेस्टिसाइड का यूज हुआ। और हम लोगों का वहाँ पे इंटरफेयर करने का सबसे ये रहा कि बर्ड सेंचुरी में जो पक्षी आते थे बाहर से, वहाँ पे उसके पेस्टिसाइड की वजह से पक्षियों को नुकसान हो रहा था। इन सभी समस्याओं को एड्रेस करने के लिए हम लोगों ने 6 इंटरवेंशंस डिज़ाइन किए, जिसके अंतर्गत मल्टीपल एक्टिविटीज़ के साथ में फार्मर्स के साथ काम कर रहे हैं, जिससे कि वहाँ पे यूज़ होने वाले पेस्टिसाइड्स कम हो। जो हमारा मुख्य उद्देश्य है कि हम सस्टेनेबिलिटी पे काम करें, तो सभी कंपोनेंट को मिला के फार्मर्स के खेत में सस्टेनेबिलिटी भी बनी रहे, हमारा इन्वायरमेंट भी सेफ रहे। इन्हीं सब चीज़ों को देखते हुए हमारा जो साथ साथ हम वाटर पे भी काम कर रहे हैं, सभी चीज़ों को मिलाते हुए इस तरीके से हम लोग वहाँ पे काम कर रहे हैं। धन्यवाद, नमस्कार।
Thank you, thank you, thank you.
Thank you so much. So we'll move towards the next one, Dr. Bhagya Vijayan from ICAR and RC on Lychee, Muzaffarpur. Also, just one information before she comes up on stage. There's a wall clock which is sponsored by Dhanuka Agritech, which is for everybody. When you are leaving this place, please collect it. It's for all. Thank you. Dr. Bhagya Vijayan?
Yes, ma'am. Ma'am, I would be presenting online.
Bhagya Vijayan is there?
Oh, she'll be giving it online, Acha. Please connect her.
Dr. Bhagya Vijayan?
Yes, ma'am. Ma'am, I would be presenting online.
Online. Yeah, you please share your screen. Dr. Bhagya, your time is only 5 minutes.
Yes, ma'am.
So try to complete within the schedule.
Ma'am, can you please keep it on slide?
Yes, yes. Now is it visible?
Yeah, you slideshow you can go.
Okay, yes, ma'am. I'll put it in slide. Namaste, good evening to all. I am Dr. Bhagya Vijayan from National Research Center on Lychee.
Dr. Bhagya, 1 minute. You please go to slideshow mode.
Actually, ma'am, I have slideshow.
Okay, you can continue.
Is it moving, ma'am?
Yeah. No, not moving.
Okay.
Ma'am, you can continue just like that. No problem. We can still see it.
Fine. Today I will be presenting on life cycle-based sustainability assessment of waste integrated farming system towards carbon neutral agriculture. Basically, as you know, Bihar is famous for Bihar, especially Muzaffarpur district is famous for lychee, and Muzaffarpur has got its GI tag on lychee. When we started the research and when we talked to the farmers, majority of the lychee orchards are given on lease agreement. Hence, improper pest and nutrient management uh, is on the rise. And it is leading to over-adoption of pesticide and fertilizer uses. Thus, it became inevitable for us to identify the hotspots of improper management through life cycle assessment. And with this objective, the locale of the research were Muzaffarpur, Vaishali, Samastipur, and Bihar districts, which were purposely sampled. Sampling size was 251. We then clustered the groups. We got six clusters, and basically the cropping pattern was lychee, other fruits including mango, amla, guava. We also cultivated cereals, pulses, oilseeds, and other crops such as potato, etc. The cluster was validated, and we got six clusters. And based on the energy use efficiency and carbon footprint and the subsequently BC ratio, we found that different clusters were having different characteristics, and cluster 2 emerged as the most efficient cluster. As you can see, the carbon emission was 4595 kilogram of carbon dioxide equivalent per hectare per year, and cluster 5, wherein you can see it was cluster 6 was it was cluster 4 was the highest. Even though we got carbon emission like this, we then move towards the sustainability part of it. Spearman analysis revealed that landholding size exerted a stronger influence on carbon footprint, and a composite sustainability index was done, and we could find that cluster 5 emerged as the most sustainable one. It came as little contrary to the above result, wherein cluster 2 was declared as the the climate optimal archetype. The point of the study and the finding of the study is that one fit size carbon mitigation strategy is not suitable for the different farming system. Different farmers follow different types, as you may be knowing. In The lychee orchards are given on lease, and half of the part of the lychee planting till fruit bearing, the farmer uh, uh, sees it, and otherwise the trader is the one who does the pesticide management, insecticide management. Everything is done by the trader. Hence, the imbalance in lychee orchard system is existing in Bihar system. Uh, Based on the sustainability calculation, we found that cluster 4 has excellent carbon credit suitability, and mitigations such as solar irrigation, drip or micro sprinkler irrigation, etc., were the mitigation strategies based on the practices followed by the cluster 4 farmers. Similarly, for other clusters also, we have found out the practices followed by the farmers, and similar mitigation strategies are being disseminated to them. Thank you.
Thought you are explaining with this slide, but I think your slides not moved.
Oh,
Okay.
okay, madam.
Anyhow, we could listen to you, and it's a good presentation. And yeah, you can unshare the slide, Dr. Bhagya.
Yeah,
Thank
yeah,
you.
yes, yes.
Thank you for your presentation.
Thank you.
Thank you so much. So now we'll request Dr. Shashwat Kumar Kar from ICAR IISWC, RC Sunabida, to please give his lecture. Please change the screen. Slide, please.
Dr. Shashwat Kumar, yeah, you please restrict with 5 minutes.
Okay, okay, madam. Respected chair, co-chair, and rapporteur, and all the participants. I am Dr. Shashwat Kumar Kaur from ICR, Indian Institute of Soil and Water Conservation, Koraput. So my studies on the soil hydraulic conductivity change on different land use practices for the Eastern Ghat region. Actually, due to land degradation, it has created a major threat to food security. In this regard, we have also committed to restore the degraded land by 2030. In this study, we have found out some suitable, sustainable land use practices for the Eastern Ghat region, which can improve the soil hydraulic properties. So we have two objectives, like to study the soil hydraulic properties under different agriculture and horticulture land use practices, and also to study the pore size distribution, how pore size is changing under different land uses. So this is our study area in Eastern Ghat region. So we have 4 land uses like mango orchard, cashew orchard, agriculture and pasture land. So this is the Hood infiltrometer. To do the study, we have basically studied the hydraulic conductivity of soil. To do this, we have used that Hood infiltrometer. This is a non-destructive type of instrument. This is the advanced type of instrument which is used to study the soil hydraulic conductivity. This is UGT made by Germany. So along with hydraulic conductivity, we have studied the soil physicochemical properties for the 4 land uses. As you can see, in case of horticulture land uses like cashew and mango, we have observed higher ball density, higher organic carbon, and also the higher mean wetted diameter as compared to other land uses. This higher value in case of horticulture land use may be due to continuous leaf litter addition and due to less use of heavy agriculture machinery, which led to the improvement in soil porous structure. This mean wetted diameter, it depends the soil aggregate stability. Higher mean wetted diameter, it leads to more stability, so it will lead to less erosion of that soil. So basically we have studied the steady state water flow. As you can see, we have done this study at different pressure heads starting from 0 to -3. Means we have gone in different suction head. We have studied how water is flowing inside the soil profile. So as you can see, in case of 0 means at complete saturation stage. That in case of cashew, we observed higher flow followed by mango, then followed by pasture and agriculture land. Similar pattern was followed rest of the 2 pressure head, that -1 and -3. This is the hydraulic conductivity. We have also observed that similar as followed in water flow, hydraulic conductivity was higher in case of cashew, then mango, then agriculture and pasture. So with this, we have studied the pore radius, means how much pore in that soil, that is percentage of soil volume, is carrying this water. As you can see, under different pore radius, as you can see, 2.5 to 0.5 mm pore radius, that is the macro pores. We can observe it is micro pore. We can see in case of horticulture land use, it consists highest percentage of macro pores as compared to other agriculture and pasture land uses. Similarly, this macro pore, again we have studied how much percentage is contributing towards the total flow of water inside the soil profile. As you can see, in majority of cases, around 70% of total flow is contributed towards this macro flow. Although it constitutes only very negligible percent of total soil volume, but it carries around 70% of total flow is contributed towards the soil profile. So that means if by any change in land use, if we are improving the soil macro pores, that means we are increasing the porosity, then we can control the runoff pattern. That means we can further control the soil erosion and also improve the soil moisture. Further, we have also studied the Pearson correlation study to see that which factors are impacting the soil hydraulic conductivity. As you can see that that mean weighted diameter and organic carbon, which is highly significant and it is positively correlated with hydraulic conductivity, whereas that ball density, it is negatively correlated towards the hydraulic conductivity. So we can conclude that in case of Eastern Ghat region, that by adopting cashew, which is dominantly adopted, dominantly followed, if you can replicate this cashew and mango other land uses, other areas, so we can improve the soil hydraulic conductivity, thereby we can improving the more water retention in the soil profile. That's all. Thank you.
Okay, good presentation. Yeah, yeah. Okay, it's okay. Yes, yes. No problem. Kindly go ahead. Please restrict in 5 minutes.
Yeah, yeah. Good afternoon. Myself, Dr. Raghu BR. I'm a senior scientist at IHR Bangalore, working in division of vegetable crop. My topic is genetics and nutritional diversity in curry leaf. You know that I don't need to introduce that how the hidden hunger is affecting whole world, including India and also Asia. Mainly the zinc and iron and calcium are the, and iodine and vitamin A are the major nutrients and causing enormous damage to human health. So with this, there are several strategies are there. Among them like biofortification and industrial fortification, food fortification. The biofortification is the one, it's very important, where not only you can enrich the food nutrient content and also the bioavailability. In other cases, there is no guarantee for bioavailability. Here, because of their natural health promoters, you can enhance the bioavailability. With this, see, for bioavailability, we need a nutrient dense leafy vegetable like drumstick, curry leaf. They are highly rich in nutrient and they are culturally accepted. So these are all the criteria where you can easily use them. So we know that throughout India we use curry leaf for many other purpose. So this is a good candidate for biofortification. It is rich in many minerals like calcium, iron and zinc. So with this we आपने बताया था कॉपी करवाना उस टाइम ओके he's unable to copy. Now he is copying. Okay, I will start from here itself. So as I said, the curry leaf can be used directly as a fresh leaf we are using. In addition, it is one of the best candidate for other spices product. So it is culturally accepted. So it is a very good candidate for food to food fortification where you can enrich our staple diet for nutrient and other health promoting factors. So with this, what are all the research gap? There is a limited information on genetic diversity for mineral nutrient, and we know that it is a rich, but we don't know what is the extent and how it is distributed. And few studies are only on the small scale of this one, germplasm. So with this, we have started nutritional diversity analysis and also identifying the LH genotypes. So this is the, in this study, we have, what happened? In this study, we have, what happening? No, not photos. Okay. So around 80 genotypes we have used, mainly from Orissa collection we have, Himachal Pradesh collections we have, Tamil Nadu, Karnataka. In Karnataka we have used both Western Ghats and also from the mainland. And this is the around 80 genotypes have used, and we have collected and we have established in ex situ condition where you can provide a uniform environmental condition. So this is the methodology we have followed. We have estimated for 5 macronutrient and 5 micronutrient, and statistical analysis we have followed to identify distribution, association, as well as to identify the LH genotypes. Coming to the results, you can see that ANAVA, it is clearly telling that all 10 mineral nutrients are highly significantly different. It shows that there is a huge genotypic differences, and heritability, everything, it is very, very high. So it indicates that through selection it is possible to improve the nutrient content. So coming to the region wise distribution, before that in among micronutrient, the calcium is very high. You can see that the range from 2 to 5% it is there, followed by potash and other. In the micronutrient, we have a very good, an important micronutrient element, that is iron. It is very good, up to 400 ppm we have estimated. Coming to the region wise distribution, we can see that from the Karnataka, Tamil Nadu, Orissa and Himachal Pradesh, there are different content for different regions. Like in case of Karnataka, the calcium is very, very high, and also the same amount of iron also we have seen, followed by in Himachal Pradesh, iron is very high. So this is a regional distribution we have. Coming to the correlation, we have identified very good correlation for some of the important nutrient, like for calcium and iron. We have seen copper is very, very close to very significantly and positively associated, and also the iron is positively, the calcium is positively associated with magnesium and other. So coming to the association studies, we have, you can see that easily, the nutrient like calcium, iron and calcium and iron, important nutrient, are clustering in one, and they are positively influencing the overall. It indicates that the positive content of the nutrient, these important nutrients, are highly influencing the diversity among the germplasm collections. And you can see that the scattering, though we have from Tamil Nadu collections and some of them are from Himachal collections, are clustered together, but from Karnataka and Orissa they are scattered. It indicates that huge variability between the regions also. So this is the heat map we have generated, both nutrient content as well as the genotypes. We can see that the outer cluster, the 2 genotype, they are extremely high for boron as well as phosphorus. Besides, the middle one, that is the cluster 2, the genotypes, large number of genotypes, they are good for 1 or 2 nutrient, but they are very, very low for other nutrient. But this is the cluster 1 containing only the few genotypes. They are multi-nutrient rich genotypes. They are very good candidate for by food to food fortification, like iron, calcium, zinc. They are highly rich in this nutrient. So coming to the our significant, so this is the region wise. As I already told, region wise we have a variability. This is very important for further exploration and conservation, and this is the heat map, this is the dendrogram we have generated. So this is the table tells, we have identified some of the genotypes, we have received IC number also to conserve them, and for each genotype, mainly I focus on calcium. In case of macronutrient, we have identified 3. You can see that the nutrient content, this is 5% around, but the reported one maximum is only 2.2%. So this is the significant improvement in the our germplasm collections. Similarly for iron, you can see that the maximum reported is 181 ppm, but we have reported 2 genotypes, they are more than 400 ppm. So for others also we have identified some of the allied genotypes. So in conclusion, I can say that for significant variability we have mapped in among diverse germplasm collections. We have identified some of the elite nutrient, multi-nutrient genotype, like 2 genotypes like IC652419 and IC652461. So thank you very much.
Thank you, Dr. Raghu, for excellent presentation. And
sorry for some
it's okay,
technical issue.
But the presentation was good. The topic is open for a discussion. We can invite 1 or 2 questions.
Yeah, actually in all, we have collected from Kangra, Sonal, so what is that, Kangra, Palampur, Shimla, three area we have collected, all are wild type only. From the forest area we have collected. Around 35 collections we have from Himachal Pradesh. And they are very good in iron.
Doctor, I have one question.
Yeah.
Actually, nowadays in urban population, they used to preserve these curry leaf in the fridge for months. So could you find any difference among the cultivars what you have selected for study for the storage?
Yeah, we have studied, madam. Observational study we have done it. The genotypes vary for powder also, because powder is one of the thing, and also fresh leaf. Some of the can retain very good freshness with good attractive chlorophyll content with nutrient. Some genotypic variations are there for this. Some can extend up to 1 week.
Okay.
Some can easily deteriorate within, they became black and other moldy within 3 days.
You are talking about that room condition or in
room condition, ambient condition.
I am telling about the refrigerated storage.
Refrigerated we have not studied. Only in the ambient condition we have kept it, because normally vendors, everyone will keep in the refrigerated condition, and they want up to window period up to 1 week from cutting to the suburban area condition where we have noticed this kind of things.
Okay, thank you.
Thank you.
So I am Professor B.K. Sahu,
Yes, sir.
a retired professor of agronomy from OUT. So thing is that in curry leaf, propagation is little difficult because whenever the curry leaf plant is there, even from the roots the plant generate.
Yes.
But if you take it and transplant it, the survival is very, very minimum. And sometimes from the seeds also it is being propagated. So my query is that what is the best method of propagation?
Sir, very good question, sir. Actually, commercially only one variety release is Suhasini from US Dharwad, sir.
Achha.
And they are commercially propagated through rooted sucker, as you mentioned.
Okay, okay.
They have a half-acre area. I have visited mother block they have maintained. From that, but the propagation ratio is very, very low. As you said, only 5 to 6 you will get. As you said, the seed is the very important thing. Now, wherever the commercial high density planting is happening, wherever in case of Anandpur area, in Andhra Pradesh and other some Telangana area, and also in Coimbatore area, they are following the seed. But seed is very, very low viability. Only 1 week it can be retained. It will lose the viability if you reduce the water content less than 30%. So, the seed is one of the important the propagation method provided if you could able to complete the sowing within 1 week, sir. One thing. But the only one issue in the seed is that this is both seed propagated as well as vegetatively propagated. Most of the vegetatively propagated, they always maintain higher heterozygosity, sir. The moment we go for seed production, huge variability within the population we will get. For a farmer is okay, but for as a breeder, if you want to make it as a variety, at least we have to bring, stabilize the population where it is suitable for seed propagation and also maintain the uniformity. So this is one of the area we can focus on.
And another thing, what Madam was asking regarding the storability. So, because I got one curly plant in my own home, so after we pluck the leaves, if you keep it as such inside the plastic cover, it can be kept for some time. But if you wash it, then immediately within a few days it will deteriorate.
Yes, yes, it is there. So we are also working with our post harvester. That is there.
Thank you.
Thanks.
Thank you, Dr. Raghu.
One question from my side. Regarding quality of leaf, whether it varies under different cropping system or farming system?
Yeah,
under
sure, sir.
agroforestry system or
Yes,
in
sir, yes.
intercropping,
Yeah.
whether quality varies?
Yes, sir, definitely, sir. Because the quality is the one trait, it is highly influenced by environment, sir, including its essential oil content. We have reported one paper also we have published, how the even cutting time also. If you delay 1 week also, the quality will be vary. So cropping system, our cultivation method, even prevailing environment, everything will be influenced.
So whether the legume intercropping with legume,
Yeah.
the quality is good or any other crop?
That we have not, intercrop we have not studied. Yeah.
Fine, thank you.
Yeah.
Thank you so much, sir.
Thank you, Doctor.
The next two presentation, the lectures are online. So are they there? Can we connect to Dr. Manjunatha Gowda?
Dr. Manju?
Yeah, I'm online.
Okay. Please share your screen.
Yeah, I'm sharing, madam.
Okay. Dr. Manjunatha, your time is 5 minutes. Kindly restrict with 5 minutes. Kindly go to slideshow mode.
It's a slideshow mode, madam.
Yes. Please proceed. Yeah, you can start.
Yeah. Good afternoon to all. I welcome you for my presentation on identification of SSR markers for fruit furnace linked QTLs in Indian melons for marker state breeding. If you see the background, muskmelon or melon, we botanically known as Cucumis melo, belongs to the family Cucurbitaceae, and it exhibits wide range of diversity due to outcrossing and cross pollination. And if you see the diversity, we can able to find for fruit traits majorly for rind color, rind pattern, and netting intensity, fruit shape, as well as fruit flesh color and fruit firmness. And here we are looking for mainly fruit firmness to improve the breeding value of the fruit firmness in muskmelon. It is a important trait. Why it is so important in muskmelon breeding program, fruit firmness? Because it's a highly complex trait governed by quantitative inheritance, and it influences the majorly on shelf life as well as consumer preference. And fruit firmness also influence on the mechanical damage during handling and transportation as well as storage. So it is very much important key trait in the melon breeding program to enhance the fruit firmness, and that's why we are majorly focusing on improving the fruit firmness in muskmelon. And now in this activity, in this research program, the major activity, major objective is to identify the markers linked to the fruit firmness. Especially we are focusing on SSR markers to dissect the trait, especially high firmness as well as soft fruit firmness. Two different contrasting traits we need to dissect with marker co-segregation, so those markers will support further fruit firmness. This is the major objectives we are looking in these activities. And in this activity, we are using materials, especially Arca series, a released variety from IHR, which is a soft flesh as well as orange flesh color. And another breeding line from IHR, that is IHR OPM1. It's a highly firm with white flesh fruit. We made a cross and developed a hybrid between these two. And these three population, especially parent 1, parent 2, and hybrid, were validated with several markers to identify the SSR marker, which is linked to fruit firmness. And in the methodology section, we have used validated 130 markers among the two parents and hybrids. Out of 130 marker, we could able to get amplification with the 10 markers. Out of 10 marker, there was a one marker we could able to find co-segregated with fruit firmness, especially high fruit firm plant as well as low fruit firmness with 180 base pair as well as 200 base pair. That marker is CMSSR 13308. Here, the 180 base pair indicates the low firmness linked to the low firmness, and 200 base pair amplification size linked to the high firmness. You can see the markers validation in parents as well as hybrids. We have amplified markers viewer. Here we have displayed here, and there is a one marker CM SSR 13308 has been showed polymorphic for parents, parent 1 and parent 2, and this was co-segregated in hybrid. In the key findings, or we have identified one polymorphic marker, that is CMSSR 13308, which is a very useful marker because it's co-segregating with high firmness trait as well as low firmness trait with amplicon size of 200 base pair as well as 180 base pair respectively. And this could be a very good marker for marker selections in the melon breeding program. In case of applications, we can this marker effectively apply in the marker selection to improve the fruit firmness as well as marker selection in the breeding populations in any segregating populations. It will accelerate the breeding program to speed up the fruit firmness improvement in case of musk. Conclusion section, in this experiment, we have identified one marker, that is CMSSR 13308. It's a highly tightly linked fruit firmness linked marker in muskmelon, which is located on the QTL FF505.1. It's on location on the chromosome number 5. It's a location located on chromosome number 5, and it is a very highly polymorphic marker, useful marker, since it's co-segregated with the fruit, different contrasting firmness of the fruits, high as well as low. and it could able to use in the marker assisted selection directly along with for the fruit rates. It cannot. It can also help to reduce the reliance of the only phenotypic screening. We can use this marker for the genotypic selection to advance the marker assisted selection to improve the fruit firmness in case of muskmelon breeding program. This is the overall study of this work, and I thank you for organizers for giving this opportunity to present the work. Thank you. If any queries, we can address.
Dr. Manjunatha, thanks. Good presentation. And
Yeah.
the session is open for discussion. One or two questions we can invite.
I think we don't have any questions, so thank you so much, Dr. Manjunatha.
Yeah, thank you, madam. Thank you.
Do we have Mrs. Poonam Preeti Pradhan online?
Miss Poonam Preeti, are you there online?
Hello.
Okay, then we can move to the next presentation.
Hello.
Okay, are you there?
Yes, ma'am.
Yeah. Okay.
Good afternoon, session.
Yeah. First is share your screen. Dr. Manjunatha, please unshare. Outside. Dr. Poonam, you have to restrict your presentation in 5 minutes.
Okay, ma'am. Am I audible? Hello.
Yeah, audible. You please start your presentation. You
Uh,
can go to slideshow mode.
My presentation is on my PhD thesis work, and that is effective solubilization of rock phosphorus organic amendments and PSB on groundnut maize cropping system. Basically, it was a 2 years work which was carried out in our college agriculture research station at Chhatabad, and the use of we had used basically focused on use of low grade rock phosphate and its effective solubilization under lateritic soils of Odisha. So if you are coming to the introduction, phosphorus is a critical element, as we all know, and here basically we had used both the organic and inorganic form so that we can
Dr. Preeti? I think she lost the connection. I think she is talking. It is coming like that. Net connection, I think, is not stable. Dr. Preeti? Are you facing any problem?
currently internet issues, I guess.
Now we could hear your voice. What I suggest you directly, you can go to the results and discussion. Yeah, you can spell out the objectives and go to results and discussion.
Here my objectives for the work that I carried out is the effective application of rock phosphate and combination, where I studied about its yield and yield attributing character. Basically, my focus was on phosphorus transformations, where I analyzed different fractions of phosphorus in soil and its availability at different stages of different growth stages of both groundnut and maize, and later on the cost effectiveness of or use of rock phosphate. Hello.
Yeah, you are audible. You please go ahead.
Thank you. These are my treatment details, where I used 100% rock phosphate and it was lone application of rock phosphate, and another was absolutely controlled. And here are different uh, combinations which I used with organic amendments and humic acid, organic amendments of vermicompost and farmyard manures. Along with that, inocular, I used phosphorus solubilizing bacteria. Then coming to the measure application, where I congregated all the organic amendments and microbial inoculums along with rock phosphate.
Dr. Preeti, we could not listen to you. Your voice is not audible. I think she got exit. She is coming again. I think your internet is not stable. She left actually. Can we go for then the last presentation?
Yes. Dr. Swarnalata Das, OUAT Bhubaneswar. Ma'am, please.
Sir, last presentation. No, sir, two are not there. Satyapriya Singh and one more presentation.
Yes.
Sir, actually, presentation of certificate. Presentation, certificate distribution like this. Deepa. Ma'am, please start. Deepa, senior, sir. You are younger to madam, sir. Okay. Yeah. Ma'am, mic you take.
Okay. Good afternoon, everybody, and welcome to sir, our Dean of Extension, sir, and to sir, and also other delegates. Myself, Dr. Sonalata Das, and my presentation is oxygen cultivation of fodder rot. Okay, all of you know that most of the presentations are concerned with the sustainable food production regarding the human beings, and no one is for, we are not bothering about the animals. So it is regarding the food security of animals, and nowadays actually animal rearing is a important livelihood for our farmers. So when the farmers are interested for animal rearing, so we have to increase the productivity of animals like milk and egg and also meat. And to improve this productivity, we have to also worry for the good quality foods. So among the different good quality foods, actually green fodder is an important fodder. Green is an important food for the animals. So among the different green fodder crops, particularly oat is very much suitable because it is digestible highly and palatable for the animals. So actually this is oat is actually a cool season crop. it prefers cool season for its high biomass for green biomass production and also seed set. But if we can cultivate it during the off season, then we can get a sustainable food for our animals. The farmers can provide some food to their animals. So in this investigation, actually, we are interested to to select some genotypes or varieties which are suitable for off-season cultivation. And for that, actually, we have screened them at the early seedling stage on the basis of their germination response, because germination and seedling vigor, these are two important criteria for selection of those genotypes. So we have studied 14 genotypes. We have studied 14 oat genotypes and germination count, and these are the observations we have recorded. These are the weather parameters. Actually, we have conducted this lab study during the month of April to June, and in which case the temperature ranged from 35 to 42 degrees Celsius. And so these are the fodder crops, and some panicles of some different genotypes, seeds of different genotypes. And nutritional quality of fodder rot under normal condition. These are the quality parameters, where is a neutral detergent fiber, acid detergent fiber, and crude protein. These are very much important parameters for animal feed. And if NDF will be more, then feed intake will be less. And if ADF will be more, then digestibility will reduce. So these two factors will be less, and crude protein will be more. And generally, in case of oat, it varies from 8 to 10% crude protein. So what happens, actually, when we conduct the experiment, we see that germination under high temperature condition and in normal condition. So in normal conditions, we see that germination response was better, and under temperature stress condition, most of the genotypes, they fail to germinate, very poor germination. But in one variety, that is UPO 24-2, which is from the Panthnagar, GB Panthnagar, it gives better result. So we search what is the reason behind it. So when we see this is the germination count in graphically presented, you see that two genotypes G2 and G3, germination totally zero, and very poor germination. And in case of GPO 24-2, germination is high as compared to the control other genotypes. In control condition, germination is good, but in temperature stress condition, only one variety shows good performance. And also regarding the SV1 seedling vigor index 1, also this genotype shows very good performance. Next, we have also studied what is the reason behind this. So we have gone for some enzyme activity studies. So alpha amylase, dehydrogenase activity, and also antioxidant activity, catalase, peroxidase activity, also superoxidase activity. All are high in case of that EPO 24-2 variety. And finally, we will conclude that this variety may perform better under stress off-season conditions. So we have also tried the correlate germination count with different enzymatic activity, and we observed the significant positive correlation. And we have identified all the traits we have accumulated, and we have found that this EPO 24-2- is the good variety. And this is my conclusion. And so we can choose this, if we can identify this variety. Actually, I have searched many papers, but I did not find regarding the off-season cultivation of oat. And during off-season, we cannot get the seed production is very much limiting. We cannot get any seed, but we can get the biomass, green biomass production, and we can provide the animals for their nutrition. And this is for sustainable food production of the animals. Thank you everybody for questions.
Thank you so much, ma'am. It's a very good presentation. And once again, thank you.
Any questions?
Any questions? One or two questions we can.
Sir.
Yes.
Sir.
Now throughout our country, the national check variety is scant. And it is, even if we have developed lot of varieties,
Yes, sir.
But Kent is taken as a national check.
Yes, yes, Kent is a
So,
national.
if feasible, please analyze
Yes, sir.
all these
This
things.
is in infant stage, and we
Yeah,
will
yeah.
try to
Yeah.
proceed again.
Because that is a national
Yes, sir.
check, right?
And many more genotypes, if we can include, then we can get a better result further.
Thank you. Thank you.
Thank you, sir.
Thank you, ma'am. Hereby we are coming to end of the session. I request the chairman, Dr. Professor Prasanajit Mishra, sir, Dean Extension Education, OUAT Bhubaneswar, to give his comments.
I request, ma'am, please, you are present the whole session. You give your remarks, then I'll conclude in 1 or 2 minutes.
Actually, the session first we started with biopesticides-based sustainable nematode management by Dr. Uma Maheshwari, and myself, Dr. Sangeeta. I have given overview of diseases infecting dragon fruits in India, and it was followed by a presentation from CSIR lab, Bhubaneswari. She has given a CRISPR-Cas-based genome editing in Solanum, and from HCL Foundation, Dr. Dibakar has given a good presentation, and after that, Dr. Bhagya from NRC Litchi, she has presented life cycle based sustainability assessment in Litchi-based integrated farming. Then Dr. Shashwat from ISWC regional station, he has given his presentation on soil hydraulic conductivity under agricultural to horticultural land use change in the Eastern Ghats. And then we could hear from Dr. Raghu, from senior scientist from ICR IAHR, on curry leaf genetic resources, exploring nutritional and genetic diversity. And followed by another senior scientist from IAHR, Dr. Manjunath Gowda. He has given presentation on identification of SSR marker for fruit firmness in melon for marker assisted breeding. Then finally, Dr. Swarnalata Das ma'am has given excellent presentation on off-season cultivation of fodder oats. And with that, we came to an end of the presentation. Though Ms. Poonam has joined online, I think her internet connection was not stable. She could not complete her presentation. Mrs. Poonam, still you are available? Informed. So with that, we are coming to the end of the session. Now I request Dr. Professor sir to give Chairman sir to give his remarks.
Thank you, Sangeeta ma'am. So, thank you for nicely conducting this session. As per the theme of this event, Sustainable Agriculture for Food Security, in this session, the complete cover of all the themes, starting from food crops, maize, groundnut, to vegetable, fruits, lastly to fodder crops. So far as crops are concerned, we have covered all the crop categories. Not only that, we are also the presenters well presented about the soil management, disease and pest management of vegetable crops, germplasm, and also up to the curry leaf. So a complete sustainable agriculture for food and nutritional security presented in this session. So I congratulate all the presenters for nice presentation, justifying to the theme allocated, and also timely completed their talk. And a special thanks to Sangeeta ma'am for strictly adhering to the time and giving equal opportunity to all the presenters. What happens normally, first few presentations took more time, and the last moment we pressurize the speakers to complete in 1 or 2 minutes. But nicely handled, all presenters are given equal opportunity in this session. Thank you once again. Thank you also the organizer for inviting me for chairing this session, and distinguished guest here, Vice Chancellor, Sri University, Professor Ved Prakash, Bhishma Pratamaha, so far as our agriculture research is concerned, Another Vishwapatama sitting beside me, Dr. Sen Das sir. You have seen in all the occasions of agrivision, Ishad Das sir is there. 3 years stalwarts so far as agriculture, research in our country is concerned. So we are fortunate to have all three in our session. So thank you once again to all. Thank you.
Thank you so much, sir. So with that, we conclude with the first day of plenary and technical session. We'll take a tea break now. We'll assemble in the ground area for the inaugural ceremony, sharp at 5. Thank you so much, everybody.